Good morning and welcome to the second day of the ABG Life Science Summit. My name is Adam Carlson. I'm an equity research analyst covering pharmaceutical and biotech companies. Our first session of the day is a fireside chat with AstraZeneca, and I'm very glad to welcome Leon Wang, EVP International and President China for AstraZeneca. Leon joined AstraZeneca in 2013, and during that time, AstraZeneca has grown to be the largest pharmaceutical company in China. Prior to joining AstraZeneca, Leon had marketing and business leadership roles at Roche as well. A warm welcome to you, Leon.
Hi everyone. Yeah, nice to meet you.
Great. I thought we'd start with a few questions on the current situation in China. China's obviously had a resurgence of the pandemic and fairly strict lockdowns, at least on a regional basis in recent months. Shanghai now, I understand, aiming to reopen on June 1st was the last I read. Despite this, on the Q1 call, that full year guidance for mid-single digit decline was maintained. So my first question is, has the picture on the ground changed at all since then, and in terms of the impact you're seeing? And was a lifting of restrictions according to sort of the timelines that we're seeing now, was that sort of factored into that maintained China outlook?
Yeah, maybe I can only talk about AstraZeneca, not the overall macroeconomic. So I think a lockdown has an impact, and the delay of VBP also have some positive impact. So I think the headwind of lockdown definitely delay our new product launch and uptake, and also diagnosis of newly diagnosed patients, and also the infusion, injectable, and hospitalized drug, and the nebulization center in the hospital basically have a limited access in around 20-30% of the places in China. It's not whole China. Shanghai is, of course, 100% lockdown, but outside of Shanghai is also not every city, but there are provinces or cities more or less impacted. So it's limited patient access, and less impact on oral oncology products, and less impact on long-term chronic disease patients like diabetes, cardiovascular, high blood pressure. So these drugs are less impacted, but more impact on injectables. But delay of the VBP also helped. So that is why we do not think this year we will change our guidance in China.
Okay. No, that's interesting. Thanks for that. And clearly, there's some uncertainty on how long these lockdowns will remain in place. But as you're saying, there are these mitigating factors. There's the delay of VBP this year. There's particular strength in Tagrisso, Farxiga, Breztri, you were saying in the Q1 call. I guess asking you maybe a difficult question here, but that sort of mid-single digit decline, is that vulnerable to lockdowns that are prolonged much further than what we're currently anticipating? Or if in a scenario where it drags on beyond sort of this summer, how should we be thinking about sort of the impact that has in relation to those favorable tailwinds that you were mentioning?
If lockdown forever scenario, so that's like the end of the world scenario. So I hope not, but I think by the end of June, more or less this lockdown should be gone. And hopefully supply chain and also patient visit to the hospital, which is our main business source, right? So should recover, right? So I think if it's only happen, mainly the lockdown in the second quarter, right? And in 20-30% of the places. So the impact is limited to 1 quarter and in 20-30% of the city, right? So should be manageable to still keep our guidance. But if the lockdown is a whole year, so I think that will be really disastrous, because the supply chain cannot sustain, right? So if it's like this kind of 100% lockdown, but I don't think the government in China is also very sensitive to the situation, to economic recovery. And I saw the recent newspaper publish quite a clear guidance. So the economy will rebound in June. So that's what the government already clearly predicted.
Okay. No, that's positive then. On the outlook, as we're saying, 2022, still expecting mid-single digit declines. 2022 potentially the toughest year, but that doesn't necessarily imply growth from 2023, I guess. And I know there's no sort of guidance for beyond 2022, but I'm curious to hear what are the sort of the main moving pieces, the puts and takes, if you will, that will sort of impact the trajectory for China next year or going forward.
I think 2022 obviously is the 2021. I think September. I think it's actually VBP already happened to Berlinta, to Crystal, which is also our large products, and also Nexium, right, in the past. And also last September is Pulmicort, right? So this is our largest product historically, so in China, so it's also impacted. So now is actually the impact of Pulmicort and all these previous VBP are actually still taking impact to the business. And while the other part, reimbursable products and non-VBP product portfolio, oncology, they're growing now, right? And the Forxiga is growing, and Reno, Roxa, Localma, they're also growing. So we're launching new products. And next year, the excitement, I think, is there are some good things in next year happening. Is Evo Shield, I think, which will be approved this year, so hopefully can benefit next year. Right now, we can expect a private market and also a provincial or city government purchase, right? So I don't know how this will be treated. And if government allows, we will also try to negotiate for an idea in the second half of the year. And also we will be launching Evo Shield in HER2, right, next year. And also the two rare disease, Coselago, Solaris, and some indication of rare disease products. I think I also have high hope on the two rare disease products, which set the footprint of our rare disease view in China. So we have high hope of rare disease business in China. So because Alexian business in the past is zero. So zero is a bad thing, right? But zero is also a good thing to start. And we also have Calquence, so which is our very important step into hematology space. So I also have very high hope, expectation on Calquence. So I think next year will be quite a good year. I mean, of course, still enjoying impact of VBP. The VBP delayed to the second half of this year will have further impact in 23. So this is a negative win. And NRDL renew happening in the late this year. We also have some price cuts impact, which you still need to grow further volume to overcome. But in China, it's not short of patience. If you can continue launching new products and digesting the impact of VBP tender loss, so that's exactly what we're doing. I think we might be one of the very few companies can still maneuver between these headwinds, right? So I think AstraZeneca will still remain #1 company in China.
Okay, well, that's very interesting. And you mentioned VBP7 being pushed back, I guess from H1 to H2. It's looking like now.
I cannot hear you. I lost your voice.
Oh
no, I cannot hear you. Now it's okay.
Okay, perfect.
Now it's okay.
Great. I was saying VBP7 being pushed back from H1 this year to H2. Do you have an idea or knowledge of sort of when in H2 it's expected? Is it late H2 or is it more towards the middle? Well, yeah, I guess that's the question.
Now it's really harder to say because the VBP technical institution is actually in Shanghai, but Shanghai has been locked down. So I think definitely has an impact on the business operation of the VBP institution. But I still believe it's a very important task for the government. They will not allow this delay too much, right? So I think late Q3 implementation is still our expectation. Maybe September, something like that.
And does it have a spillover effect on sort of the next round of VBP as well? Or do you think they'll stick to the timeline that would have been if it had happened in H1?
I think every year one VBP, they will do it anyway, sometime around that. So I don't think it will have impact on future. So it's not like a sequential delay.
Okay, alrighty.
One-off delay.
A one-off, yeah. And you were mentioning Evusheld and that expectation of a China approval at some point this year. And I was curious, given the state of the pandemic and the impact for the lockdowns and so on, is there a sort of increased urgency from the Chinese regulators, or some kind of expedited review or so on, that could sort of bring that potential approval forward in time? And then sort of secondly on Evusheld, how should we be thinking about pricing of Evusheld in China, sort of qualitatively, perhaps relative to what we're seeing roughly in the West, both on sort of government contracts and self-pay?
Yeah, I think Evusheld is the only approved prophylaxis antibody for COVID-19. So I think it also has impact on Omicron. So I think clearly it has evidence and indication which is very suitable to the current situation in China, which is to protect the vulnerable people of the whole population. So the Omicron is actually less an issue as a more milder kind of COVID-19. But the problem is the aged people and the vulnerable people, they still need some kind of protection, right, against the Omicron. So in order to reduce deaths, so that reduce mortality is still the government objective. So I think in the dialogue with government in China, they show great interest in our drug. And we are also working together to make the manufacturing in China happen. And now the drug substance is actually manufactured in China, in Wuxi Biologics, right? So in the future, we're also aiming to have finished products also if the supply is strong. And right now we get some order from Hainan pilot zone because they want to accumulate some initial evidence and experience on some patient population, and we get some small order. So I hope we can really through that thousands of cases, we can really accumulate some kind of real world experience. I think the approval will be still expected towards the end of the year. And right now, we believe there could be three kind of reimbursement. One is NRDL, national reimbursement. It's more treated like a drug rather than vaccine, right? So it's an antibody, right? So we still will get not a vaccine license, we'll get a drug license for this product. So maybe it will go through the NRDL kind of process. And also provincial and the city government can also purchase through their own financial budget. That's what we believe. If there's a outbreak of a new mutation or current mutation into the city or province, they need to protect the older people and the hematology patients, cancer patients, right? So I think that they might just want to get it, right? But we will still have a field force to promote it if we are able to launch it and try to get into an NRDL next year.
Okay, alrighty. That's interesting. And that, so NRDL and then on a provincial or city basis, and then was the third one also a self-pay?
Yes.
And how should we?
I think self-pay could be quite huge. So because patients, if we really promote to the doctor on the benefit of this drug, so I think still lots of patients will get prescribed. And we have quite a strong team in renal space and also in oncology space.
Yeah. Okay. No, interesting. And on the vaccine side of things, Vaxzevria, obviously it's sub-licensed in China, so any sales would be collaboration revenue, I guess anyway. But and you were mentioning on the Q1 call that there's work ongoing from your side and the sub-licensee side to get that booster study underway. Do you have a sense for sort of the timelines there when a potential booster approval might be realized, and sort of what is your expectations around how materials or material sales of sort of booster vaccines would be in China given the state of the vaccine market there?
Yeah, right now we're only able to export 30 million, not 30 million, some vaccine from BKT to some emerging markets, right?
Mm-hmm.
So, and I think the key now is it's a sub-license holder, so we're applying for approval actually of this technology in China.
Mm-hmm.
I think there's no clear timeline because it's really not in our hand, in our control. we are going through expert panel hearing in CDE of the government, and we try our best to become a third shots booster, and the fourth shots maybe renew, right? So at the moment, the problem maybe is another problem is patients are not really taking third shots widely in the emerging markets. I think that's also happening in China. So the government needed to make up their mind to push really third shots, right, in the travel passport and the travel limitation, right? So right now, I think 2 shots are being promoted. So that is something I think should be coming.
Okay, no, interesting. And I guess that feeds into potentially.
I have more expectation, more hope on EvoShield because it's a clearly more differentiated offering, which is a prophylaxis treatment antibody. So, and nobody else can offer this. Yeah.
No, that's interesting. Thank you. Perhaps a question on Farxiga, which has obviously been strong in emerging markets overall and in China in particular. Ruud was saying on the Q1 call, market shares in CKD sort of 70-90%, and outside the US and in heart failure, sort of the majority of the market anyway. So I was curious to hear sort of your estimates of the market share for Farxiga in heart failure, at least, where you launched last year. Is it sort of broadly in line with what Ruud was referencing globally? And then secondly, it was mentioned in the Q1 call as well, Farxiga having benefited from increased screening in China. So my second question then, how underdiagnosed are the conditions for Farxiga? So I guess diabetes, CKD, heart failure, perhaps relative to the US or Europe. And I guess what does that imply in terms of further tailwinds for Farxiga or screening of those conditions?
I might not have the details of how the heart failure penetration, but I see every market launching quite aggressively into CKD patients. And now I think starting to get into heart failure outside the US. I think we're starting now. And I think these two are great opportunity, not only helping us in heart failure in CKD, but also strengthen our position in diabetes space, which is we still have room to grow. So lots in the emerging market are still underdiagnosed in diabetes, high glucose, rather than just the CKD and heart failure. So if you ask me to sequence, is even diabetes you have space to go up, and CKD definitely. Heart failure is seriously underdiagnosed.
Okay. No, that's interesting. Another question then on perhaps emerging markets ex-China. I think some people were probably a bit surprised by that very strong growth outside of China in the quarter. And you said in the Q1 call, at least in part driven by new product launches in oncology. So I was wondering whether you could elaborate a bit on what those launches were, and whether there are many more sort of material launches in key emerging markets coming up for some of those key oncology products.
Yeah, I think you see clearly we almost doubled our business outside the China emerging market, but largely owing to the vaccine in the Q1, and we're starting to see some EvoShield business. Right? So I hope vaccine will be a bit less in the next few quarters, but EvoShield will definitely grow. So I think this is one aspect. And the second one is rare disease. I think rare disease is a very important growth engine even for the emerging market, because the base is small, but it's also quite a significant growth. So outside the US emerging market, so it's quite important growth engine for us. And lots of emerging market rare disease policy is actually quite friendly. So I think we have chance to grow. And certainly, and the base business, of course, is oncology, launching new indication, new products in oncology. Right now, is still Lynparza new indication, Tagrisso new indication still, right? First line. We're merely getting to first line treatment across the emerging market. So, and now starting to get into Adura patients for Tagrisso. Tagrisso remain the most important oncology launches. And then Lynparza, followed by Lynparza, and then Imfinzi. And now we also see markets launching Enhertu in a very successful way. And also Calquence, because lots of emerging markets are young people. So age is relatively young. And breast cancer and hematology patients are actually quite important indication, and not just lung cancer. So I think with Calquence and Enhertu is really ideal. So I think with these 5 opportunities, Enhertu has many indications, and Calquence also has 2-3 indications. So I think when you roll out in the emerging market, if we can make accessibility less an issue or burden, we can definitely accept more patients in the region.
Okay, no
and beyond oncology, I think Synergis and Fasenra, the biologics also starting now, right? So and also this anifrolumab. So I think these are also very important future. Of course, the Forxiga is still doing very well, very important. And also emerging market products actually outside China. China has a VBP. Outside China is actually even off patent. You don't see the kind of patent cliff, right? So so still I see many markets, Pulmicort, Crestor, and even Silicon are growing very nicely. So I think emerging market is a very different rhythm, kind of a sustained release of business, right? So I think it's quite promising. I think this kind of strong momentum should continue. And because a lot of untreated patients in these markets, unaffordable, undiagnosed, untreated. So it's many improperly treated and not diagnosed, and even diagnosed, not affordable people around the corner.
Yeah. Okay, no, interesting. And I'll pick you up on this thing you were saying, but rare diseases also being sort of a growth engine going forward, an increasingly important part of Astra's business, of course. But in terms of how access and reimbursement is looking in China, and I guess in the wider emerging market space, is there a push or a will from, I guess, maybe starting with the Chinese authorities to improve access to rare diseases there? And are you seeing any sort of tangible progress there in terms of regulatory processes, pricing, reimbursements?
Yeah, I think China government is issuing guidance and also some policy in draft to collect opinion on rare disease, and it's clearly saying that rare disease is their priority on the government agenda in this 2-session meeting this year, and also private insurance are coming into the game, and lots of provinces and cities they have a surplus in budget, and they're supporting rare disease now, right? And I think the government is a real intention to do it, but I think we also needed to think to leverage charity and leverage private pay, and also we should, as a company, we also need to show gesture to help patients, right? So at the end, it's a 1/3 government, let's say 1/4 government, 1/4 charity, 1/4 company, and 1/4 individual, right? So to make a large majority of people affordable to the rare disease treatment, to make it work.
And more broadly, the self-pay market in China, how big is it relative to the government funded, and sort of what proportion of Astra sales comes from self-pay? Do you have that number?
Yeah, I think right now in the whole, you can definitely see from the NRDL budget, right? So private insurance and individual payment. And I think if you put these three things together, private insurance is less 10%, less than 10%, right? So, and still large majority is NRDL, which is the national reimbursement, and 30% is out of pocket, right? So you still have a quite significant self-pay. So if you self-pay co-payment is 30% usually, right? And if you put outside NRDL, maybe you still find another 20-30%, totally out of your pocket. You don't even co-pay, right? So so if you put together, at the end is like 45, 10, right? So so private insurance 10 and 45% the government, and 45% is out of the individual pocket.
Okay, no, interesting. We're nearing the end, but maybe just one or two last questions. Perhaps one on approvals or regulatory timelines and so on. I think you were saying if it was the Q1 or the Q4 call about that lag time between approvals in the US and the EU and what we're seeing in China, oftentimes sort of a year or more, and that sort of aim to bring those timelines more in line. And I think Mark was saying for rare diseases, specifically in the Q1 call, that there is a move to include China in more of the global studies as well to sort of harmonize those approval timelines. So I guess is that something that you're hopeful will result in sort of approvals being more in line with the US and Europe in terms of the timing of them in China going forward, or will we always have this sort of bit of a lag before you access the Chinese markets?
I think I think my dream, my expectation is that we launch ahead of US, right? So rather than waiting, so we already have products launching ahead of US, like Roxa, right, and like the CMET drug, right? So we would co-develop with Fibrogen and Hutchison in China. So in the future, more and more China innovative products will be originated from China and launched first in China. So in the future, you will see part of our portfolio will be launched in China first and gradually roll out globally. So this is one part. And right now, global portfolio, we are able to participate in 90% of global studies. So 9 out of 10 studies we are either joining with a tail. So I think that resulted in, hopefully, I mean, we are not that far from European approval, right? Or Japan approval, right? So more or less same time in the future. So that is definitely our goal, right? So and I think that is achievable. That is achievable. And the global headquarters engage China earlier and earlier, right? So and China becoming more important. So if there's a company China is important, it's AstraZeneca, right? So so I think we're gaining the kind of attention and the priority within the internally.
Okay, no, interesting. Maybe just one last one then before we have to wrap up. We're seeing increased competition from innovative Chinese companies trying to bring drugs onto the US market, often through partnerships. And I guess the most relevant example for Astra was EQRx and Omalotinib. The FDA seems to have taken a little bit of a different stance when it comes to China-only studies. We saw the Sintilimab ODAC outcome. So my question is sort of how should we be thinking about that increasing threat from sort of Chinese innovated drugs accessing the sort of global markets? What's your thinking around that?
I think we shouldn't feel it like a threat, regardless of whether this drug is from Europe or from Japan, right? So I think Europe, Japan are also launching lots of new products into the US with a global standard quality studies, right? I don't think China is exception, right? So I think if we are on the same line, so I think it's a very friendly competition, right? So whichever drug is better, right, in the outcome, right? So if you launch a diabetes drug, you need outcome study. If you launch oncology products, you need not only PFS, DFS, and OS, right? So if you are satisfying the needs of approval, I don't see this as a threat because it is quite costly to develop a global standard drug to be open, right? So especially in large indication, in large competitive indication like lung cancer, right? So a standard of care is very hard to beat, right? So you have to have a clear comparator and a benchmark, right? So I don't see that as a problem. And especially for AstraZeneca, we are quite deeply rooted in China as a more localized multinational company. And we might be the company benefit from China innovation, right? So we already, right? So Roxa we launched even ahead of US and Japan, right? And the breastfeed we also, our own products also launch ahead, right? So, and Hutchison's product CMET also launch ahead. So at the end, we have a CICC AstraZeneca joint venture capital fund, right? So we're jointly investing into China innovative companies. Ultimately, want to help these innovative products to achieve global standard with our network and become AstraZeneca products globally outside China.
Okay, no, interesting. Perfect. I think we're at time. Thank you so much for a very interesting conversation, Leon.
Thank you. Thank you for all the good questions. Thank you.
Great.
Bye-bye.
Bye now. Welcome back. The next company to present is Synact, and we're joined by their chairman, Torbjörn Bjerke. Synact is a Swedish-listed company in sort of mid-clinical stage development with its lead asset, AP1189, for rheumatoid arthritis. I'll hand over to Torbjörn to tell you a bit more about it.
Thank you very much, Adam. And good morning, everybody. It's a great pleasure to be able to talk about Synact Pharma for you. So as quite a few of you probably know already, Synact Pharma is all about developing the new treatment for inflammatory diseases, and we hope to be able to help quite a lot of patients within that space, which is really, really big and with big medical need. So what I want to do today is really to go through, focus on 1189 and go through where we are, focusing on rheumatoid arthritis, and I'll explain why this is our focus, but also talk about the next clinical trials as well as the milestones coming up. So if we look at Synact Pharma, we are at the moment listed on Spotlight. We're working, and hopefully we will be at the Nasdaq main market in Q2 this year, which gives us many more opportunities to do investor relations and hopefully attract institutional and specialist investors into the stock. We believe that we have a very exciting development program in the company, and as you can see on this slide here, there are quite a lot of different milestones coming up also in 2022. We had a fabulous 2021 with a lot of different milestones, giving the basis now for further development of 1189 in rheumatoid arthritis and other inflammatory diseases. I also want to point out that we just raised 150 million Swedish kronor, very much helped by ABG and Kempen, so thank you very much for that. That gives us a runway to the end of 2023 and gives us the funding to do the next clinical trials. This is the pipeline, and as we all know, this is really the value of any companies in drug development. And as you can see on the top ones here is rheumatoid arthritis. We go for both first-line treatment as well as second-line, or those patients that have not responded well enough to the disease-modifying agents out there today. We believe that this the 1189 could really help here as well. And why rheumatoid arthritis? Yes, obviously because it's about 1% of the general population in the world. The market is about $30 billion, and there's a big medical need. As you also can see here, we also have a more orphan drug focused with the nephrotic syndrome, and we also have virus and respiratory insufficiency, as we came out last year with positive data in COVID-19 induced ARDS. In addition to that, we do have a discovery program with new compounds coming up as second compounds to 1189. So what is the mode of action here? And this is quite important in order to put it into perspectives to the treatment of rheumatoid arthritis. We believe that 1189 is a compound that can correct dysregulated inflammation without inducing immunosuppression. Now, that's quite important. We don't want immunosuppression. We want the immune system to rebalance itself. And this is exactly what we believe 1189 does. So on the cartoon here, you can see both the excess inflammation. When you get a inflammation in your body, you get a lot of inflammatory cells into the site, and it's important to decrease the pressure on the immune system and on the tissue itself. So we do that with 1189, and then we clear up the inflammation as well. So we get away all the debris and that kind of stuff. So this is the dual action of 1189, which we believe is very important and could play a major role in further treatments of these patients. When you do clinical development, you need to show how this compound is taken up by your body, by your gastrointestinal tract. And this slide shows you that we have fantastic, good DMPK, meaning that you can get a good curves in terms of different doses, and importantly also, as you can see on the right side of the slide, you can get 24 hours coverage. So once daily it's a tablet formulation. We have worked quite a bit on that. So this is really the good thing. We often kind of compare us to other compounds within this sector, and we believe that 1189 can circumvent quite a lot of the obstacles in the current treatment. This slide shows you the tablet formulation. When you go from a suspension that we did in the beginning to a tablet formulation for further clinical trials, it's important that they have the same curve and they are being taken up by your gastrointestinal tract at exactly the same manner and with the same curve. And as you can see here, absolutely, that was a very important finding in 21, and further finding, we came out with a press release not that long ago, also restating the importance of this tablet formulation. In addition to that, in 2021, we did the tox studies preparing for the 12-week study that we are initiating now. This slide is just to show you that this is not the first time we show proof of concept in severe inflammation. These results are from the COVID-19 trials I talked about. We did that in Brazil, came out very positive with patients coming back from the hospital to their home much quicker than with normal state-of-the-art treatment for these patients. However, due to the fact that COVID-19, thanks God for that, and the biotech sector, that we now have going from a pandemic to a more slower stage of this virus. So the business case for 1189 in COVID-19, we believe is not right there right now. But we learned a lot from these results, and we are going for influenza, which also kills quite a lot of patients every year. So you will hear more about that. So let's talk a little bit about the BEGIN study. The BEGIN study is a study where we took newly diagnosed rheumatoid arthritis patients. We took a patient population, more severe patients, and patients that were also treated with methotrexate. Methotrexate is a disease-modifying agent, which is often the first line of treatment, however, linked to quite severe side effects. So importantly, we got positive results and significant positive results on the C-line score. C-line score and ACR20, as I'll show you later, and DAS28 are different ways of measuring the disease activity of rheumatoid arthritis, and it's important that you get significance here. And as you can see from this slide, in 100 mg, we tested 100 mg, we tested 50 mg, and we tested placebo. So placebo, remember, is with methotrexate plus the placebo. The 50 is 50 mg of 1189 plus methotrexate, and the 100, of course, 100 mg 1189 and methotrexate. And we saw clinical significant result already after 4 weeks of treatment, because BEGIN study is 4 weeks of treatment. So it's the initiation of the treatment of a more chronic disease. This study, or this slide here, shows you the ACR20, 50 and 70. ACR20 is like again, a composite of different measures on disease activity. And as you can see here, in 100 mg of 1189, you can see a significant 60.6% efficacy, meaning that 60.6% of the patients got a 20% relief of the symptoms. Again, in terms of the DAS28, it's also another way of measuring disease activity. We saw a nice dose response, and this is also quite important. So all three ways of measuring disease activity came out quite positive. Another element of the rheumatoid arthritis that's not much talked about is really the fatigue. These patients get really fatigued, and we measured that as well in the study. And as you can see here, after 4 weeks of treatment, 100 mg per day came out with a nice dose response here. So the conclusion so far is really that this shows really, really promising positive data after 4 weeks of treatment only. Safety is important, and just a take-home message from this slide is that the safety profile we saw was really benign. We didn't see any severe safety incidents here, and that is really promising for the next courses of treatment for the 12 weeks trials. So begin study, summary, already said it, good initial clinical data, really promising for the next clinical trials, good safety profile. We believe that if we can continue to show this efficacy and safety profile, and maybe even better efficacy, then it could become a important medicine for rheumatoid arthritis patients, and hopefully also for other inflammatory diseases. So let's talk a little bit about the positioning and the next development steps. So this is quite a busy slide. However, the take-home message here is that the treatment options in rheumatoid arthritis today is quite a few. There are disease-modifying drugs like methotrexate. There are also biologics like antibodies to treat the single aspects of the disease. However, they are all linked to severe side effects, and including the JAKs that are quite efficacious, however, linked to severe side effects and do have black box warnings in the US, which hampers the introduction into the market and the treatment there. So the emerging profile of AP1189 is once daily oral dosing, tick, that's great. Quick onset of action is also very important. We saw that in the COVID-19 study, and we saw that also in the BEGIN study. So it is obviously important that it has a quick onset to action. Efficacy approaching JAKs, which are the best treatment in terms of efficacy today, absolutely. However, we want much better safety profile. So safe and well tolerated. Steroid-free mode of action is also important, and compatibility with methotrexate for potential combination treatment. That was exactly what we evaluated also in the BEGIN. So when we look at the 1189 compared to JAKs today, after 4 weeks of treatment, we can see that it has the same efficacy, but much better safety profile. Now, during the 11 or 12 weeks of treatment, we will see how far up we can get in the efficacy and how the safety profile is. This is extremely important for us. We have good vibes on that, looking at the profile today. So comparable to JAKs. We have done a couple of market research in the US and in Europe. after the data came out, and high prescribing rheumatologists in the US, for example, are really keen on 1189, the profile of 1189, and they predict that about 45% of the DMARC patients, those that do have disease-modifying agent, for example, with methotrexate, that does not really work anymore, then 1189 can come in, and that's the biggest market anyway. So absolutely very promising on that. So novel mode of action, oral route, and clean safety profile is important. So let's talk a little bit about the further development in RA. The BEGIN study will go into an EXPAND study. We need to see how well, in terms of efficacy, can we get after 12 weeks of treatment. And we have chosen the 100 mg because that was the best dose in the BEGIN study, and we'll compare that to the placebo that is methotrexate. So it's very much like the first BEGIN study. However, here we look at 12 weeks. We'll also look more upon the mode of action. How do we interact? How do we help these patients in terms of their inflammation, and get more data on that as well? So really exciting. We'll have ACR20 as a primary readout now. As you remember, that was significant in the BEGIN study, and that is really what FDA wants to see. The next study we'll start is the DMARC IR, where we go in on these patients that do not respond anymore to the disease-modifying agent, the first one they got. And then we'll add on different doses of 1189 with the tablet, of course, and see where we go before we go into a full 12-week study. So this is divided into two, with ACR20 as the primary readout here as well. Important milestones in 22 and 23. As I said, 21 was a fantastic year for SynAct Pharma with a lot of different milestones. Now we have the funding in order to do the next clinical trials in this very challenging market from a financing point of view. We did the right issue. We are talking to the FDA, and we'll have feedback on the clinical trial that will also be going on in the US. That's important because we're looking at the global market. And we'll have a redesign of the nephrology syndrome. Thomas is thinking about that now, and we'll have a discussion on that, and then we'll come out with what we believe is the right way of going up. NASDAQ uplift is important in order to capture even more investors. We are really happy with all the shareholders we have today, but we would also like to attract more, and then the NASDAQ main market is important base for doing that. And as you can see here, quite a few different milestones until fourth quarter 23. So highlights: we continue to develop the 1189 in rheumatoid arthritis and other indications. We believe we have 2 proof of concept studies in 2 different indications, which is quite important when you look at drug development and the likelihood for success in the next studies. We know quite a bit on the mode of actions. We would like to know even more, and that's important. We'll also have that answer during the next trials, and we're looking forward to a eventful 2022 and 23. So thank you very much.
Great. Thank you very much for that presentation, Torbjörn. We have time for a few questions. I was wondering if you could just help us understand sort of the rationale of proceeding with both the first-line RA study and the DMARD IR study. Presumably, given methotrexate's sort of very firm and position in the first line, it's a cheap drug, is it more likely that it would be a sort of a DMARD IR setting for 1189? Yeah, but you're doing 2 studies.
Adam, really good question. So our way of thinking there is that now we showed really good results in after 4 weeks of treatment in these begin patients. Why are we then expanding it to the expand and measuring 12 weeks in the first line setting? They are twofold. We are really dying to see results after 12 weeks because we believe, and this is our belief, that we can get even better efficacy after 12 weeks in these patients, first line. And then we also learn more about the combination with methotrexate after 12 weeks. And then, you know, when you look at the safety profile so far on 1189, it could maybe fit into a tool for first-line treatment as well, maybe together with methotrexate. And that's quite a substantial market. The JAKs cannot do that today because of the safety bad safety profile. And we don't have much tools for these patients. So that's the rationale for the first one. And for the second, the DMARC, you know, when this is the big market. So we believe that 1189 with the existing safety profile and efficacy could play an important role as a tool for doctors and patients when they have failed in the first disease-modifying agent. So, and we want to do that in parallel, not to lose time. So, so that's the rationale.
Yeah, yeah. Okay, no, gotcha. With this expansion into 12 weeks, 3 months of treatment, help us think: is there a sort of a useful proxy that we can look at in terms of what we would hope to see in terms of an efficacy add-on from those extra 2 months? I mean, you can obviously look at JAK inhibitors and what they do at 1 month versus 3 months, but then there is this difference in mechanism of action, which could give you better, you know, presumably, potentially. What's your thinking in terms of what you're using to try to gauge?
Yeah, Adam, I myself, and I think the whole Synact Pharma team would very much like to see efficacy comparable to the JAKs, which, as I said, are the best compounds today in that setting. However, with severe side effects. So if we can compare it to JAKs in that range, I think that would be marvelous, and that would be a home run. And then you have all the different, you know, percentages in between. But that's the aim. That's the aim. And when we look into the begin results, we get a clear feeling that we haven't seen the max efficacy yet. Because remembering that the mode of action here is really to balance the immune system. Right. Yeah. And it takes time because these are very severe patients. So that's why we need to rebalance the immune system. We saw a significant efficacy after 4 weeks, but you know, it's a good background for believing that we can get even better efficacy. And with the JAKs, you know, they get up to about, in ACR 20, get up to about 70 to 80%. We saw more than 60% in after 4 weeks. So I believe in order to be competitive here, we need to get into that range, but with better safety profile.
Yeah, yeah, okay. And maybe just one final question then. The RA studies, do you know where in the world those studies are running? What countries will you be recruiting from?
Yes. So if we look at the EXPAND study, we are anticipating it to be where we, or especially in Bulgaria and Moldova. Now Moldova is on the risk, so we have backup plans for Moldova, unfortunately. However, at the moment it looks really nice to start, and they are really good. They are educating their patients quite well, and we saw a only a very small placebo effect in those countries. So we are continuing on that. On the DMARC IR, it's also including US sites. And that's why we are excited to talk to FDA. That's in the process now. We'll come to the market with the feedback from the FDA when that is, when we have that, and then start that. So this will be Europe as well as the US. So we're getting into a global development path.
Yep.
For a company like Synact Pharma. That's a very positive challenge.
Sure. A good stepping stone. Great. I think we're at time. Thank you very much for the presentation and the Q&A, Torbjörn.
Thank you, Adam. Thank you.
Welcome back. The next company to present is Xbrane, and I'm pleased to be joined by their CEO, Martin Åmark. Xbrane is a Swedishly listed biosimilar company moving into commercial phase, you can say. I'll hand over to Martin to tell you more about it.
Thank you very much. Yes, indeed. We are a developer of biosimilars, and starting off with just a snapshot of who we are and where we are. Xlucane is our lead product candidate. It's a biosimilar candidate to Lucentis, ophthalmology drug that sells for some 3 billion euro annually. We are in the registration phase currently. We submitted the application to EMA in Europe in September last year, and recently to the FDA. And we have partners that are going to commercialize the product, if approved, of course. STADA in Europe, and then Bausch + Lomb across North America. So we're excited about that lead candidate moving towards approval and potential launch later this year and beginning of next year. Then we have a preclinical portfolio that targets really 50 billion euro of annual sales of the respective originators, if we look at 2026 estimations. And the next upcoming one we call Ximsane is a biosimilar candidate to Cimzia. It's partnered up with Biogen, and it's kind of going through the preclinical development and looking forward to hopefully being able to initiate clinical trials during next year. Then we have a portfolio of oncology biosimilar candidates, biosimilar candidates to Opdivo, Keytruda, and Darzalex respectively. And we have our business model really rests on trying to out-license our product candidates at the preclinical stage, and that is what we've done with our 2 first product candidates. And we have an ambition to really become one of the leading players in the world when it comes to developing biosimilars. We have a patented platform technology which gives us higher productivity, we believe, compared to competition, and thereby lower production costs. We have now a team of more than 65 professionals, and really all the capabilities and competences required to take a product all the way from cell line development to registration. And we have state-of-the-art development facilities out in Campus Solna here in Stockholm. And we will now have the ambition to initiate 1 new development program annually, and still on the back of income generation from our leading product candidate Xlucane, become cash flow positive towards the end of 2023. Okay, so I'll go a little bit first into talking more about biosimilars and the biosimilar market, then going into Xlucane, and then talking about the preclinical portfolio. So what is a biosimilar, just to clear that concept? You have a biological drug which consists of a protein, a large molecule as active substance. It is produced via so-called recombinant DNA technology, so you introduce a gene into a living host cell that then instructs the cell to express or produce the target protein. Now, a biosimilar is a highly similar product to that originator biological drug, but since it is such large molecule and complex molecule, you cannot demonstrate that you have an exact identical copy, but you need to be talking about high similarity. And of course, as the concept of generics and small molecules, you can launch the biosimilar post-patent expiration of the originator biological drugs, and they're typically at the price discount. We like this segment. It really allows us to develop drugs faster, at lower investment, at lower risk compared to novel drug development. It takes us 6 to 7 years to develop a drug, 100 million euro roughly, speaking in investment. But the most important thing is, by the time we go into the required phase 3, historically speaking, 95% of the programs going into phase 3 have ended up with an approval. So the risk is much lower when the majority of the capital required is deployed. And biosimilars are addressing typically big markets. I mean, many of these biological drugs are blockbusters with several billion euros of annual sales. And what we've seen now recently is very quick uptake. So you can look here on this page in Europe and US respectively. The volume share of the biosimilars combined over time from launch versus the originator product. And 40% market share, volume market share, 12 months after launch has been achieved recently by the biosimilar. So that's rather extraordinary, I would say. And on average, 2 to 3 biosimilars per originator product, and then price discounts of 20 to 40%. So that's important to remember. This is different to small molecule generics, where you can have 10, 15 generics coming into market post-patent expiration and prices thereby going down 90%. This is different, higher barriers to entry, and thereby fewer competitors and lower price discount versus originator. We believe we have an edge here in our patented platform technology, which gives higher productivity and thereby lower cost compared to what we believe competing systems. And we have a growing IP portfolio around this technology, 10 approved patents and 15 pending. And I think that our platform technology and our capabilities as a team has been validated by partnerships. We have Stada, Bausch + Lomb, and Biogen as partners with over 150 million euro of paid and committed license fee and cost sharings for our respective programs. And if I look at then our portfolio, it's targeting really in total 53 billion euro of peak annual sales of the respective originator products. So I think it's a very big market we are addressing with our portfolio. So I'll go more into depth on Xlucane first, and then the preclinical portfolio. So Xlucane is then a biosimilar candidate to the eye drug Lucentis. It's used in treatment of mainly age-related macular degeneration. So it's a disease affecting elderly, leading to deteriorating vision and in worst case, blindness. And it's a big disease, roughly 18 million eyes affected globally, and a big portion actually going untreated due to high costs of the originator products in combination with restrictions and caps on reimbursement, which I think is something we like to see when we develop a biosimilar candidate, because that tells us that we can do something meaningful. There's a need for a more cost-efficient product to come to market, and we can address an unmet medical need in that situation. So where are we? We did a large phase 3 trial comparing efficacy and safety to the originator product, which now has been concluded. We submitted the file to EMA, and if all goes according to plan, we shall get approval second part of this year. We recently submitted the file to FDA, and we're going through the acceptance period. So if all goes well in the acceptance period, FDA shall be able to file the application by end of May. But we'll be able to get back on that one during the coming weeks, of course. So that's the process, and I do believe that we have a strong package. We have comparative analytical assessment, which is really composed of a panel of over 20 analytical methods, and then what you have to do in this context is to produce 10 or more batches of your biosimilar candidate and procure on the market up to 20 batches of the originator product, and then compare the ones versus the other. And I think we have a high degree of similarity if you look at the main attributes that you're looking at: amino acid sequence, structure, binding properties, potency, and purity. I mean, of main importance, of course, binding properties and potency, where you actually control so that your biosimilar candidate binds into what it shall bind into in the human body, compared to the originator. In this case, it's a growth factor called VEGF-alpha, and we've demonstrated that Xlucane binds in a highly similar manner to that growth factor compared to Lucentis. And also in the phase 3 trial, we demonstrated equivalent efficacy. The study met the primary endpoint, and also comparable safety to the originator, and similar product characteristics really to the originator. And big market, 11.7 billion euro really of market, roughly 3 billion composed of Lucentis sales, and the rest is really Eylea, which is a similar competing product. And I think really we are addressing this whole market of 11.7 billion, since Eylea and Lucentis really are used in similar fashions with regards to number of doses per year in a real clinical setting, despite their label being different, with Lucentis having a 4th-week and Eylea an 8-week injection frequency label. Pricing is similar, efficacy and safety similar. So really I do believe that although in our base case, when we believe we shall reach to 100 million euro of net income generation year 3, we look only at the Lucentis sales today. I do believe that the sales of Eylea and the market of Eylea is composing an additional upside for us. And I think we're 2 great partners that are going to commercialize the product. Bausch + Lomb, that is an eye product specialist really across North America, and really the only company that are going to have a Lucentis biosimilar on the market in North America that has a sales force already targeting these retina clinics, and they have relationships to the retina clinics and a very strong brand name in that setting. And then STADA are going to commercialize across Europe, select countries across Asia and Middle East, generic and biosimilar specialist with a handful of biosimilars on the market already, so they know well how to sell and market biosimilars. So that's Xlucane. We're excited, of course, to see that product develop during the coming year, and looking forward to finally being able to bring that product to market. So if we look ahead here and kind of take a step back, really what we do here in building Xbrane in the longer term perspective is to fulfilling a purpose of addressing the issue currently present with low accessibility, globally speaking, of biological drugs. So biological drugs have really revolutionized the treatment of several severe diseases, including in the oncology setting, eye diseases, as I talked about, but there is a limited accessibility, and there's in the US, 2% of American population is being treated by biologics, but still it accounts for 40% of the cost of the prescription drugs. So it's a massive problem, of course, both from a kind of a pharmaceutical spending perspective, but also an issue with accessibility, and that's what we are trying to address with Xbrain. So the purpose is really to bring about a change and enable healthy quality, globally speaking, and bring people back to healthy lives through then developing affordable biologics. And I think we now have an R&D engine, so to say, with the platform technology giving us lower production cost, strong team in place, and our facility where we have the equipment needed to develop biosimilars. And then looking at the preclinical portfolio, Xcimzane is then a biosimilar candidate to Cimzia, and Cimzia is a differentiated TNF-alpha inhibitor used in treatment of mainly rheumatoid arthritis and psoriasis. 2 billion euro of annual sales, roughly speaking, so it's still a sizable product. And what is interesting here is that we have the only program globally with a biosimilar candidate to Cimzia. So it's a unique situation where I do expect that Xcimzane will come alone as a biosimilar on a 2 billion euro product. And the reason, really, I do believe, is related to our platform technology, because this is a setting where productivity and cost is crucial. And we set out the criteria when we started this program that we need to be able to get to 80% gross margin with a 50% price reduction versus originator, and that you can calculate backwards what it means in terms of productivity in your expression system. And we had that as a go, no go in our program, and we were able to reach that, but it was very challenging, and it was thanks to our patented platform technology. And I do believe that others have tried but struggled and therefore dropped the program, and I believe that this is the reason why we're alone here. And that was, of course, crucial for us to be able to strike a deal in such an early phase with this program with Biogen. And I think this is a great deal, and something we would like to replicate across our portfolio. So it's an out-license deal of the global rights to Xcimzane, and we're going to be responsible for the preclinical development, after which Biogen takes the responsibility and also funds the clinical development of this product. And we got $8 million of an upfront payment, and additionally $80 million as milestones throughout development, and then royalties on net sales. So I think if you look at this from a return on investment perspective, it's extraordinary. So I think it came very handy for us, and if we really shall focus on what we believe we're best at, which is the preclinical development and applying our superior platform technology, then doing deals like this, that's the way to get maximal return on what we are best at, our real core competitive advantage. So that's what we want to continue to do if we look ahead on our further preclinical portfolio. We're then looking at a portfolio of oncology biosimilar candidates to Opdivo, Keytruda, and Darzalex respectively. And it's a huge market, I would say. 48 billion euro of expected peak sales of these three products combined in 2026. It's a huge market, and we have a huge issue here as well with high costs on the respective products, which leads to an issue with accessibility and even the concept of financial toxicity. So persons surviving over cancer, but then struggling with the the debt that they accumulated during the time of the treatment, leading to, in the worst cases, of personal bankruptcy. So these are the situations which is a reality, particularly in the US. And here it's really a strong case for us to try to do something meaningful for this patient population. Okay, so stopping up here where I started, we're excited, of course, about Xlucane and the prospects for that product long term, with reaching this 100 million euro annual net income generation year 3. And it's a very exciting year, of course, with the regulatory processes, which of course we need to navigate through successfully, and then looking forward towards a launch of the product. And then I think we have a great preclinical portfolio. I think the Xcimzane program is very compelling, with it being the only biosimilar candidate to Cimzia, and also with such a partner like Biogen. And then the oncology portfolio with really huge market we are addressing there. So I think we have a very good setup, and we're standing a very, very exciting time now in our development. So thanks for listening.
Perfect. Thank you very much for the presentation, Martin. A few questions then. You mentioned this thing about focusing on what you do best, and I guess just a question then on what that suggests about the potential to sort of expand into other growth verticals, manufacturing or commercialization. Is the preference then to sort of stay with what you do best, where you get the most bang for the buck, or how should we think about those potential growth verticals?
Yeah, no, I mean, these are questions that we have asked ourselves and continue to ask ourselves. As a matter of fact, we're going through a strategy process annually. We're going through that now as well, and we have those questions on the table. But I feel that our core competence really lies in clinical development and our platform technology. We can do all the other steps in the development, clinic and registration. In fact, we've done that and undertaken that responsibility for Xlucane. But I really do believe that the company should focus on what it's best at, and then I think that's what we should do. And I think we've demonstrated now that we can do early stage deals. We've done it with both our 2 leading programs, so that's fully possible. And I rather do such deals and get the lower royalty than having to invest the whole programs through with a higher royalty or profit share, of course. But I'd rather do the buy-in like this and broadening the portfolio, because there's plentiful of opportunities. So the best thing we probably can do is just to address all those opportunities out there with regards to biologics going off patent now into 2030 and onwards, focusing on the preclinical development.
Okay, no, interesting. And a question on, or 2 parts there, maybe a first one, this concept of interchangeability, and the approval of the first interchangeable biosimilars. What is the sort of the significance of that to Xbrane? And then a second sort of theme, this thing of indications from the UK at least, of sort of opening up for approvals based on solid preclinical and phase 1 data, and omitting a full-on phase 3 study. Do you see that trend, I guess? And then what are the sort of implications for you if that gets adopted more wider by the FDA and EMA as well?
Yeah, no, I mean, both those 2 trends you're mentioning will happen. I'm certain over time. How long time it will take is hard to tell. There's already guidance with regards to interchangeability from the FDA, and in Europe it's a country-by-country decision, but I'm sure that will come more and more. And as you said, there's a guidance from MHRA in the UK to omit the phase 3, and I think that EMA will follow, and eventually also the FDA. So what will it mean? It will mean more competition and lower prices as a consequence. So it's the generalization of this market. I mean, that is what I believe is going to happen over time. But I think what's good here is that the only thing it will mean is that production costs will matter more.
Yeah, right.
And there is an opportunity here to differentiate on production cost, because you can have a more productive system with regards to the yield you're getting out in the expression system than competition. When you talk about small molecules, that's probably not possible. There, everybody will be at the same production cost. But here you can differentiate in production cost, and we do that, and we believe we're best in that. So I do believe that the only thing it will mean is that our competitive advantage will only be more important going forward. And we need to continue to invest behind that to be better and better and better, and really be the best on cost going forward. And of course, also what it will mean for us, and what we talk a lot about, is we need to lead the industry in omitting the phase trees. And that means that we need to focus on analytics, because omitting the phase tree will rely on a very solid comparative analytical package. So we invested a lot out in Solna in analytical equipment and having the best people to run those machines, to be able to achieve what you're talking about as fingerprint biosimilarity. So a really, really similar analytical profile.
Okay, no, interesting. And this dynamic of that competitive edge that you have now in terms of that higher yield and better margins, with the cell lines that you have been working with and the ones that you're sort of, as I understand it, moving towards in terms of mammalian cell lines, does that impact that competitive edge that you have in terms of higher yields? Do you need to sort of rediscover that edge, or are you bringing that sort of advantage with you into these new cell lines that you're using?
We have historically the strongest edge in E. coli. That's true. But we're moving now into CHO cells. That's the host cell we're using for the oncology programs. And we're trying to take the concept and philosophy of our LEMO technology in the E. coli world to CHO cells. And mainly what we're doing now is to experiment with the vector, or the gene vector that you introduce into the host cells, in order to take off limitations in the cells in expressing as much as possible of the target protein. And already now, if we compare to a lab which we worked with in the development of our Optivo biosimilar program, we from scratch, with that thinking, saw an advantage versus them and their setup, which they've been using for 15 years or so. It was an experienced place. So I think we had something already from the get-go there, and that's also now 5 approved patents on the basis of that technology. But we need to continue to invest. We are going into having our own cell line. We're working further on the way that we are putting together the vector to introduce or genetically engineer the host cells for maximum productivity, but then also how we set up the development of the production process. So we need to invest across this whole chain to really be best at cost or productivity, and then in the prolongation cost.
Okay, no, perfect. That's very interesting. Thank you so much for taking the time. We're all out of it now, but very interesting. Thank you.
Thank you.
Hi, the next company presenting is Ultimovacs, a company developing novel vaccine therapies against cancer. With us, we have here today Carlos de Sousa, the CEO. Carlos, I'll give you the word now.
Good morning everybody, and thank you for the opportunity to present you Ultimovacs. If we can move to the next slide. As we are a publicly listed company, I need to show you the slide, but let's start moving to the next slide, introducing you to Ultimovacs. So we are a company operating in the field of immunotherapies with a next generation universal cancer vaccines that are really off the shelf, easy to use. And what we do, our vaccine, is basically enable the immune system to identify and kill cancer cells. And we do that by flagging cells expressing telomerase. And this enzyme is expressing 85 to 90% of all cancers at all stages of tumor life. We have a strong phase 1 data, a good long-term safety, more than 5 years, strong signals of efficacy in our phase 1 studies, and also received recognition from the FDA by receiving fast track and orphan drug designation. We have quite a broad phase 2 pipeline, 5 phase 2 clinical trials enrolling more than 650 patients in 15 countries and addressing cancers with high unmet medical need. Also very exciting is that the readout of the first trials, these first phase 2 trials are going to start in the first half of 2023. We have a potential to be used as a baseline therapy because we work synergistic with immune checkpoint inhibitors. And our phase 2 trials are combining UV1 with 4 checkpoint inhibitors. We also received recently an extension of our patent in the US by having now a patent covering UV1 in combination with checkpoint inhibitors that will last at least until 2037, even before patent extensions. We have a strong cash position, very important in these volatile times. At the end of the Q1 2022, we have about 523 million Norwegian krone, what gives us a runway to the first part of 2024. If we move to the next slide, the treatment of cancer, you can see here that we have a very strong support from very important shareholders, long-term perspective, that have been with the company for a long time. We are a small team, 25 people, but very experienced from 5 nationalities and with offices in Norway and Sweden. And as I mentioned, a financial runway very comfortable to the first part of 2024. And very importantly, as you will see, across big potential near-term inflection points. When we move to the next slide, just to see really the development of the treatment of cancer. You see there, until years ago, the chemotherapy was the treatment of cancer, as you know, very toxic. And really, the treatment of cancer was revolutionized with the introduction of the immune checkpoint inhibitors, the purple line. That have big promises, but unfortunately didn't really work in all patients. So the treatment paradigm has been moving more to the combination that you see there, the dotted red line, to really increase the response to the patients and particularly extend the survival. But in these combinations, we are part of that with our UV1, our cancer vaccine, because we will be working synergistically with CPIs when combined. And this is very important, as you see in the next slide, because the checkpoint inhibitors, next slide, please. Yeah, the checkpoint inhibitors are the fastest growth class in all of pharma, expected to reach close to $70 billion by 2026. And here I just want to highlight that we are not going to be competing with these pharma companies. We're going to be used in combination. So a huge potential for UV1, because at the moment in our phase 2 program, we are combining with 4 out of the top 5 checkpoint inhibitors in the market. The next slide, you can see that in this field that is very competitive, we have a very good positioning because we have very strong benefits. One of them is that it's easy to use a UV1. We just do intradermal injections. So no need for complex hospital infrastructure, no need to do biopsies. The product is also truly off the shelf, ready to be administered when needed, and the patients don't need to be pre-screened to see if they would be able to take the vaccine. So very, very important. Also, from a perspective of commercial success, the product is easily manufactured. We are now in the final stages of moving to a commercial process with a very experienced manufacturer. And the product has more than 3-year shelf life so far, and a low unit cost, particularly when you compare with other approaches in the immuno-oncology space. As I mentioned, because we target cells expressing telomerase, we help the immune system identify these cells, and these are present in 85 to 90% of cancer cells. Really, we have a very broad usage across multitude of cancer types. And what we do, we activate the called helper T lymphocytes, or CD4, that are really the maestros of the immune response, and very important, are also responsible for the immune memory. In a recent presentation we had last week, we could see T lymphocytes still trained, educated against telomerase after more than 6 years. So very important to have this immune memory. So moving to the next slide, you can see our broad pipeline. We have 3 phase 1 studies previously already completed many years ago, but these are the active clinical trials. I will give you a little bit of details on the results and how these studies are ongoing. The important part here that we want to emphasize is that now we have these 5 phase 2 trials. The Initium is our trial. It's controlled by us. The other 4 trials are what is called investigator-initiated studies. We have had a lot of interest from specialist groups of oncologists, and also from some of the big pharmas. And this is a very extensive program, as I mentioned, that will enroll more than 650 patients. And we also have a second technology, the TET technology, where the first clinical trial in prostate cancer is running at this moment. So if we can move to the next slide, as you see, melanoma is our lead indication. These are previous 2 phase 1 trials. On the left, with combination with ipilimumab, this is now in more than 6-year follow-up. Very good safety shown in the long term. And also the same thing for the trial on the right, that is still ongoing with Keytruda, pembrolizumab, also showing a very strong safety and data that was very positive and presented at the big conference ASCO last year. But let me show you just briefly a little bit of the results in the next slide, please. You see here we reached recently in the study in combination with ipilimumab a median overall survival of 66 months. This is a small study, 12 patients, but very important for our data. And you see there, we of course we have to compare at the moment with historical data. We don't want to make more than what it is, but you see there's a big difference in terms of the median overall survival, but also in terms of the median progression-free survival, that is how long the patients take to progress. So in the next slide, you can see also from the second study with pembrolizumab, and the excitement about this data last year is that 57% of the patients responded to the treatment. And respond here is to reduce the tumor size or totally clean the tumor. So, and when you compare with pembrolizumab alone, again, historical data, it's more really important when you look at the complete response rate. These are the percentage of patients that clear totally the tumor. 33% when we added UV1 to pembrolizumab versus 5 to 12%. And also, the overall survival, this is how many patients are still alive. This is very important. You see here as the study progresses and we can follow up these patients, and we are going to have additional data this year on overall survival. If we move to the next slide, this data that I just showed you was reviewed by the FDA and was found of value to really grant us the fast track designation in 2 combinations with ipilimumab and pembrolizumab. And also this data was part of the data that was used to support the patents of the UV1 in combination with checkpoint inhibitors that we receive now recently allowance. So we have a patent for the use of UV1 in combination with anti-CTLA-4 like ipilimumab, anti-PD-1, and anti-PD-L1s. And as I mentioned, this extends further the patent protection to at least 2037 before any patent extensions. In the next slide, you can see now talking about the phase 2 program. Again, 5 phase 2 programs. And these studies is important to mention that are all randomized, comparative, and statistically powered. So all of these studies will have a control arm, what is extremely important not only for the regulatory authorities, but for our potential partners when we get the results and activate even further the discussions. So the initial study, as I told you, is our controlled study. Is in combination with Ipi/Nivo, that is standard of care in treatment of malignant melanoma, is running in the US and in Europe. And we are close to finish enrollment. We have now 137 patients out of the 154 that are required to be enrolled. So we expect the top-line results from this study in the first half of 2023. The second study, NIPU study, this is an investigation study that is Oslo University Hospital, the sponsor. And in the study, we collaborate also with Bristol Myers Squibb. Bristol Myers Squibb sponsors, provides Nivo and Ipi to the sponsor, and we provide UV1. We have now 78 patients out of the 118 patients to be enrolled. And we also expect the top-line results to be communicated in the first half of 2023. And this study is in mesothelioma and is running in the Europe and Australia. Then a recently started study in December is the DOVAC trial. This is also a study where we have a collaboration with the sponsors, that is the Nordic Society of Gynecological Oncology and the European Network of these type of cancers. And AstraZeneca supplies olaparib and durvalumab, and we supply UV1. So these patients are treated with UV1, olaparib, durvalumab in comparison in different arms. We have now 4 out of 184 patients enrolled, and the study is now in the phases of activating different centers in different countries. So it's always a little bit of a lengthy period. We will give a guidance on when we expect results with the quarter 4 report of 2022. And of course, we'll continue to provide updates in every quarterly report as we normally do. And in the next slide, you see the other 2 additional phase 2 studies. The FOCUS study is in combination with pembrolizumab in head and neck squamous cell carcinoma. This is running all in Germany. The sponsor is a very experienced team. We have now 18 patients out of the 75. And we will also be providing updates on the patient enrollment, and we will give an updated guidance also with the Q4 report of 2022. And the most recent study to be added to our broad pipeline is the LungVac study. LungVac study is a study in advanced or metastatic non-small cell lung cancer. The study is in combination with pembrolizumab and is going to run all in Norway. Basically, all hospitals in Norway that are involved in treating lung cancer patients are going to be part of the study. And we expect the first patient to be enrolled now during the second quarter. And the trial will enroll a total of 138 patients. In the next slide, you see just a brief update on our second platform technology. Is a very innovative adjuvant technology where the adjuvant and the peptides are combined in the same molecule, and we can add different peptides, so has a huge potential to derive different vaccines. We are now testing a vaccine in prostate cancer in the TENDU trial. That has now 8 patients enrolled at the highest dose cohort, and we expect to extend the number of patients to 12. So this study is ongoing, and it's very important give us safety, immune activation for this platform. And so far, no safety concerns. So if we move to the next slide, very important in terms of talking, what is coming for the company. We had a very successful 2021, a very successful first quarter of 2022. So what you can expect until the end of the year is that we will inform the market when the first patient in the LungVac study will be enrolled. We will provide in the fourth quarter some interim safety data on the TENDU. We will inform the market when the last patient in the initial trial is enrolled. But very importantly, we will provide an update on the overall survival of the 103 slide study, the one in combination with pembrolizumab in melanoma. So in the third quarter, we will be able to provide a 2-year overall survival update for all 30 patients in the study. And in the fourth quarter, we'll be able to provide an update on the 3-year overall survival for the cohort 1 of 20 patients. But of course, we are all excited with the first half of 2023, when we expect to be able to communicate the top-line results for both Initium and NIPU. And this can be transformational for the company in terms of partnering activities, but also discussions with the regulatory authorities. And of course, we will continue with the other phase 2 studies, the DOVAC and FOCUS and LungVac in the months after that. We continue also to present at several medical and scientific conferences results. It's very important to inform the scientific community about our progress. And if we move to the next slide, just to give a wrap up, as a company, we have a cancer vaccine platform that really enhances the impact and durability of the immunotherapies, broadly applicable in different cancer types and in different therapeutic combinations. There is a strong commercial potential as shown because we can use really in combination as a baseline therapy, off the shelf, easy to use. Very important, we have a good safety profile when more and more patients are treated with multiple drug combinations. A broad phase 2 program that have 5 indications, more than 650 patients. A recognition from the regulatory authorities to our existing data by receiving the fast track and orphan drug designations. Also the validation from a large pharma as they accept to participate in 2 of our trials, where we collaborate with oncology specialist groups that are also very validating. We have a high interest from these groups. We are very pleased to continue developing the TET platform where we see a huge potential. And it's very pleased for me to have an experienced team supporting the company, a strong shareholder base that has a long-term perspective, and very importantly in these difficult and volatile times, a good cash position that really takes us across multiple key value inflection points. And with us, I want to thank you for your attention and interest, and happy to answer any questions.
Thank you very much, Carlos, for such a nice presentation. And now we have time for a few questions. And I will start asking you about, it's more like a broad question. By pushing the immune responses with UV1 targeting telomerase, you are tackling, let's say, a shared point among a broad spectrum of cancers. How much can we extrapolate from your trial, for example, Initium to NIPU? Based on you're tackling the same pathway, but it's different cancers.
Well, all cancers, of course, are going to be different. I think that, and I mentioned this, if we have positive data in some of the cancer types, doesn't mean that it's going to work in all of them. But I think that if we start having some studies that have positive results, in a way, that proves the concept that we really helping the immune system to target cells expressing telomerase and helping them identify cancer cells is a concept that is valid and that can be applicable to other cancer types. And, as we know, all cancer types are different, all patients are different, some are harder to treat than others. So I think the benefit for us, for patients, and also for our investors is that we have all these multiple clinical trials happening, and in a way spreads the risk and de-risks the investment.
Very interesting. And in terms of your phase 2 pipeline right now, What will be your strategy, assuming that, for example, you get positive results across all the trials? What will be your strategy to move forward? Will you pick one and develop it, or are you planning to develop them all at the same time together with partners?
Well, I think that, as a company, we are prepared to move ahead by ourselves, but then, definitely, we need to focus in one or two indications. But we have been very public and open to say that we see the way of maximizing the potential for UV1 and for patients is really to enter into a strategic partnership with a partner that can really move ahead with different cancer types. What we are doing basically is to provide all the pieces in terms of data, a product that will be manufactured by a commercial process, very important before you move to phase 3, all the discussions with the regulatory authorities, that all these pieces will be in place if the data is positive for this partner to really move into phase 3 with multiple cancer types, not only the ones that we are testing now, but a lot of other cancer types that could be used. So really, we believe that a strategic partnership will increase the potential and the benefit for patients.
Interesting. My next question is related to your TET platform. You're coming to an end of your phase 1 trial. Could you let us know, or could you give us some color, and how are you planning to move this platform further? Are you going to combine it, for example, with UV1 peptides, or are you planning to, for example, also out-license the technology?
Well, without going into specific details, what we are going to combine with, because again, we are still in the process of getting patents. What I can say is that the TET platform has the potential to help us as a company to expand our pipeline of products. We're using for our own products. We can enter into collaborations with companies, other biotechs, for instance, that have peptides or antigens and need an innovative adjuvant. So that's another way of expanding. And we can also, enter into collaborations with pharmas or any companies that want to develop their products and they need an adjuvant. So we see different avenues to monetize TET. But of course, at the moment, we are looking at putting all the data together. We are also, of course, have other activities happening in the background in terms of preclinical CMC and together with this data. But we see a huge potential for TET for our own benefit in terms of our pipeline, but also to partner with other companies.
Interesting. My follow-up question is regarding the new study that you announced, I think that a couple of weeks ago, on melanoma after completing enrollment for Initium. Could you give us some words on what are you aiming with this trial, and could this trial in a way define the outcome of the platform of the program?
No, this is, I think the first part I want to clarify is that, again, the study is a supplement to Initium, has no impact on Initium and the data, but is going to pay more attention and go into more detail, with looking at biopsies, solid biopsies, liquid biopsies. The main goal of the study, and all patients will be treated with ipilimumab, nivolumab, and UV1. So it's very important for patients to see a benefit, because they are going to be required to do a lot of extra tests. So there are selected centers that are participating in Initium that will also then include these patients. And the big benefit is that we want to better understand how we activate the T cells, what happens to the T cells that are activated in the tumor, in the blood. And this is very important to understand better and more the mechanism of action of Initium, not only for regulatory authorities, but for potential partners, and to strengthen the package of data that we will have to support, the melanoma indication.
All clear. And one last question. Could you remind us what's your current cash position and your cash runway?
So we have a little bit over 500 million Norwegian krone, in cash now at the end of the Q1, and very importantly, because we are very, very disciplined and prudent how we use our cash. This will take us as a runway to the first part of 2024, And as you see now after the presentation, this is important because it takes us across the period of time where we will be receiving data from the first 4 phase 2 trials. Very good place to be.
Carlos, thank you very much for being here today and for your nice presentation.
Thank you so much to you and to all the listeners. Have a great day.
Welcome back. The next company to present is Zealand Pharma, and I'm pleased to be joined by their CEO, Adam Stensberg. Adam, I'll hand over the word to you.
Thank you, Adam. And thanks for the invitation to present today. I've really been looking forward to this. And you're right, I'm the new CEO of Zealand Pharma since March 30th, but I'm not new to Zealand Pharma. I've been the Chief Medical Officer and Head of R&D actually since 2010, so I know the company extremely well and the pipeline. But of course, I'm new in the role of as CEO. So I was announced at the same time we announced a refocused strategy for our company, where we're going to refocus on R&D. We have for a few years also been investing in building up some commercial efforts in the US, and there we will actually change the strategy towards R&D, and then we'll find commercial partners for our products, which are on the market, but also strategic collaborations along the pipeline, of course, starting with the late-stage assets, which I'll come back to later. So we have also undergone a quite significant restructuring over the last weeks, which we will complete by end August, where we are downscaling our US operations, which were around 130 employees. That will, and again, by end of August, will be down to around 10 employees in the US and then 220 employees in Denmark. Before I get into my presentation, I just want to highlight to you guys that since I became the CEO and also over the next few months, I'm really focused on operations in the sense that controlling the reorganization, getting our financial situation in good shape, which you can say, first of all, we were really happy to announce renegotiations of a debt deal with Oberland early last week, and then earlier this week, we announced that we managed to actually find a partner for Vigo, which is one of our commercial products. Then the next focus for me is to continue these partnering discussions, so we find, hopefully within the next months as well, a partner for Segalog, which is a product we have on the market, and then upcoming clinical readouts in our phase 3 program. But I'll come back to those things. So that's my short-term focus. But really on Zealand Pharma, we are a peptide-based drug discovery and development company. So it's our key capabilities are within peptides, which is a special type of drugs that you would also know if you know in Novo Nordisk, who work with peptides a lot as well. Let's go on with next slide. I will be making forward-looking statements, and here are the disclaimers. Next slide, please. And one more. Yeah, and let's just take the next slide as well. So we have a very, very strong pipeline, which I'll come back to a little bit later. If you try to envision where Zealand Pharma could be in let's say 2 to 3 years from now, then it's actually a company who have very significant products on the market through partnerships addressing, you can say, very significant disease areas. First of all, we have fantastic assets addressing management of type 1 diabetes. Then we have, and these are the clinical readouts that we have coming out this quarter and also next quarter, we have 2 phase 3 assets in the rare disease space, which we look very much forward to share more data on later in the year. And then we have a very exciting obesity portfolio, which have not gotten that much attention because it's still early, but we will actually see phase 2 readouts this year and also hope to progress some assets we have in phase 1. And if you have followed the obesity space, it's really an area that is taking off since the launch of Wegovy from Novo last year, and also with recent data from Lilly and so on. So this, I believe, will become a very significant value driver for Zealand in the future. And then we have exciting opportunities in the early pipeline addressing chronic inflammation. And all this is built on our proprietary peptide platform. Next slide, please. This is highlighting the 2 products we have on the market. And Vigo, if we start to the right, is the product we just found a partner for and announced that a few days ago. So this little insulin pump is now in the hands of mankind. And you can say it was a product we acquired 2 years ago when we acquired some commercial infrastructure in US. So it's never been, you can say, it's not something we have developed ourselves. It was just a product that was complementary to our efforts in diabetes. But a key priority for me here, of course, when we changed our focus towards R&D and downscaling the US was to get a new home for this product. So really, really happy to have announced that earlier in the week. Segalog, that's a different story. This is actually the first product that Zealand have taken all the way through development and registration in the US. It was approved by the US FDA last year in March, and we launched it summer last year. It's been a difficult launch for different reasons, and but we still believe that it has a very, very attractive clinical profile, demonstrating a mean of 10 minutes to rescue when people have had a severe hypoglycemic event. So I'm actually excited about the opportunity now to discuss with potential partners around Segalog, And I truly believe that in the right commercial hands, this could be a very significant product for patients with diabetes who takes insulin. So a rescue therapy for those people. So one of the things that we spend a lot of time on right now is to find the right partner for this product. So more on that hopefully over the coming months. Next slide, please. And next. So this is the pipeline, which is really where you guys should put the attention when you evaluate Zealand Pharma. So I believe, considering the size of the company, market cap, and also number of employees and so on, we have one of the richest pipelines that you can come across. We have quite significant assets in late-stage development and also a rich early pipeline. As I demonstrated or discussed on the slides before, these assets are targeting type 1 diabetes, rare diseases, obesity, and then inflammation. And today, later in the call, I will spend a little bit more time on the congenital hyperinsulinism, where we will have phase 3 data readouts very, very soon. Actually, we have at our quarterly call last week, we announced that it would come within the next few weeks. Then on glepaglutide, which is a GLP-2 long-acting analog, we'll have phase 3 data readouts in September this year. And that is likely, or that is the product that carries the highest value right now in our pipeline. So that's going to be a major value trigger for Zealand Pharma when we see those data, of course, depending on if they're positive or negative. Then in the obesity pipeline, we have a collaboration with Boehringer. I'll come back to it, but just again highlighting that we will actually see phase 2 data from that program this year. They have 3 ongoing studies. So that could be important again for the case of Zealand Pharma. And then our amylin analog and GIP agonist. Then we have in the preclinical area, we have a very good collaboration with Alexion, which is now part of the Astra group, on a complement C3 inhibitor. Again, something we would expect to drive significant value in the future for Zealand Pharma. Next slide, please. Just a few words on our peptide platform, because it is really what you can say differentiates Zealand from many other biotech companies. So you might know of companies, or at least you know medicines that are based on small molecules, which are classical tablets or molecules that a pill you would take. So that is one group of medicines. And then some of you may also know large proteins, which are very famous in cancer therapy and chronic inflammation therapy, and is often injected or infused into a vein or injected into the muscles or subcutaneous depot. Peptides, that is somewhat in between there. So it's a very small protein. Our human body actually utilizes peptides. So if you have cells that want to signal to other cells, that often happen via peptides. So they release a peptide into the bloodstream, and then that peptide have a receptor at a different cell or a different organ, and then it sends a signal. A common theme for peptides when they're endogenous is they have short half-life. So they only live for like maybe 3 to 5 minutes. Our, you can say, source of inspiration when we look to the left is actually either endogenous peptides, and there we have examples of glucagon, GLP-1, GLP-2, which you can see in our pipeline. But we can also use peptides from venom of spiders or snakes. They have used it so that's where we get into the chronic inflammation, because some of these peptides can actually inhibit pain sensations and so on. And then one of the things that have started to get more and more attention, and it's also why I would actually describe that Zealand's platform is now, you can say, it's really getting to a completely novel and super exciting period. That is to use computational design and phase display. So these are completely new modalities where we don't have to have an endogenous peptide. We can actually, as long as we know what we want to attack, like 2 molecules that interact, and we find the binding groove, then we can design peptides completely novel, unknown to the world, that can then inhibit or stimulate such interactions. So this is really taking our innovation ability to the next level. So something that you will see much more on in the future when it comes to identifying new targets and new hits. Then a thing that really differentiates Zealand from many other peptide companies is our proven ability to actually make peptides into drugs. And it's not an easy task because, as I mentioned before, typically a peptide have a half-life of only 3 minutes. And if it's such a short half-life, then you would have to, you can say, take a pill or inject yourself every hour throughout the day. So what we have really become experts in is to extend the half-life. So maybe you can do an injection only once a week. We have also made sure that we can make formulations so they can be formulated in a way with high concentrations and so on. So it's easy administration of these drugs. And then you can say we have demonstrated our ability to take them all the way through development to registration and approval, as exemplified by the rescue pen I discussed before. So all in all, this is really what differentiates Zealand beyond our pipeline towards other biotech companies. Next slide, please. And I think we should just actually, in the interest, go a few more slides forward. One more until we get to the rare disease. One more, and one more. Yes, so maybe next slide. Because I really want to make sure that we have time to discuss our rare disease assets, which are in phase 3, both of them. So we have dasiglucagon as a subcutaneous infusion via a small pump addressing congenital hyperinsulinism. That's where we will have our phase 3 readout within the coming week. And then we have short bowel syndrome, where we have a long-acting GLP-2 analog, glepaglutide, where we will have phase 3 results in September. So these are two of the major value drivers in our pipeline this year. And congenital hyperinsulinism, that's an ultra-rare indication. So these are small children born with a genetic defect in the pancreas, where they produce too much insulin. And if you produce too much insulin, irrespectively of your blood glucose, then you will end up with dangerously or life-threatening low blood glucose, because insulin lowers glucose. So it's really a devastating disease where there are no good therapies available for these children today. So many of them would have to have their full pancreas removed within the first few months of life in order to survive. And that's, of course, a very serious operation, which carries other issues with them. So we hope with dasiglucagon that we can address that, because dasiglucagon is doing the exact opposite to insulin. So it raises plasma glucose. So it's basically a battle where we will try to battle the excessive insulin by infusing dasiglucagon. And we see a huge unmet medical need and also a potential for fast approval with FDA if we meet the primary endpoints in our next phase 3 study. Then short bowel syndrome. That's a disease where patients have had surgery to the intestines for different causes. It could be Crohn's disease or cancer or just an acute lack of oxygen in the intestine. So they're left with too little intestine to absorb fluid, energy, and electrolytes. And then many of these patients become dependent on parenteral support, meaning that they will have infusion of these energy and water and so on, what have you, to a central venous catheter for up to 17 hours every day in order to survive. And a GLP-2 have already been shown to be able to increase the absorptive capacity of the intestine, meaning that that short gut that is insufficient actually becomes more effective in absorbing whatever you eat and drink. And by doing so, the patients will become less dependent on the parenteral support and can survive in a more normal situation like the rest of us by just drinking and eating. So we are extremely excited about this program because there's a short-acting GLP-2, Gattex, on the market today. It's sold by Takeda, and it's selling for around $650 million US dollars every year in a few patients. And with our program, if we meet the clinical attributes that we hope to see, it's going to be a very significant, we believe, improvement to care because it's a long-acting agent. So Gattex you have to inject daily, and you have to mix it before you inject it and dose it per weight and adjust for kidney function and so on. With our product, we believe we could have a once or twice weekly injection in a very simple auto-injector and not having to adjust for weight or kidney functions and what have you. So it's really about making a quite complicated treatment much easier, but hopefully also having some fantastic clinical readouts that we should see here in September. So next slide, please. So this is the program addressing congenital hyperinsulinism, and we only need to see the data from the last phase 3 study, which is the one in the middle, where we will see data in the coming week. And that is a study in neonates down to the age of 7 days. So if that study comes out positive, then our ambition is to engage with the US FDA over the summer and then hopefully submit the NDA by end year. And that could be a fast approval. So second half next year, if everything goes well, we could have this product on the market in the US, addressing, helping these small children and their families to better cope with this, you can say, devastating disease. It's really, really something where we can do a huge difference for these children and their parents. The good thing, or the interesting thing here, is that it's so difficult to live with the disease in the early years, but as these children, they approach the teenage years, many of them grow out of this condition, and it's a known phenomenon. If you have a beta cell that produces insulin day in and day out without being controlled, these cells have a tendency to burn out. So in type 2 diabetes, that's an issue, and that's why you see many patients with type 2 diabetes, they end up on insulin late in life because they have produced too much insulin for a long time. But in this situation, it's actually a good thing, because then suddenly these beta cells that produce too much insulin, they die out, and then they become less severe affected, these children. So if we can maintain them and help them throughout this early childhood into the teenage years, they might actually be in a situation where they can live rather normal lives later in life. So I'm extremely excited about the opportunity to help these patients. Next slide, please. This is the most important readout we're going to have this year in September. That is our long-acting GLP-2 analog, glepaglutide, where we have demonstrated good data in phase 2 some years ago. So if you take the next slide, and the next slide, please. This is the trial program. I just want to highlight that program because we have EASE-SBS1, which is the pivotal study where we will have results in September. And this is a 6-month study, and we have actually allowed then patients to roll into EASE-SBS2 and then further into EASE-SBS3, meaning that we have had patients on treatment for now more than 3 years on glepaglutide. So of course, for us, that you can say underscores the potential, because we see a lot of these patients who were enrolled originally, they still stay on treatment. So you can say that's exciting to see that patients choose to remain in the program. We still need to see the clinical data readout and see if it's a twice weekly or once weekly treatment, and of course also see how effective this product is compared to expectations and so on. But all that we will know in September. Then we have a smaller EASE-4 study running as well in parallel, which you can say addresses some, you can say, more technical features of the program. So if you take the next slide and go to slide 24, actually. This is just to highlight our approach in obesity, because I think within the next year or so, it will be very obvious to the world that we have some fantastic early to mid-stage assets in obesity, and it's really an area that is taking off right now. So we are still in the early days of obesity. I personally believe it's going to be one of the very, very big opportunities for the industry, and we will see a lot of large players wanting to get into this space, why it's also interesting now with our strategy of partnering that we have such interesting assets in obesity. We have either molecules that stimulate 2 receptors, as the one we have with Boehringer, that's the one you see in the top, or we have products which are monoagonist against receptors in obesity. So if you take the next slide, that shows data that we presented last year for the obesity programs that we have with BI. And as I said, we have 3 phase 3 studies. Hopefully within the next year, Boehringer will take phase 3 decisions. So could be very exciting. Next slide, please. And that is just to highlight our amylin analog, which is right now in phase 1 development. And the reason that there's so much excitement around this one is that Novo is going to start a phase 3 study with an amylin analog that they have. And we just heard that Lilly is also progressing an amylin analog. So this target carries a lot of hope, I would say, within treatment of obesity. And Zealand is the only other company beyond these 2 large players who have amylin analogs in clinical development. So with that, I would actually suggest that we go to slide 32, which is the last slide of my presentation, and just highlighting that we have a number of significant milestones lined up over the coming months and into the later part of the year. And I'm truly excited about the opportunity to lead Zealand in this next phase. And should we open up for questions then?
Perfect. Thank you very much for that presentation, Adam. First question then on obesity, and I'm curious to hear how sort of able or comfortable you feel with sort of the data you have on hand for your obesity candidates in relation to what we've seen with Wegovy and now most recently with tirzepatide. Quite fierce competition, I imagine. But can you put some of the data that you've seen in relation to that, perhaps?
Yeah, no, I feel very, very confident, but of course I also acknowledge that it's early days. But for me, it's actually just a confirmation that we are on the right track, that we see these great data from both Wegovy, which is a GLP-1 receptor agonist, and then tirzepatide, which stimulates two receptors, both the GLP-1 and the GIP receptor. And if you look into our pipeline, we actually also have a GIP receptor analog that we could take into phase 1. So obesity is a super complex disease, and you can say that ultimately my belief is there will be different modalities needed in order to address each individual patient with obesity to achieve their goals. And reality is 15 to 20% weight loss is really, really a lot, and that's also why you see this demand kicking in. But patients actually, many patients, they would like to get into the 30 to 35% weight loss, which is what you've seen with bariatric surgery. So there's room for more. And I've heard this question for a long time, and I think the questions are actually starting to change a little bit, because people have asked over the last year, how are any company going to catch up with Novo or potentially Lilly? Reality is, it's not a matter of catching up, it's a matter of not missing out, because if you're a large pharma company in this surrounding space, you cannot afford not to be in the obesity space. And I think companies are realizing that right now when they see the dramatic demand for treatments that have these kind of effects. And I see our molecules as being differentiated. And the GLP-1 glucagon has a different, you can say, opportunity than a GLP-1 GIP. So ultimately, hopefully, there will be 4 or 5 players who will be very significantly doing something for treatment of obesity, and I see us being able to help out there.
Okay, no, that's interesting. And I guess among the sort of large cap pharma, which ones do you see as potential new entrants into the obesity field? I guess ones with a sort of a metabolic disease focus, or what are the sort of prime suspects wanting to catch up there, as you were saying?
Yeah, no, you're absolutely right. It is, of course, companies who have a metabolic disease focus, but there's quite a lot of companies who have had like obesity comorbidities on their plate, such as Astra, Merck, J&J, Pfizer, and what have you, some of the really large players. But I would also say, I mean, look at the companies who were high in hypertension, on cholesterol medications, and so on. I mean, these are the kind of companies in my mind who cannot afford to miss out on this one, because my sense is that this could become even bigger than these areas became, you can say, 10 and 20 years ago.
Okay, no, interesting. And maybe just a question on the GLP-2 analog for short bowel syndrome. What does the development landscape look like there? Are there other GLP-2 analogs, or indeed other type of drugs also in development for short bowel syndrome, or is it a quite open landscape, so to say?
Yeah, so there's Gattex on the market sold by Takeda today, and then there's our product, which we will have phase 3 readout this year, and then there's one competing product in phase 3 as well. That's from a company called Vectiv Bio. The molecule is called apraglutide. They expect data readout late next year if they manage to get the patients into the phase 3 study. And then there is, as you can say, some earlier efforts with the long-acting GLP-1s and so on, which I don't put as much attention to, but so I think these are probably the 3 assets you should look most into, and I think Zealand is in a very good position here with our molecule, because we have the readout coming up here within the next few months, and then hopefully we'll pursue an NDA submission fast. with positive data.
Okay, no, interesting. And maybe finally, for those who are unfamiliar or sort of just peripherally aware of Zealand Pharma, your cash position and sort of financing needs and so on, looking ahead, what can you say around that?
Yeah, I think we just renegotiated this debt deal with Oberland, which took us into, you can say, our financial runway into 23, and then we announced the Vigo sell here earlier this week, which extended our cash runway, of course, even further. And then, so it's I would say it's probably around a year right now, but then we have all these partnership opportunities, starting with Segalog, which could bring in again additional non-dilutive funding. So I'm actually quite comfortable right now, even considering the current environment, which is quite hostile towards biotech. But I think with the restructuring that we announced in March, also reducing our OPEX with expected with more than 35% starting next year, we've really set ourselves up to be one of the successful companies that will come out of this crisis as actually a stronger company than when it started, when looking at the biotech industry.
Okay, no, great. Adam, thank you so much for an interesting presentation.
Thank you for allowing me to present here today. Have a good conference.
Thank you.
Bye.
Hi again. Our next company is Targovax, a biotech company developing therapies targeting cancer with engineered oncolytic viruses. With us, we have here today Erik Digman Wiklund, the CEO. Please, Erik, the word is yours.
Thank you. Good morning and welcome to Targovax. Let's dive into it. Next slide, please. That's just a safe harbor. And next one, please. So immuno-oncology is really revolutionizing the way we treat cancer. Only 10 years ago, immunotherapy was still a pipe dream. And today, more than 500,000 patients per year are benefiting from immunotherapy. There are more than 3,000 ongoing trials, and 40% of patients in the US of cancer patients are eligible for immunotherapy. And this is quite remarkable when you think of that having happened in just the last 10 years. Next slide, please. So when we talk about immunotherapy today, we're usually referring to checkpoint inhibitors. And checkpoint inhibitors are great because they interfere or block the tumor's first-line defense mechanism against the immune system. And the most common strategy to achieve that is, as you can see on this slide here, is to use monoclonal antibodies that interfere with the interaction between PD-1 on T cells and PD-L1 on tumor cells. And this has proven highly effective, and it's become really the cornerstone of current cancer treatment today. In some cancer forms, this is really what is used and works, and it delivers deep and durable responses. The first PD-1 inhibitor was approved 7-8 years ago, and in that short period of time, it has grown to a market where there's $25 billion of sales per year for this modality alone. And there are to date 8 different PD-1 or PD-L1 antagonists approved in the West. Next slide, please. However, even though they work really well, this is great for patients, still only a minority of patients respond. So depending on the cancer type, somewhere between 0 to 40% of the patients will have a benefit of the checkpoint inhibitor. And of those that have a benefit, more than half end up relapsing. So they become resistant over time. So it's becoming clear that monotherapy with PD-1 checkpoint inhibitors simply isn't sufficient. So how do you make PD-1 checkpoint inhibitors work for more patients? And that's what we're doing at Targovax. Next slide, please. We offer a solution to this problem via our oncolytic immunotherapy, ONCOS. And ONCOS really, what it does is reverses the immunosuppressive defense mechanisms that the cancer deploys against the immunotherapy or the immune system itself. And it does that by driving inflammation in the tumor, priming fresh new T cells that can recognize and target the cancer cells. And at the same time, we can deliver immune stimulatory genetic payloads directly into the tumor. Next slide, please. Here's an overview of our development program. ONCOS 102 is the name of our lead clinical asset. This is an oncolytic virus that we deliver intratumorally, so we inject it right into the tumor. We try this in multiple indications. And the leading indication, which I will show you a comprehensive data set from today, is in PD-1 refractory melanoma. So this is melanoma, which has been prior treated with anti-PD-1 checkpoint inhibitors, but then the patient relapsed and is no longer having benefit. And we're testing the hypothesis whether ONCOS 102 can reinstate the response to the checkpoint inhibitor and thereby expand and improve the response rates and the clinical benefit for the patients. Next slide, please. And next. So to give you a little bit of flavor of the size of the unmet need here, there are roughly 100,000 new cases of melanoma or malignant melanoma every year. And half of these are unresectable, and those patients are treated primarily first with PD-1 checkpoint inhibitors. So 50,000 patients get the PD-1 checkpoint inhibitor. Half of those have a durable, deep effect, but half of them, so roughly 25,000 patients per year, will become resistant and no longer have any good treatment alternatives. And we estimate that somewhere between 10 to 20,000 patients in melanoma alone would be addressable with an intratumoral therapy like ONCOS. And if we look more broadly, you know, I mentioned depending on what cancer type you look at, you may have more patients that become resistant or never respond. If you look at lung cancer, for instance, more than 100,000 patients per year become PD-1 resistant, 50,000 patients in head and neck. So this is a massive unmet medical need that we are trying to address, and we're doing it first in melanoma to show this as a proof of concept can be done with ONCOS-102. Next slide, please. In a phase 1 trial we recently completed in 20 patients, we showed 35% response rate in this population. So patients that stopped responding to PD-1 inhibitor, we give them ONCOS-102 and then retreat with the PD-1 inhibitor. So you don't expect these patients to respond to the PD-1. Out of 20 patients, we achieved a response in 7, 35% ORR. And this compares very favorably to other published data in this indication. So here we benchmark our data against all the published data we can find from other companies active in the same population. And if you compare here, you see our closest competitors would be Replimune and BioNTech. They achieved similar response rates to Targovax, and the rest are lower and we would consider less promising. Interesting to add here is that Replimune is also an oncolytic virus, although based on herpes, whereas ours is based on an adenovirus. So it seems like maybe an oncolytic virus approach here is particularly effective in PD-1 resistant melanoma. Next slide, please. One aspect which is very important with these type of local therapies is that you're able to get a systemic effect. So you're injecting one tumor, but you have impact in other tumors that you didn't inject as well. And that's something you achieve if you're able to generate T cells that recognize the cancer and that these can travel around the body and find other tumors. So on this graph here, we're showing you all the tumors that we tracked from all the patients in the trial, overall 36 tumors were tracked. And then the blue are injected tumors and the gray are non-injected tumors. And you can see here that we get some deep and solid responses in the blue. See 4 of the blue are completely disappearing, so 100% change from baseline. But we also see this in the non-injected sites. 2 non-injected tumors completely regressed, and overall 12 out of the 36 non-injected lesions reduced in size. So we consider this a very important evidence of systemic activity by ONCOS-102. The mechanism is working as we're hypothesizing that it should do. Next slide, please. So let me show you a case example. So here we have a concrete patient that's enrolled in the trial. This patient, if you look in the down right corner, you can see this patient has had several lines of prior therapy. First surgery, the patient has been treated with TVEC. That's interesting because that's another oncolytic virus, and the patient failed to respond to TVEC. Then it had ipilimumab, CTLA-4 checkpoint inhibitor, and then pembrolizumab, a PD-1 checkpoint inhibitor. And then the patient was enrolled in the trial. And then we have here 5 pictures over time. You can see baseline that shows the tumors as they were when the patient enrolled in the trial. And then in week 3, so in the first 3 weeks, we only treat with ONCOS-102. It's before starting the anti-PD-1. And particularly in the top picture here, you can see there's a clear inflammation and necrosis going on. It's a dramatic effect just after 3 weeks from the ONCOS-102 injection. And then over time, the tumor regresses, and by the end at week 27, the top lesion here has completely disappeared. The second lesion had reduction, and then overall, this patient had a partial response. Next slide, please. So we focus very hard on doing deep translational analysis to really understand what's going on at the cellular and the genetic level in these patients. And we did 3 biopsies in this case. We have a baseline, so pre-treatment. We have a week 3 biopsy, that's on ONCOS-102 only, and then a week 9 biopsy, which is on ONCOS and pembrolizumab combination. And then we look at T cells, we look at other cell subsets also, but here I show you T cells. So you can see at baseline, very little T cell infiltrate, almost no T cells present in the tumor, neither the CD8 on top or the CD4s on bottom. And then already by week 3, you start seeing T cells coming into the tumor in response to the injection of the virus. The virus is driving inflammation. T cells are coming in. And at week 9, it's even stronger. By week 9, you can see almost everywhere in this picture, you have these red dots, and that illustrates that the CD8 T cells are really now penetrating into the lesion. Right-hand side, we're quantifying the whole slide, and you can see that we're not just showing the nicest pictures. There is indeed an increase overall in both the CD8 and the CD4 positive T cell population. So we can corroborate the shrinkage we see in the tumor with infiltrative T cells, and that's what we're after. Next one, please. That was a case example. Now I'm going to show you aggregate data for all of the patients. And here at on this graph, you're seeing box plots averaging all the patients, and we split them in patients with disease control and progressive disease. So disease control has the responders and the stable disease patients, and red is patients that progressed. And you can see if you start at baseline, there is the tendency to somewhat higher T cell infiltrate in the responding patients compared to the non-responders. And then by week 3, this increases further. So at week 3, you see everyone has an increase. It's still less in the progressive disease, but it went up from baseline. So everyone had T cell infiltrate at week 3. Then interestingly, what happens at week 9 is that in the responders, T cells keep increasing, whereas in the progressors, they come back to baseline. And this shows that in some patients, we're able to really drive a sustained pro-inflammatory remodulation. We're getting T cells in consistently, and they persist over time. And then this translates into a durable clinical benefit for the patient. So this was the CD8. You can see also the p-value here is clearly statistically significant, the analysis, even though we just have 20 patients. Next slide, please, or click for the perfect. Here is the same data for the CD4 T cell population. And again, the pattern is identical, right? You see the baseline, it goes up at week 3, and then it continues to strengthen in the responders at week 9. Next one, please. And then when you look at T cells, you have different T cell populations. Some are advantageous, some are disadvantageous. It's a complicated picture. So you want to understand in a bit more detail what type of T cells you have present. So therefore here we look at granzyme B expressing T cells. This is a marker for activated cytotoxic T cells that are engaged in tumor killing, as opposed to an exhausted or negative T cell, and this is an important difference. And encouragingly, we see here the difference is actually even bigger when you look at this activated T cell population. You can see at week 9 that there is a strong upregulation compared to baseline in activated T cells in the responding patients. Next slide, please. So we also look more closely at the T cells to see what they recognize. And here I just show an example with an ELISpot analysis. We look for T cells recognizing relevant tumor targets. And in this case here, this is from our complete response patient. You see that we are able to isolate out and propagate up a population of T cells recognizing MAGE-A1. So this shows that we're not just generating a T cell response to the virus, but there is actual tumor targeting T cells being primed and activated and then differentiate. And then this is very important in order to actually have the systemic activity. And then we can show on the next here, if you click one more time. This is hallmarked by a very interesting individual here, where you can see on the schematic. In this case, there were 4. This patient had 4 lesions. We injected the 2 red ones with ONCOS-102. And what happened remarkably was that there was no response in the 2 red injected tumors, but the 2 blue ones, they disappeared. So we had a complete regression in a non-injected site in this patient. And then it turns out, when we look closely at the genetic profile of the tumors in these patients, the injected tumors had lost the ability to present antigens on its surface by what's called a beta-2M loss of function. So this means the tumor was no longer presenting any targets for the T cells in the injected site, but those targets were presented in other places, and there the T cells could have its impact. So this shows that the activity of ONCOS-102 is driven by an anti-tumor T cell response. It's a very important observation mechanistically to understand how this product works. Next one, please. This looks rather detailed. I'll just make a very simple statement about it. We look at gene expression. As I said, we go very deep into the analysis of these patients. Now, what we see by gene expression confirms the cellular data I just showed you on these biopsies. We see a broad upregulation of relevant immune gene signatures, both at week 3 and at week 9. So ONCOS-102 drives broad pro-inflammatory remodeling. You can see it by cellular data. You can see it by gene expression. And next one, please. And as I showed before, you know the T cells went down in the progressors but kept growing or increasing in the responders. We see the same here at the genetic level. Blue is patients with response, red is patients with progressive disease, and you see very clearly, also on the gene expression level, clear separation at week 9. In the responders, we're able to drive a sustained immune response that lasts and delivers a durable clinical benefit for the patient. Next slide, please. So looking more deeply at this, it becomes really interesting because you can look at particular molecules or targets that you can use for future combination. And we're certainly very interested in looking at what happens to these checkpoint inhibitor molecules because they present opportunities for future combinations that we can do. So on this slide here, I show you then 5 individual the most frequent or most well-known checkpoint inhibitor targets. And we talked about PD-1 before, but CTLA-4 is another interesting target which is already approved in melanoma. Click, please. Interestingly, here you can see that CTLA-4, now we're comparing responder to non-responder. Right-hand side means higher in responder. CTLA-4 is upregulated more than 25-fold in our responding patients. So this now presents a target for us that maybe we can combine with in the future to deepen and prolong the response. So what we expect is if we put the CTLA-4 inhibitor into this combination here, we should deepen and prolong the benefit for the patient. Next slide, please. CTLA-4 also offers another interesting benefit. CTLA-4 is known to reverse inhibitory regulatory T cells in the tumor, which is an advantage. It improves your likelihood of having an effect. And also CTLA-4 is very important in priming these tumor-specific T cells that I showed you we can generate and that we get into the tumor. So CTLA-4 enhances this process early on to both prime and expand tumor-specific T cells. So we believe the totality here argues for moving to combining with the CTLA-4 as well as the PD-1. And that's what we're planning to do next. Next slide, please. So based on this very encouraging data set, we're now designing a multi-cohort phase 2 trial where we plan to both confirm what we have seen and explore novel combinations. And we formed a clinical collaboration with the US company Agenus that have a very interesting portfolio of checkpoint inhibitors available. And we're doing this trial in collaboration with them, and we're going to combine with their 2 checkpoint inhibitors, balstilimab, which is an anti-PD-1, and then more importantly, botensilimab, which is a second-generation CTLA-4. So this is an engineered, improved version of CTLA-4 that deals with many of the issues with the first-generation CTLA-4. It's a unique molecule. It's by far the second-gen CTLA-4 with the best data available and most advanced. So we're very excited to be able to access this molecule. So what we're planning to do in this trial is show step-by-step how ONCOS and these combinations work. We start with cohort 1, you see here, ONCOS 102 monotherapy. Then we add on a balstilimab, so the PD-1 combination. That's the same as we did in the phase 1 that I've showed you data for. As number 3, we add in the CTLA-4. And the 4th cohort is really the one where we expect or hope to knock it out of the park, the triple. ONCOS plus balstilimab plus botensilimab. And we think this triple combo really has the potential to boost the response rates up and beyond the 35% we have seen and really put Targovax in a competitive position. And we think this is a highly differentiated approach. No one else is trying at the moment, and we have access to this unique molecule, botensilimab, from our partners at Agenus. Next slide, please. In addition, we plan to explore also more combinations together with collaboration partners. We're in discussion with other companies to put in here cohort 5. We plan to do a co-stimulatory molecule to see if we can find other strategies that can enhance this activity even further. And the idea is, next please, is that all of these cohorts, as we get data, they can independently form the basis for subsequent registrational trials to get to the market eventually. So we see this as a smart way of building stone by stone, assessing multiple combinations, opportunities for partnering and differentiation, and find the best possible combination approach for ONCOS 102. Next one, please. Let me very quickly tell you how we're planning to build on this program, and we're moving into delivery of circular RNA, which is an exciting new area of biology. Next slide, please. So RNA-based therapeutics really have a moment now, driven by the COVID vaccines, which are based on mRNA. But RNA-based therapeutics still face several critical challenges. RNA is chemically unstable. It's quickly degraded. It's also difficult to deliver. You need delivery systems that both get them to the right site and make them stable enough. And in terms of a cancer, it's difficult to get them sufficiently penetrating into solid tumors. Now, we believe Oncos offers a very attractive delivery system for RNA-based therapies. We can effectively express RNA from our Oncos DNA delivery viral system. And this is what we're now planning to do. Next, please. And we're doing this through circular RNA. So the big advantage of circular RNA is that these are highly stable transcripts that have a half-life of days or weeks rather than just hours, which is the standard for mRNA. And circular RNA was actually discovered originally by myself and my colleague here, Thomas Hansen, about 10 years ago. And this has now evolved into a very hot area, illustrated on the right-hand side here by Flagship and Moderna actually setting up this company Laronde, which raised nearly $500 million last year to do gene therapy based on circular RNA. So this is not just something we came up with. This is a really hot area at the moment, and I think we have a unique angle for delivery of circular RNA with our Oncos platform. We hired Thomas here. Thomas is the pioneer in circular RNA research. He discovered the field and sort of built the whole thing into what it is today. And he now works for Targovax and is setting up our team to push Targovax in this direction. Next one, please. In the interest of time, I think we can skip this. Move to the next one. So we are planning here a multi-pronged approach. We are currently working on developing circular RNA Oncos vectors. So it's an Oncos construct that expresses a circular RNA for various purposes, for protein expression or gene therapy, for vaccine purposes, encoding tumor antigens, for boosted immunological activation, and also to regulate other non-coding RNAs. Because these molecules are highly stable, they offer a broad range of opportunities. It's really a toolbox that you can use as a platform to achieve multiple goals that are not possible with a linear RNA. We are currently demonstrating in vitro the advantages of circular RNA, and we will have proof of concept data of this second half of the year. And I think this is Oncos 102 is our lead clinical program. That's what we're pushing forward and exploring clinically and getting to the market with the right type of combinations. And this program here is really learning from what we do with Oncos 102 to enhance and expand this into a platform that we can build significant value from in the future. Next slide, please. And next one. Next. Yes, so I will just wrap up with this slide to explain how we plan to create value from our different programs. So we have our Oncos 102 program, as I said. We have circular RNA at the bottom. What I haven't touched on today is that we also have a KRAS immunotherapy program. If we start with Oncos 102, our aim is to do this one multi-cohort trial that I showed. We gear this trial to answer important questions from regulators, as well as make it interesting for partners, both in terms of what we combine with, as well as the sizing. So our aim is that based on this trial, we will partner out the program, out-license it, and then someone else will do the registrational development. We will use the funds generated from that for our own pipeline development. With the KRAS program, we're actively setting up low-cost investigator-initiated trials, and we're bringing this into the clinic in multiple indications and combinations. And this will generate both news flow as well as partnering opportunities as we move forward. With the circular RNA, our aim is to establish this as a platform, get IP filed, and show we can broadly use Oncos as a delivery system for circular RNA. Then we plan to form early partnerships on this program, developing circular RNA constructs for partners, and also develop our own in-house products. So the aim is to make this as cash neutral as we can by early partnering, use that to finance our own in-house development, and also expand out more broadly what can be achieved. We are focusing on oncology, but we think this technology can be applied much more broadly. So with that, I conclude my presentation, and I'm happy to take any questions from the audience.
Thank you very much, Eric, for such a nice presentation. We have time for a couple of questions now, and I will start asking you about your collaboration with Agenus that you will start now, as you mentioned, the clinical trial combining ONCOS 102 with their immunocheckpoint inhibitors. What is the rationale including all these arms in the trial? Is it something that was a requirement to test ONCOS 102 with the different checkpoint inhibitors as was separated and combined, or it's something that you decided for a specific reason?
So if we begin in melanoma, PD-1 checkpoint inhibition is the cornerstone or backbone treatment. You have to have the PD-1 in there. It's almost impossible to do development work without combining with the anti-PD-1. So that's a must. But we think both scientifically and business strategy-wise, it makes sense to try additional combinations on top. And as I showed you by the data, and hopefully convince you that both mechanistically and from what we see, CTLA-4 is really on top of the list of what to combine with in addition to the PD-1. And also CTLA-4 is an approved modality in oncology or in melanoma. Now, the first generation CTLA-4, ipilimumab or Yervoy, if you will, has a lot of drawbacks. It's coming off patent soon, it's toxic, it's difficult to combine with other treatments, and it also doesn't work in all patients. So we really looked for a partner that has the best possible CTLA-4 antibody out there. And the advantage of the engineered CTLA-4 of Agenus is that it interacts with both or more Fc gamma receptors, so it broadens out the number of patients that can respond. So it's expected almost certainly to give better response rates than ipilimumab. And also it's engineered to not activate the complement cascade, which means that it will have a better tox profile. And this has already been shown in phase 1. So we worked really hard to convince Agenus to let us use this molecule. They're not partnering with anyone else on it. So we will have unique access to this second-gen CTLA-4, which really is leading the pack. So I think this is what drove us to talk to Agenus. And then they have a PD-1 blocker also, which means we can access everything we need from just one partner.
All right, that's very useful. Thank you. And last question for today. You have shown beautiful data in melanoma, in mesothelioma, and as you said at the beginning of your presentation, there are other companies working within the same space. What is a key point that Targovax has that makes it different from competition?
As far as we are aware, no one has looked as deeply as we have done translationally. We have sequential biopsies, like I showed you. We did both cellular and genetic work. We also have systemic data that I didn't show. So I think when you have small data sets like we do, it's always in question whether you're lucky or you're unlucky. But I think we can show mechanistically exactly why the responders respond. It's not a fluke. These responses are real and they're driven by mechanistic activity from ONCOS 102. So it gives us confidence in what the product is doing, and it's not just by pure luck. And also, this depth provides us with, I think, a unique opportunity to really be smart about the next development steps, which I don't think is necessarily that simple for others to replicate. Plus, a third advantage, if you compare to especially systemically delivered antibodies, a local virus like this has very few side effects. So you can there's limited toxicity beyond what you get with whatever you combine with. So this means it offers quite broad opportunities for combinations that is more challenging for others to achieve. So I think the totality of that is what sets us apart and gives us a clear path to future success.
Eric, thank you very much for being here with us today and for your nice presentation.
Thank you very much. Bye-bye.
Hi again. Our next company is Cantargia, a Swedish biotech company developing drugs for cancer, as well as for inflammatory and autoimmune diseases. With us, we have here Göran Forsberg, the CEO. Please, Göran, the word is yours.
Thank you so much. It's a pleasure to be back and present Cantargia and also introduce those of you who are new to the company to our story, but also to show the progress we made and, let's say, the very nearby upcoming events. So next slide, please. And then we can go to the next slide. So for those who are not familiar with the company, we're an antibody company. We are focusing very much on a unique molecular target called IL-1RAP, which has lots of opportunities, not only in cancer, but also in autoimmune inflammatory diseases. And the background is that it's overexpressed on cancer cells. It has very low normal tissue reactivity, but it's also found on several inflammatory cells and is involved in signaling of both IL-1, IL-33, and IL-36. We currently have 5 clinical trials in oncology. The lead indications are pancreatic cancer and non-small cell lung cancer. We've seen very exciting data in those indications, which has been, let's say, inspired us to broaden the development a little bit to look into more indications. We also have a very solid IP position and with not only IP on our molecules, but also on the target in itself when it comes to cancer. We have a good cash position. We had about 60 million US on a bank end of last year, and that will take us at least until mid next year, and we can extend the runway if needed. So if we go to the next slide. So when it comes to cancer treatment, there are lots of opportunities for a drug like nadunolimab, the lead compound. But we're taking the approach to look into resistance mechanisms and where we have a really interesting opportunity. So if you, to the left, look at a tumor which is, let's say, more or less newly diagnosed, almost by definition, it's immune suppressive because otherwise our immune system would attack it and eradicate it. And obviously the most logical approach, if it's possible, is to start working with immune therapy to, let's say, make it more visible to the immune system and potentially eradicate it. The resistance mechanism which comes up is that you very often stimulate T cells to eradicate the tumor cells, and then very obviously mechanisms trying to control an overactive immune system like immune suppressive cells and MDSCs. And MDSCs overexpress our target and is, let's say, biologically controlled by this system. If immune therapy is not possible, chemotherapy is a very good choice. And chemotherapy is basically designed to kill cancer cells. If cells are exposed to danger, there are automatic signals that will come into play. That's more by nature. And one of those signals is interleukin-1 alpha, which is a very primitive way or an old-fashioned way of for let's say cells to defend themselves. And again, interleukin-1 alpha is the ligand for the signaling mechanism we are utilizing, and also targeted agents would involve IL-1RAP when it comes to resistance. So we believe that we have a very universal tool here to actually increase the efficacy of existing therapies. So if we go to the next slide, our pipeline is very much, when it comes to oncology, very much based on what we've seen preclinically and this hypothesis of sensitization of existing therapies. So in pancreatic cancer, we're working with first-line patients being treated with gemcitabine Abraxane. We've seen really interesting results, and there is much more to come to this story. And we are, based on those results, entering a pivotal trial in the US, starting up later this year if everything goes as planned. In lung cancer, we've presented the first data set in combination with cisplatin gemcitabine. We, based on preclinical data and also that's our own data, suggest that platinum-based compounds there's something unique here when it comes to activating the IL-1 system in the defense. So again, that's one way where we've seen interesting results, and then we expanding, we're including more patients with lung cancer, and we'll come back to that. But we're also broadening the development into other cancer forms and other chemotherapies to really get the first indications of how broadly this type of therapy can be used. We're also doing some studies with immunotherapy, and we'll come back to that as well as we are presenting the first data at ASCO here. Our second program is called CAN10. It's being developed for myocarditis, systemic sclerosis, and is entering clinical studies early next year. And then we have a platform of new antibodies, and we will come back to a little bit why we have so many opportunities. So next slide, please. And then we can go to the next one. So when it comes to IL-1RAP, it's, as I said, it's part of the interleukin-1 system. And the way it works is that there are two forms of interleukin-1, alpha and beta, and this is really, really important and really differentiating our compound from competition. So it could be either IL-1 alpha or IL-1 beta, and both of them have relevance when it comes to cancer, and both would be found in the tumor microenvironment. And when they bind to the IL-1 receptor 1, there is a freedom conformational change, which leads to binding to IL-1RAP. And then there is a signal, as you can see to the left. Our antibody nadunolimab binds to IL-1RAP in a way so it cannot dimerize with IL-1R1, which means that there is no signal whatsoever in this context. And this signal typically would be immune suppressive when it comes to cancer. Since we also have an IgG1 antibody, if you look to the right, it has ADCC activity, so it will stimulate NK cells to eradicate cancer cells. And then through the blockade of IL-1 and IL-1 alpha and beta, it will inhibit myeloid cells, it will inhibit the crosstalk between fibroblasts and cancer cells, and it will have an effect on angiogenesis. So if we go to the next slide, so here's one example where we are combining with chemotherapy, and docetaxel is interesting because it's widely used, also called Taxotere. It's also a chemotherapy where IL-1 beta blockade has been studied clinically and not reaching primary endpoint. So in this system we're working here, it's a mouse system where we treat mice with the docetaxel, and as you can see to the right, there is a treatment effect. The tumor growth is affected. If we add an antibody against interleukin-1 beta only, there is no additive or synergistic effect with the chemotherapy. But if we add our antibody, which then has the more broader mechanism of action blocking both IL-1 alpha and IL-1 beta, and also having the ADCC, clearly we have a synergistic effect with the chemotherapy. And one reason here is that chemotherapy docetaxel increase IL-1 alpha in the tumor context. So if we go to the next slide, what we see here is a similar type of experiment, but we used platinum. And I think the important message here, which we can look at in the slices in the middle, What you can see is if you treat animal with gemcitabine cisplatin, which is a combination we're using in non-small cell lung cancer, there is an increase in interleukin-1 alpha. And once we're activating interleukin-1 alpha, there is an expectation that interleukin-1 beta and the inflammasome would get activated. And you can see that to the right, as a green staining, that we do get an upregulation of this system in the tumor stroma. So in principle, what is shown here is that both interleukin-1 alpha and interleukin-1 beta is increased in the tumor microenvironment by the chemotherapy. And if we add our antibody, as you can see to the right, there is a synergistic effect on the tumor growth. So if you compare the two lower lines, the gray and the orange one, you can see that there is a clear difference here. And interestingly, we also counteract some of the side effects associated with chemotherapy, which can be, let's say, explained by our ability to block IL-1RAP and the IL-1 system. So there are lots of opportunities here, not only to treat the cancer, but also to have an impact on quality of life. And there is a relevance here, which we'll come back to. So next slide, please. So what we're showing here is the first clinical data set in pancreatic cancer as a combination therapy. And obviously a big data set, but the most important findings is that when you look at the survival, these patients have lived much longer than is expected from chemotherapy alone. So we have a median survival of 12.7 months, which is approximately 3 months longer than you would expect from chemotherapy alone, which is clearly a really interesting observation. The progression-free survival is also longer, about 1 CT scan, so about 2 months longer than expected from chemo alone. And what you can see in the waterfall below is that the responses are deeper, and it's not obvious from this one, but we've seen that they are perhaps more long-lasting when you would expect from chemo alone. So again, really pointing out towards what we're trying to achieve, an increased efficacy of the chemotherapy, which is used anyway in these patients. We've seen also that patients benefit in 2 different ways, so either through classical responses or through some kind of delayed responses after initial tumor increase in size. The tumor has started to shrink after 2 months, and these patients are really benefiting from the therapy and live a long time and longer than expected. So this is a really nice result, and this is based on 33 patients. We have now treated 73 patients, and the results on these 73 patients will be presented at ASCO in 2 weeks from now. So next slide, please. Safety profile, again, very interesting. And if we start on the positive side, one of the serious side effects with the chemotherapy gemcitabine Abraxane is that patient develop neuropathy. In fact, in the phase 3 trial leading to approval of Abraxane, it was 17% of the patients developing grade 3 or higher neuropathy. We have not seen a single patient in this initial group of 33 or 36 patients that developed neuropathy, despite that they've been on treatment longer than in the phase 3 trial. So this is really exciting, and maybe one aspect why patients are doing so much better. They cope with the chemo and can stay on therapy much longer. On the other side, we've seen that neutropenia is higher than expected, and it was to some extent expected because IL-1 blockade is already known to have an effect on neutropenia. Nevertheless, this was the first trial where we examined the combination, and it was important to characterize the side effects. But let's say moving forward, we know that there is a therapy called GCSF to counteract neutropenia, and we're, let's say, have learned more how to use GCSF and have built that into the treatment concept. So going into the phase 2/3 trial, the potential pivotal trial, we're obviously going to use GCSF to control the neutropenia. So next slide. So the upcoming trial then is a great opportunity for Cantargia. It's a trial run in the US together with the leading clinical centers, and it's run by PanCan. It's going to be an initial phase of 100 patients in each arm. If taken forward, it will add another 75 patients in phase 3 trial design. And we will expect to reach or to have results no later than 2027. It's a design where 4 different active compounds are used in different arms towards one common control arm, and it depends a little bit on how many of these arms that are advancing to a phase 3 part. It's also a trial which is financially very attractive for Cantargia. We are only paying a fraction of what a normal phase 3 trial would cost. So next slide. And lung cancer, again, we see stronger results than expected from chemo. We have a response rate of about 50%, which is about twice as high as expected from chemo alone. And this is in patients that have not been treated with chemo before. Progression-free survival, about 1 CT scan longer or 2 months longer than expected. And side effect profile similar to pancreatic cancer. So, and these results will be updated again at ASCO and will be, let's say, much more mature for long-term effects as well. So next slide. So here we looked at what are the subgroups. So there are lung cancer is far more competitive than pancreatic cancer, and we need to, let's say, find our niches for development. And interestingly enough, we see a much stronger signal in non-squamous non-small cell lung cancer, which is the biggest subtype, and especially in patients that have received immune therapy ahead of our therapy, where lots of patients respond. So we're building on these findings to get a robust data set before we entering randomized trials early next year. So next slide, please. And the third trial, which will be presented at ASCO, is then the combination trial with Keytruda. This is the first-in-man trial, 15 patients, really designed to make sure that we have no unexpected side effects when you combine the 2 drugs. And data will be presented at ASCO as well. So next slide, please. So, and then next one. So when it comes to CAN10, then, which is second program, it differs from the lead program in that now we have an antibody not only blocking the IL-1 system, but also IL-33 and IL-36, which is signaling through IL-1RAP. And this leads to lots of unique opportunities within the autoimmune inflammatory disease field. And we decided to focus on systemic sclerosis and myocarditis. And if we go to the next slide, we can see that to the left, that biologically all these, both IL-1 alpha, IL-1 beta, IL-36 and IL-33, contribute to some of these inflammatory/fibrotic diseases. And to the left, it's an illustration of systemic sclerosis. And if you look to the right, you can see that we do have very interesting effects, both in preclinical disease models of systemic sclerosis and myocarditis. And especially in myocarditis, you can see that if you do not treat these animals, they get a very quick deterioration of heart function. If we add our antibody, the heart function remains intact. So then we can go to the next slide, which is the financials, and then go to next. So this is just what I said about the cash position, about 60 million US end of Q4. We have a market cap around 1.3 billion SEK or 140 million US. And we have a strong ownership, so we have lots of Swedish institutional investors backing us. As you can see, both pension funds and bank funds. And if we go to the next slide, here is really the upcoming news flow. So in the near term, we have 3 presentations at ASCO, both for pancreatic cancer, non-small cell lung cancer, and the Keytruda combination. Very excited to go there and present new data. It's quite a lot of new data. We are in the process of starting a phase 2/3 trial in pancreatic cancer together with the US consortium PanCan in the Precision Promise trial. We have lots of supportive translational preclinical studies ongoing, and whenever we have something meaningful to present, we will be transparent and do that. And then we have all these new trials which are up and running, studying, let's say, how we're broadening to see which combinations work best. And you should expect the results or from that during the second half of this year. And then we have a CAN10 program, which is advancing well towards the clinical trial early next year. So by that, I'd like to finish off with the last slide, which is more a summary of the company. And I'm very happy that you showed an interest to listen to the company, and I'm pleased to take questions. So thank you so much.
Thank you very much, Göran, for such a nice presentation. Now we have time for a couple of questions. You have shown very nice data on how important it is to inhibit the signaling processes mediated by IL-1 alpha and IL-1 beta at the same time. Is there any other company out there targeting these the signaling mediated by these 2 molecules out there, or you are the only ones?
No, so there are other companies looking into blocking both alpha and beta, but both in autoimmune inflammatory diseases, but also in cancer. But no one is using an antibody concept in a way that we're doing. So it's more just blocking the 2 cytokines and not utilizing the ADCC component. Also, by having an antibody, we have an advantage of, let's say, 2 to 3 weeks half-life of a molecule, while other concepts may have a very short half-life.
Okay, very, very interesting. And my next question refers to your PanCan network inclusion. What are if you could explain us, what are the key benefits of being included in such an important network?
Yeah, so basically, the PanCan trial, when started, it had about 15 of the leading US centers, and it's really the top US centers like Dana-Farber, the Memorial, MD Anderson, Johns Hopkins, etc. And it's adding on other centers. So basically, we get both, let's say, the attention as well as, let's say, the opportunity to get input from leading US physicians. So that's one very important aspect. The second aspect is the financials, which we calculate that we're paying about somewhere between a third and half of what this type of clinical trial would cost if we were financing it alone.
Okay, Göran, thank you very much for being here with us today and for such a nice presentation.
Thank you so much. Pleasure.
The next company to present is Calliditas, and I'm pleased to be joined virtually anyway by the CEO of Calliditas, René Aguiar-Locander. René, I'll pass the word over to you for the presentation.
Thank you very much. Thank you to this presentation from Calliditas Therapeutics. If you can please go to the next page. I just want to draw your attention to the disclaimer page, as I'm sure you're used to, just with regards to kind of forward-looking statements. And I refer you to all of our public filings, including risk sections, etc. So we can take the next page, please. So a brief summary of Calliditas. We're a commercial stage biopharma company. We're focused on orphan indications with initial interest really in renal and hepatic diseases where there's a significant unmet medical need. For the last decade or so, we've been pioneers really in one of such rare disease called IgA nephropathy, which is an autoimmune disease of the kidney. We are the only ones who actually had a large positive phase 2b trial. It was published in The Lancet. We also completed our phase 3 in 2020, and both of these clinical trials met both primary and key secondary endpoints. We filed for approval last year and did get approval from the FDA, which is the first and only time actually that there has been an approval in this rare indication, which of course we're super excited about. We have a strong pipeline. Obviously then also focused on orphan kind of liver and kidney indications. It's a first-in-class NOX inhibitors platform, and I'll talk about that a little bit later in the pipeline slide. We have a broad set of investors, both from kind of the US and Europe. We are listed in both places, and our cash position as of the end of the first quarter is 825 million Swedish crowns. So next page, please. So as I mentioned, we had an approval for our first commercial product. It's the brand name is Tarpeyo. And as I mentioned, it's the first and only FDA-approved drug. It's also specifically designed for really kind of locally focused on the origin of this disease, because it's not really, it is a kidney disease, but it doesn't actually start in the kidney, but it starts in the gut, which is the presumed pathophysiology here, which I'll take you through in a minute. We launched this at the end of January, and it was really based on, the approval is based on the results from Part A of our NefIgArd study that reported out reduction in proteinuria, which was the endpoint of 31% versus placebo, 34% on intention to treat basis versus 5% for the actual placebo arm. And it was generally well tolerated. Next slide, please. So here's our pipeline. So as I mentioned, obviously the Nefecon program, or Tarpeyo as it's now being marketed in the US as, it was read out in 2020. That study is, however, still ongoing and it's still blinded, is in a kind of phase 3 format because there's a confirmatory readout that is required, which is always the case for these type of surrogate marker approvals. And that study is planned to read out in 2023. We're commercializing, as I said, ourselves in the US. We have licensed it ex-US. We have a commercial partner in Detta in Europe, which we did licensing agreement last year. And in 2019, we entered into a development and commercial agreement with Everest Medicines for Greater China. In terms of the pipeline, as I mentioned, it's a first-in-class NOX inhibitor platform. We are running a pivotal phase 2/3 trial in primary biliary cholangitis, an orphan indication for the liver. And we're also doing a proof of concept study, which also is up and running in head and neck cancer. This is more to actually just characterize the the mode of action, if you like, of setanaxib. And there's been a lot of work done on the clinical side within head and neck cancer that we're now kind of doing a study to translate into clinical data. We also have a couple of investigator-led studies, one in DKD, one in IPF. And so the IPF study is also a phase 2 study, placebo-controlled, randomized, and is ongoing. Next page, please. So IgA nephropathy, as I mentioned, is an autoimmune disease of the kidney. And there's a genetic predisposition that's required, but it is insufficient. So there are triggers required, whether they be environmental or bacterial or dietary. And we know that patients are really kind of diagnosed generally in their 20s and 30s in the Western world. And more than 50% are at risk of just continuing to, they're kind of the the relentless trip really towards end-stage renal disease. This can take about 10 to 20 years, so it's a fairly slowly moving disease. And this is because it's attacking really the glomerular filtration capacity of the kidney, and we have about 1 million of those glomeruli in each kidney. And what we do know is obviously as this disease sets in, it impacts this kind of filtration mechanism, and you start spilling protein into your urine or blood into your urine. And we know that the higher levels of patients that have proteinuria, or those patients that have higher levels of proteinuria, have a much worse progression and outcome expectation. It is an orphan disease. We're estimating the US patient population to be between 130 to 150,000, with about 200,000 in Europe. In China and Asia, for reasons that are actually not well established, this is not an orphan disease, but actually quite a common kidney disease. Next page, please. So the pathophysiology, as I mentioned, so this actually starts in the gut where we have the majority of our immune system. And specifically in the part of the small intestine called the ileum, so the distal part of the small intestine, where we have a large concentration of something called Peyer's patches. So lymphatic kind of nodes with B cells in there, and they produce these secretory IgA antibodies. And they're meant to be there in the lumen, but in this particular disease, they find their way out into the systemic circulation. And as they do that, they are considered immunogenic. So there's an autoimmune response from the body, which creates these large complexes of autoantibodies, IgG, IgA autoantibodies that create these large complexes, which ultimately then deposit in the kidney. And they deposit in these kind of filtration mechanisms. It starts a kind of an inflammatory cascade, which then starts leading to fibrosis, necrosis, and ultimately leading to the need for dialysis and transplantation in the form of end-stage renal disease in a large number of patients. So next page, please. So what is then Tarpeo and Nefecon? It's designed really to target this origin of the disease. It's a capsule that has an enteric coating that keeps it intact as it goes through the stomach and most of your small intestine. And based on a pH, it releases these beads, which are triple coated with a variety of polymers, as well as the active ingredient budesonide. So budesonide you might be familiar with from Pulmicort. It's a very similar approach. It's a local treatment of the mucosa in the lungs. In this case, it's a local treatment of the mucosa in the gut. And what we know about budesonide is obviously well established. We know a lot about how it works and its safety profile because it's been used in these other indications. And what we know about budesonide is that it has a poor bioavailability. That means that very little passes into the systemic circulation. About 90% is cleared in first pass metabolism. And that's what makes it a local, potent local kind of active kind of immunosuppressant and is not really used for systemic treatment. So this actually creates all of these polymers, et cetera, creates a very differentiated release profile. So it really releases it in the very specific place where we want to and need a very high effective dose. And so you get a very kind of high uptake, and then it actually just kind of falls down kind of quite quickly. So it really kind of delivers in a very targeted way. Next slide, please. So this was our phase 3 trial. As I mentioned, we read this out in 2020. It was a daily dosing for 9 months orally with 16 milligrams of Nefecon versus placebo. And in the kind of efficacy and safety readout, included about 199 patients. And the endpoint was a reduction in proteinuria. And what we saw there in the treatment arm was a 31% reduction in the treatment arm versus a 5% reduction in the placebo group. And on an intention to treat basis, as I mentioned, that grew to 34%. For those patients who had reached 12 months in this kind of protocol, so they had been off drug for 3 months, what we saw is that the proteinuria continued to significantly reduce, and for those patients reached 52% versus the placebo group of 7% at that time. Secondary endpoint, very important. Obviously, this is the underlying kidney function. This is obviously what it's all about. Proteinuria as a symptom. and obviously has some correlations here, but obviously it's EGFR, which is the underlying kidney function that we're trying to protect. And what we saw is that in the treatment arm, there was a stabilization of EGFR, whilst in the placebo group, there was a continued decline of about 7% over the 9 months, or about 4 milliliters per minute. And it was statistically and clinically, obviously, significant on both of these. So next page, please. In regards to safety, so this is from our USPI. So basically, it's generally well tolerated. Safety profile was in keeping with the phase 2B. Majority of adverse reactions were mild, moderate. No adverse kind of clinical effects on the cardiovascular metabolic system, no severe infections, etc. So very much what you would expect from budesonide, which, as I said, is a very well characterized active ingredient. Next page, please. So in terms of the US market overview, as I said, so Tarpeyo, obviously the full indication is that it was approved for reduction of proteinuria, in patients at risk of rapid disease progression, generally characterized as UPCR equal to about 1.5 gram per gram. And obviously, as I'm saying, it is an ongoing confirmatory study that's still continuing as well with regards to the ultimate impact on EGFR. So this was approved on the December 15th. So if we go to next page, please. So in terms of the market opportunity here, the US prevalence is estimated to be between 130 and 150, as I mentioned. And we know that 50% or more actually potentially then progress or continue to progress to end-stage renal disease, despite optimized supported care, which in this indication really is blood pressure lowering agents. So RAS blockade. And obviously, this has not only a huge cost, obviously, to the patient and the family, but obviously also a health economic cost, which is very significant. Spherix was a third-party research house, actually did quite a lot of research last year, published it in terms of looking at chart audits in IgA nephropathy patients and about 190 nephrologists. And what came out of that is obviously that there is a significant medical need. More than half, because they found that there were few or no effective treatment options. This is obviously before the Tarpeyo approval. And actually from their patient population, they said that they expected 65% of their patients to progress to dialysis. And in terms of the profile, the data that was available at the time with regards to Nefecon or now Tarpeyo, the ones who were extremely familiar with this, because obviously there's quite a lot of data out there, look to actually prescribe this in a majority of their patients. So next slide, please. So what have we actually then achieved? We just reported our Q1 results yesterday. And so, as I mentioned, the first sale and prescription filled was actually at the end of January 2022, which is actually very quick from the actual approval. And what we had reported is that, we have the salespeople, we have about 40 of them in the market in the US who were there just at the end of January, fully available to kind of contact nephrologists. Have so far actually reached about 4,000 nephrologists. And so what we can report from the Q1 is that we have 134 enrollments. So this is prescriptions written by 111 unique prescribers. So this really kind of covers the 9 weeks or so that we have been able to promote the product in the US, and it resulted in about 18 million Swedish crowns of revenues, about $2 million in terms of net sales. And we are super excited about this, and we look at the number of unique prescribers in this short period of time as an extremely good sign of adoption. And we're very proud about the Q1 results that we posted. And obviously, we're also seeing, acceleration of these kind of trends as we're kind of going through and having a higher awareness, being able to kind of contact more physicians and more physicians learning about the fact that there is an approved product out there now for these patients. Next page, please. So obviously, we don't get to this situation without a lot of hard work. And obviously, we have spent about 3 years preparing for this, both in terms of we have a fully established platform in the US that includes, obviously, medical affairs. It's been in this, we've had them for a couple of years being out there talking about the pathophysiology, really kind of discussing this disease with the nephrologists. Obviously, also market access, super important, have that in place early. To really kind of prepare for all of the research, all the kind of health economic studies, all of the interactions with payers, which in the US is a fairly complex system to understand and penetrate. And we continue, obviously, with these engagements. And so in the US, there are thousands of kind of plans and insurances. But obviously, there are some key kind of targets in terms of large kind of payer organizations that we continue to interact with. And it takes about 6 to 9 months for any drug that's approved in the US to get through these processes in terms of P&T committees, so pharmacy and therapeutic committees, to review this data and actually then decide how they're going to handle this new product in their kind of specific plan. Next page, please. And how does the actual system then work? So obviously we have something called Tarpeyo Touch Points, which really kind of went live just after the approval. It is a full service patient and provider kind of hub with a lot of support programs. And really what it is, it has like a dedicated care navigator team that's there for us with kind of nurses and pharmacists and distribution, et cetera. And this is really what you need. This is a specialist product. This is not a broad kind of market. Remember, this is an orphan indication. It really should be nephrologists that prescribe this in this kind of drug for their patients. And you really need to, we have a very streamlined kind of supply and distribution chain, which we felt was appropriate for this product. And really in order to provide all of these services around the prescribers and the patients during this time that all of these insurance plans go through their review and establish how they're going to ultimately deal with the product going forward. So next page, please. So as I mentioned, we have 40 sales reps. They all have rare disease, specialty background, and actually a very large number of them, 70%, also come directly from the nephrology market. As I mentioned, we have account managers, obviously still dealing with targeted payers. And we have a team that is extremely experienced. So maybe this company as such has not done a commercial launch before, but the team that's in place, that we've had in place for quite a long time now already, have a lot of experience in terms of launching specialist drugs in the US. Next page, please. As I mentioned, outside of the US, we have partnered with Detta in Europe and with Everest Medicines in China. We are expecting an opinion with regards to an EMA opinion for conditional approval potentially at the end of this May meeting from CHMP. And with regards to China, there's a plan for them to file their regulatory file in China in the second half of this year. Next page, please. Next page, please. So in terms of our pipeline, we talked about the NOx inhibitors. So this is really a group of kind of enzymes that are solely dedicated to producing reactive oxygen species, or ROS. This is their primary and only function. And our kind of clinical candidate, which is the first clinical candidate from this group to ever kind of reach clinical stage, really focuses on NOx1 and NOx4. So it's anti-inflammatory and anti-fibrotic, really. Next page, please. So there's been a lot of work that's done preclinically and with animal models. A lot of that's taken place in the head and neck space and cancer. And what it's been shown is that for solid tumors that have a high density or concentration of what's called cancer-associated fibroblasts, actually existing immunotherapy is not very effective. The response rate is only about 20%, for example, for the kind of the first line pembrolizumab as a monotherapy, which is quite poor. And we know that NOx4 is highly overexpressed in these CAFs, and it drives these kind of myofibroblastic activation in these tumors, which results in the fact that these tumor-infiltrating lymphocytes can't get into the tumor, and therefore immunotherapy is not effective. What's been shown in preclinical and animal models is that if you actually dose acetonaxib, it seems to have this unique capability of actually reversing this and kind of quieting down these kind of fibroblasts, and actually thereby allowing infiltration of these kind of lymphocytes, and thereby also significantly improving tumor reduction, overall response, overall survival, etc. Next page, please. And this has been proven. It's actually in one of the journals where this material has been published, has also in the American Cancer Association, and the journal's been selected as one of the top referenced kind of articles in 2021-22. And we're super excited about being able to hopefully take this into the clinic. So next page, please. So we are started, as we just again reported this week. We have dosed our first patient in our phase 2 trial. This is a randomized, placebo-controlled. So it's not an open handful of patients. It's really going to be about 50 patients, randomized, placebo-controlled, and we would look to hopefully have some interim biomarker data with regards to the impact on calves before the end of the year, and the trial will be reading out next year. And there'll be a biopsy before and the biopsy after about 3 cycles of pembrolizumab to really kind of look at the potential impact of setanaxib co-administration with regards to these cancer-associated fibroblasts. Next page, please. We're also running a pivotal phase 2/3 trial, as I mentioned, in PBC. Again, it's a double-blind, randomized, placebo-controlled study. It has an adaptive study design. So there's a dose-ranging portion of it that then goes straight into a phase 3. And so it's a 52-week treatment with setanaxib at 1,200 milligrams or 1,600 milligrams daily. And we would look at having a interim kind of futility analysis conducted in 2023, so next year, when the 99th patient has completed 24 weeks of treatment. And we would expect the full trial to read out either end of 2024 or beginning of 2025, obviously subject to recruitment and general kind of macro conditions. Next page, please. So these are really some of the summarize really of some of the upcoming news items that I've already covered. So obviously there is we are up in the agenda for the May meeting for an EMA opinion from CMHP. And we are then obviously subject to an opinion that would then be approved by the Commission about a couple of months later. So in Q3, we would expect that to take place. And then that could result in a commercial launch in Europe by Stada, obviously our partner, before the end of the year. As I mentioned, there's a regulatory filing for approval in China, which we're expecting to take place the second half of this year. We're obviously going to continue to ramp our commercial presence in the US and also to actually have this interim analysis or proof of concept head and neck cancer study before the end of the year, hopefully, again, obviously subject to recruitment and general macro conditions. So with that, I think we have finished the presentation.
Great. Thank you very much for that presentation, René. A few questions then, perhaps on this Trump opinion expected any day now, potentially. Considering or looking at the labeling in Europe, your thoughts there, assuming approval, obviously. Is there any reason to think that it would be sort of materially different from the FDA label you have, with this sort of wording around risk of rapid progression being generally UPCR score over 1.1 grams per grams?
Well, I mean, at the end of the day, it's always kind of interesting to see what the label actually comes out to at the end. But obviously, I mean, our assumption is that, these agencies collaborate, they talk to each other. This is quite a significant event because obviously there had never been an accelerated approval given by that FDA division ever on this surrogate endpoint. So our assumption is that there has been discussion and kind of between the two agencies. So I guess we wouldn't be surprised if that's the case, that it's a similar label in the two places.
Okay, and for the part B readout in 23 that you're using to convert from accelerated to full approval, what is it that you have to achieve? What is the sort of the bar that you're looking for there in terms of a readout to make that conversion?
So the endpoint of that is, so it's 360 patients in total. So they would have had 9 months of treatment and 15 months of follow-up. And the point in that study is eGFR. So it is the underlying kidney function over that kind of 2-year period. So that is the endpoint that's been agreed with the agencies. And in terms of exactly what exactly they would be looking for, they have not wanted to share with us or with anybody else. So we're just going to have to, just like we did when we were the first ones to get the approval, I think we're going to have to see what they are really looking for in kind of discussions with them when we file. Okay,
gotcha. And then on Q1 sales, I was curious to hear whether there was any stocking versus sort of true demand. How did that dynamic look like in Q1, and what are the expectations for Q2? Should we be expecting some degree of stocking as well, or what's your thinking around that?
So I think so there's clearly always some stocking, because this is actually where you're giving patients only like 1 month, and so you have to get a refill obviously kind of month to month. So everybody, there has to be some stocking, otherwise you can't fulfill demand. But obviously the difference here is that it's a specialty product, so it's only really one pharmacy. So if you had a general product, you would have basically, it would be a very large stocking effect potentially, because every single kind of pharmacy would hold some stock. So in this case, it's actually not something that we would expect, to be a major component of this. But obviously there needs to be some stocking because of the kind of the refill requirements.
Okay, alrighty. And a question on sort of pricing and formularies. What can you say on pricing, and particularly on how you've been working with discounting as you try to get onto as many formularies as possible?
So actually we haven't been rebating.
Okay. All right, that's clear. Thank you. And perhaps just one last one then on uptake. I think in the Q1 call, it was said that physicians aren't necessarily calling out and getting all their patients in to switch, but it's rather when patients come for their regular visits, sort of every quarter or so, patients are assessed, and if eligible, then potentially switched on to TARPEYO. Does that sort of regimen of patient visits every quarter or so, does that imply that in a quarter, more or less, from now, most patients would have been sort of assessed for eligibility to TARPEYO, or how should we be thinking about that?
I mean, I think what we said is obviously the core kind of target market that we're targeting really is about 65 to 70,000 patients. So obviously it'll take quite a long time, before necessarily all of those patients have, kind of been to see their physician. And also obviously there is there's never been anything approved in this indication before. And it takes some education and understanding and awareness from physicians even to kind of get to the point where even they're aware of the fact that there's a product that's approved in this indication. But this is why we're so kind of encouraged by the fact that the adoption, from so many physicians seems to be quite quick. I mean, so we have 111 unique subscribers over this kind of 9-week period. So I think that really tells us that obviously they are kind of, there is a lot of awareness, and they are keen to really kind of try and get their patients onto this drug. But I think, realistically, I think we are going to see, and as we're seeing acceleration in terms of the trends, and I think we'll continue to see that for a while as actually more and more physicians, become aware and start prescribing the drug to their patients.
Great. I might squeeze in maybe just one final question on KDIGO guidelines. Do you have a sense for when they are likely to be updated, and presumably TARPEYO would be included there, given the approval? But then the second part to that is sort of given the prescribers being not overly numerous, it's sort of a specialty indication. How should we be thinking about sort of the material impact of a guideline inclusion for TARPEYO here? Presumably most prescribers will be aware and sort of have TARPEYO on the radar already. Do you think it will be sort of meaningful in terms of the uptake?
Yeah. I think it's always meaningful to be included in the guidelines. I think it's always kind of something that all companies really, try to be, and I think it is impactful. Generally, for guidelines, there's some indications that might be different, but generally, if you have a disease that is not, I would say, kind of, short-term lethal, obviously, where there is some time in terms of, the progression of the disease, these guidelines tend to kind of really rely on published, published journals, articles, research, etc., before they actually include anything in their guidelines. And so we are assuming that, guidelines will be updated as kind of publications become available, peer-reviewed kind of data becomes available, etc. And they tend to not be maybe super quick in terms of how they're updating their guidelines, even when there are approvals. So I guess we're expecting that we have to be a little bit patient to get into the guidelines. But I think you're absolutely right that from a kind of, launch and commercial perspective, It's always very helpful, but it's certainly not kind of a necessity in terms of having a very successful, launch and access to physicians.
Gotcha. Okay. No, we're all out of time. Thank you very much for an interesting presentation, Renee.
Thank you very much. Bye.
Welcome back. Our next company is Nanexa, a Swedish biotech company focused on developing new drug delivery systems. With us is here today David Westberg, the CEO. David, please, the floor is yours.
Okay, thank you so much. Thank you so much. So yes, I will present Nanexa for you and where we are today and a bit our plans going forward. And first of all, I would like to say what we are aiming at. So we have this drug delivery system that we call PharmaShell. And so we've done quite a lot of studies, mainly in animals so far, but so that we feel confident that we can take this drug delivery system, into clinic and further on into product development, and actually become a world-leading company within the field of long-acting injectables. So that is sort of our long-term aim for the company. I will present to you some of the features of the system, the PharmaShell system, and why we believe it is so strong. But first, a few words about the market. As you know, we are getting older and older, not just in person by person, but also the community as such. So if you look at this in 2015, there are about 12% of us, of the total population that is over 60 years of old. In 2050, that number is 21% and even a little bit more. So that gives us an older population, and with older population, we get more illness and more illness to treat, of course. And then I come to the long-acting injectables and how that fit very well with an aging population. So looking at in general, then long-acting injectables, what can that support in patients and in healthcare system, payers, and so on? Well, we know that the older you get, the more fragile you can be, and it's really important to have a convenient treatment, convenient treatment, so that you stay on the treatment actually, so that you don't get away from the treatment, and therefore long acting is very, very important. So also for the healthcare system, of course, having long-acting injectables, enabling fewer visits to the hospitals for treatment is, of course, of essence to keep the cost lower for the healthcare system. And that is also, of course, important for the payers and especially so, I would say, in the US market, but also, of course, in Europe and other parts of the world. So, what are we then doing? How do we create the long-acting injectables? We know that there are several different systems out there in the world that does this, but we have a totally different approach to long-acting injectables. And what we do is actually that we coat each and every particle, that is particles of different, it could be any API actually, any active pharmaceutical ingredient. It could be either small molecules, it could be biologics. And we coat them with an extremely thin coating, and that is what we call the PharmaShell. And that coating is a metal oxide that we put on the surface of each and every particle in a gas phase process. That process is called atomic layer deposition. And it's a very versatile process and also easy to upscale and so on and so forth, and has proven to give a lot of good features. And some of these features is that we can easily customize the depot length. In some instances, you would like to have a depot that's 1 week. In other, you would like one that is 1 month, or even longer, actually up to half a year to a year. And by altering the thickness of the coating that we put on, and also the material we have in the coating, the composition of the coating, we can elaborate in the process to make these depots either 1 week or 1 month and so on. Another very important feature of PharmaShell is that we can control the initial release. If you think of that you have a depot formulation that should last for 3 months, and about 30% of that is released the first day, you will get a huge plasma concentration of that specific API. We have the possibility to control that down to just a few percentage of the total dose. And that's extremely important, specifically if you work with very potent drugs. We have a drug load that is, in many cases, up to 90%, and that is sort of unheard of if you compare to other systems. Normally, that number is around maybe 30 to 40%, and this high number allows us to have long depot formulations in a very low volume, and that makes it easier for the patient and for the healthcare system to actually use the product. As I mentioned, we can work both with small molecules and we can work with large molecules like proteins, peptides. We can work with monoclonal antibodies and so on. So that is very important. And as you know, biologics is something that is coming, and it has been for many years now. So that is a market segment that we believe is very important for us. And also we have this flexibility in respect of where to administer this in the body. If you would like to have a systemic effect, you would administer it subcutaneously, most preferential. That would then allow you to have a smooth and continuous plasma profile throughout this period of time. You can always also think about administering it right into a tumor intratumorally, so that it actually dissolves within a tumor and release the cytotoxic compound in the tumor. Well, this is crowded pictures. What I want to say here is that we have a good patent situation. This technology is actually, it did evolve from the Uppsala University. We have a good patent situation. It has been the basic or the base patent has been granted in the US, Japan, China, South Korea, and Europe. And we're looking into India and Canada. And we have also a lot of new patent applications ongoing. And many of you know also that we have an infringement situation or patent litigation with Vitrovax in the US. And so far, this has gone very good for us. So we're in that process. I cannot comment on it that much more, but it looks good for us. Then our business then, how do we look at our business? Well, we have two different business models, I'd say, or we have one business model, but two legs. The first one is that we develop our own projects, our own product projects aimed at a specific indication. And that is these next projects that I have on the left side, next 18, next 20. Those two we have already started, and next 22, or whatever we call it, will be a third one. And our ambition is to have something like 3 to 4 different projects in different phases of development going forward. To combine that with another business strategy is also to out-license the technology as such to partners that have specific APIs they want to do a long-acting injectable of. And then we out-license the technology for that specific API to that company. And these deals will maybe not be as big as the deals of the product projects, but they will at least they will give us good money and anyhow. So looking at the market then, again, I showed you the population, and that's become more and more older people in the world. But looking at then the growth within drug delivery, we can see to the left that there's a huge growth, actually, about 7% yearly in that segment of the market. And these are numbers for biologics, I should mention. So there's about a similar picture for the small molecules. It's not limited to just one indication area, so it's all over the place, so to say. But we have chosen to work in our first 2 projects in the oncology field. And you see to the right hand, the next 18 and the next 20, they target quite big markets. And even though I will show you later the market on the lenalidomide case, the Revlimid, it has a huge sales today, about $11 billion. It will, when the patent goes off, erode to some extent, but even after that, it will be a huge market. Look then at our projects. The next 18, what we're trying to do there is to simplify the treatment for MDS patients. Today, they are having the Vidaza product treatment, and that includes injections, 7 injections, 7 days in a row, and then a resting period for 3 weeks, and then you go on with the 7 injections again. What we would like to do is to exchange those 7 injections with just 1 on day 1, taking away a lot of cumbersome traveling back and forth for the patients to the hospitals. Also, at the hospital, each time they get this syringe today, the Vidaza, they have to be pre-treated to skip nausea and so on. And they need to be remain at the hospital for a few hours until they go home. So that this just taking away 6 of the 7 injections will of course make a huge difference both for the patient and the healthcare system in convenience and in money spent on the treatment. So this is just to show the azacitidine use in America, in the US. What we foresee, and this is important to have this picture because it's an old product, but even though there are coming new products into the market, azacitidine will be used as a backbone treatment, and that is important because, as we know, this is an interesting indication for many drug companies, and they're developing new compounds to enter this market. But even though they do that, azacitidine will be the backbone treatment. If we then look at next 20 for multiple myeloma, what we see here, multiple myeloma, is a huge indication actually in the cancer field. And what we see here, even though it's an oral treatment, they need to take either 21 or 28 tablets each day for 28 or 28, oh, 1 tablet a day, of course, but either for 21 or 28 days, a little bit depending on what treatment they're on. But if you look at the right-hand side, it's documented that about or almost 40% of the patients do not take their medication as prescribed. And that, of course, does not give a good treatment effect if you don't take your medication. If you instead would allow you to have an injection done at the hospital once a month and take away all the tablets, it would ease up the treatment. Of course, the treatment will be done, even though so that is a way of getting a better compliance to the treatment actually. And patients are coming into the hospital once a month for taking samples and looking at the progression of the illness. If you look at the market for Revlimid then, or for lenalidomide, you see that the blue part here will decrease, and that is of course Revlimid, which is the original product with lenalidomide, which is the API in this product. But generic ones will increase, and you see this is an increase in the same way as for azacitidine. And also here it's important to know that lenalidomide is also the backbone treatment, even though new entities come into the market. And despite patent expiring, the lenalidomide market is expected to be somewhere around $2 to $4 billion yearly. So that is a huge market that we are entering also here. Then we come to some more internal information about Nanexa's pilot plant. We're very proud now to have that ready. It is a state-of-the-art GMP pilot plant located in Uppsala. It's about 300 square meters, and it allows us to work with both very toxic compounds like cytotoxics, and it allows us also to work with biologic, have aseptic processing with biologic substances. And that is important since biologics cannot withstand a sterilization treatment using irradiation, which small molecules does. So we have now built the capacity to take any project that I can think of through phase 1, phase 2, and phase 3, and potentially even use this facility as a launch facility for products to the market. So we're just moving into this, and actually we'll have our medical agency inspecting it in beginning mid of June, so that we get the permission to manufacture clinical trial material from this facility, as well as the older facility that we have. You see the number here of 1,000 square meters. That includes also R&D labs and our offices. So this picture of the organization, what I want to show here is that we have an organization that covers all the critical aspects of product development in the pharma field. And we have very experienced people in this team. I'm very glad to have that. People that have worked in the big pharma, people that have worked for doing many NDAs in both Europe and for FDA. We have business development, market analysis, and IP, all of these functions. And so we have a very strong team. Also, we have a very strong board of directors headed by Göran Andu, who formerly was the chairman also for Novo Nordisk, and earlier than that was the vice president of Pharmacia Upjohn. So we have a very good board of directors as well, and so we're a small company but very extensive experience. And why Nanexa as a last picture? Well, we respond to global need in the healthcare sector. The demographics show that. And we know that we have the possibility to be able to reduce the cost for healthcare and treatment. We also know that the products that we have actually can allow us to have a premium price on those products, even though there are generics on the market. We have a good patent situation, and we're building on to that situation, so that's good going forward. We have this own production facility, which allows us to work seamlessly in projects, taking it from ID to the market. And we have this team that I already talked about. So with that, I think I will leave for questions.
Thank you, David, for such a nice presentation.
Thank you.
And now we have time for a few questions. I will start asking you about your new plant.
Yes.
So soon it will be inaugurated, and this is exciting for Nanexa. And my question is, what are the first steps that you are planning to take there? Is it that you're going to push more collaborations, or you're going to push more like your own internal programs?
I'd say actually both. The first thing that will be done is actually our own projects. It's taking next 20 into clinics later this year. So that is an important step. We will have two different equipments, different scales from Applied Materials that we will install in this facility. And that will be done later this year, and also a larger one that's not needed right now, but it will be installed next year. But this allows us also to work with partner projects. And I'll say that it's important, since this is a new technology into the pharma field, we need to sort of prove that, yes, this can be handled in a professional way, in GMP, in not just lab scale, but also in pilot scale and also in large scale. So I foresee that this facility will also trigger that we get more interest from partners.
Yeah. No, that's very interesting. And as you said before, you will have the capacity of working not just with traditional APIs, you will also be able to work with biologics or protein, well, with a lot of other products. I was wondering about the technological complexity for you. Is it harder to work with a monoclonal antibody rather than with a traditional molecule?
Really, our process will be more or less the same, we foresee, because we are, but what is important is to, of course, keep the activity of the monoclonal antibody. We don't want to destroy that. And so, but as soon as you have stabilized it for instance, a spray-dried particle, I don't foresee that our process of coating that particle with our metal oxides will sort of destroy, or what you say, the monoclonals. But our process, I don't see it becoming harder than any other APIs.
Okay. And you mentioned that by the end of this year, you're planning to start your next 20 clinical trial. Could you give us a little bit on the overall picture of this trial? Is it going to be similar to the next 18 trial in terms of size and timelines?
Thank you. Yes, that's a good question. It will be somewhat different because this will be done in healthy volunteers. And that is a huge difference when it comes to recruiting to the study. It is a study where we will start with a low dose and then going up in dose. We will focus on safety, of course, and on PK. But it will be a study where we will get a good grip of how our formulation works so that we can, as soon as possible, get into a bigger study that will go into proof of concept. So there is a huge difference in the way that we're now working with healthy volunteers, which is different from the next 18 where we did it in patients.
Yeah. And going to next 18, that you're mentioning it, you identified the cause that basically caused the stoppage of your clinical trial. And you identified the solution, which is something very positive. How complex is to fix the problem that you found, and in order to more or less estimate a potential start again of the trial in 23? Yeah.
So we're currently running a few studies in preclinical studies to evaluate our new formulations. So far, it looks very promising for us. We will most probably need to tweak that formulation in one other step or maybe two other steps to really get to a situation where we can eliminate what we saw in the clinical study started last year. So I would say that I would need to have the results from what we're right now doing. Hopefully by the third quarter or so, have a very good grip of how we proceed going forward.
Okay, interesting. And regarding your next project, the next 22, I think you mentioned.
Yes.
So next 22, it will be born basically in the new plant, if I understand it correctly. And so there is a high potential that the product will not be a traditional molecule, but it could be potentially a biological.
Yeah, it could potentially be a biological. Absolutely, I can say that we're looking at maybe 3 to 4 different projects right now, evaluating them, and there are biologics within those 4. There are some traditional APIs as well. So we're still evaluating and talking to some of the key opinion leaders for those different indications. Try to gather all that data into a sort of a matrix and then make a decision by the end of this year.
Exciting. And then my last question, could you remind us what's your current cash position and your cash runway?
So our current cash position is, what I get it, about 80 million. And we will have We will have runtime at least for the first quarter next year and probably also for the second quarter next year.
Okay. David, thank you very much for being here with us today and for your nice presentation.
Thank you so much.
Welcome back. The next company to present is Vicor, and I'm pleased to be joined virtually by their CFO, Hans Jeppsson. Vicor is a Swedish company in sort of mid-stage clinical development with a lead asset, C21, that recently reported some promising data in idiopathic pulmonary fibrosis. I'll hand the word over to Hans to tell you more about it.
Thanks a lot for that, Adam. So my name is Hans Jeppsson. I'm the CFO of Vicor. So Vicor is developing a new class of drugs, AT2 receptor agonists, which we think, if data continue to hold through, could really lay a new foundation in medicine. So next slide, please. And next slide. So the team and the board, I think we have made three additions last year and built a complete management team with the addition of Jessica Schall, head of digital therapeutics, and leading those efforts. Mikael Nygård, vice president business development, and Åsa Magnusson at the bottom right in leading our commercial efforts in COVID, but also to build in that in the IPF program as we advance. So I think all in all, we are very strong on the science side and significant experience of working within rare diseases. Next slide, please. So this is just a glimpse of the current pipeline. We are starting at the top. Our COVID-19 program is in midst of phase 3, and we anticipate readout from that study the second half of 2022. C21 in IPF is where we really presented potential paradigm shifting results earlier this year. Is also more than halfway of that study, and we anticipate readout during the second half of 2022. We also earlier this year announced that we're extended also into a relatively closely related indication to IPF, PAH, which is PAH. And I think that's supportive with very strong preclinical data and really natural extension to our efforts with C21 in IPF. And we anticipate to start that one around Q1 2023 or Q4 2022. We also have another program in DTX, a cognitive behavioral therapy, treating anxiety. I mean, this is a deadly disease, so this is a really under-treated area, and we very recently announced the first patient included in the pilot study in the US. Also have a program in IPF cough, and we're still in the formulation phase there. Last but not least, I think we have a very exciting program with new selective AT2 receptor agonists, which, given the wealth of preclinical data that we have there, is really an unrecognized value that is really unrecognized and not valued out there. And I think, given the wealth of preclinical data that we have, and now when we are kind of building the data with C21 as a front runner, I think that makes so much more sense, and we're entering phase 1 with the first follow-up molecule or new AT2 receptor agonist there within very short. So I think that is very exciting. We're listed at the Nasdaq Stockholm main market cap of approximately $175 million. We have a cash balance of around 300 million SEK end of March, and I think we reported operating expenses of 93 million in Q1, and that is not subject to extrapolation, so that was higher than we anticipate going forward. So we anticipate with our plans that the cash runway is well into the second half of 2023. So that means well beyond key readouts in IPF and COVID-19. We have a very strong list of shareholders, generalists and specialists, including HealthCap, HBM, Invus, and others, which I think we have transformed and we are very satisfied with. Next slide, please. And next one. So we are part of, so the AT2 receptor is part of a well-known system, the renin-angiotensin system, which is well known because of the phenomenal success with the ACE inhibitors and the AT1 receptor blockers, which you see on the left side here, developed against hypertension. We see the AT2 receptor as an inducible regeneration and repair receptor associated with very positive effects, including, for example, antifibrotic, immunomodulatory, regenerative. So we really see this as a novel target with untapped potential. Next slide, please. So I'm not going to go into all of those potential multimodal effects through which the AT2 receptor activation are mediated. And we see 4 here, so it could be all of them or relatively 1 or more of them. So I think starting or focus on the top left, we have in the lung, the AT2 receptor is highly expressed on AT2 cells, and these AT2 cells or progenitor cells, they are involved in the repair of damaged alveoli, and they also fill a critical function in secreting surfactant, which we see here on the picture, and that reduces the alveolar surface tension and thereby makes it easier to expand the lungs. And I think that is extremely important to keep in mind when we now have disclosed the very exciting and intriguing interim data in IPF, to have that in mind. But also there are other potential mechanisms of action such as vascular remodeling and vasodilation, the second one at the top corner, through NO release, and also indirectly the NO pathways are impacting TGF-beta, as we see on the in the bottom left corner. And TGF-beta is a master regulator, and we see here also a direct effect of C21 on this master regulator. So I think all in all, the science is extremely exciting with the AT2 receptor. Next slide, please. And this is, I think, what is really unique is that there are more than 100 plus publications with C21 in different preclinical models, and probably thanks to the company being very drip-fed. Funded in the past. We have shared the compound with lots of academic research worldwide, but that has given us a wealth of data to choose from when we focus the activities. But I think also now when we're building the link between preclinical data and clinical confirmation, those preclinical data make so much more sense and gives really Vicore a great opportunity to lead that development with new in the AT2R space. So we can, for example, see on the left side here different organs involved, and if we only focus on fibrosis, for example, we do see prevention of fibrosis in a number of preclinical models and organs, for example, the lung, the kidneys, the heart, the blood vessels, the skin, and so on. And in fact, we have even seen in the preclinical models a reversal of fibrosis in the lung, which I think is very interesting given the data that we disclosed earlier this year. So I think all in all, I think we are just in the beginning of a very exciting journey with when we now build the clinical confirmation based on these huge number of preclinical data that is out there. Next slide, please. And I think this is, we see ATRAG or the AT2 receptor agonist as really a new class of drugs and has what we think the potential to be as successful as, for example, other drug classes like the beta blockers approved in some 50 indications or the SGLT2 inhibitors. So I think has very good prerequisites, and Vicore has really the great AT2 receptor expertise and is in a unique position to pursue these opportunities in these. So I think we are really as a front runner here, and there's not much competition out there in AT2 receptor agonism. So I think we're well positioned there. Next slide, please. So at C21, we have orphan drug status in IPF, which gives us 7 years in the US and 10 years in Europe, and we have also market exclusivity. It's worth mentioning that we also have several pending patents. Regarding the new compounds, we really see them, would like to emphasize significant growth opportunities, and they, of course, have patent protection well into 2040 and beyond, 2041 and beyond. And they have slightly improved physiochemical and metabolic properties, and we're very excited to announce that we are moving the front runner of those molecules into the phase 1 within short here. Next slide, please. And next one. So I think this is just a way to show how we work, and I think we do have a really patient-centric focus. I think that is important. I think there are learnings to be made from, for example, Actier, which was acquired by J&J back in the days for $30 billion, and they developed Bosentan and others in PAH, and didn't necessarily have better data than competitors like the big pharma companies, J&J, GlaxoSmithKline, but they worked very closely with the patients, patient organization, and were able to win the loyalty of those. I think this is what we think is important when we compete with big pharma, leading companies out there, Boehringer Ingelheim and Roche and others, really to take a 360-degree and work close to the patients and address the huge unmet need out there, not only in fibrosis, but also the anxiety, which is a big complication for these patients, the cough, which also impacts quality of life, and also the PH, pulmonary hypertension component. And PH is what ultimately kills these people. And I think if we can show or have an impact, as we do have in the preclinical models, I think it's really exciting in totality. Next slide, please. A little bit on IPF. It's a deadly disease, a life expectancy of 3 to 5 years. It's a common rare disease, so approximately 250,000 patients in the US and Europe. There are 2 approved drugs, and they generate some $4 billion in combined sales in 2021. Incidence is growing, market is growing. The 2 approved drugs, they slow down the rate of deterioration, but do not stop disease progression and do not reverse it. And they also come with significant side effects, including GI, such as diarrhea, appetite loss, weight loss, nausea, photosensitivity, and so on. So I think the patients do not, from a quality of life perspective, do not feel that they are better, but they experience the side effects. And one potential reason for that is observed that we know that approximately 40% of US patients are not on approved drugs. So that could be one reason. So therefore, we think that it's really a tremendous commercial opportunity here, just something that has a on par efficacy, but better safety profile. Next slide, please. Yeah, so this is just the preclinical data on the left, and we can just tick the boxes. We have just ticked the boxes there on the bleomycin model. We have also good data in the monocrotaline model, and to the right here we have in more disease-like with the precision-cut lung slices from IPF lung transplants. And here we see the effect on TGF-beta protein, as I mentioned before, which is considered as the master regulator in fibrosis. So I think we have ticked the boxes and also have relevant with this IPF lung transplants data. Next slide, please. So when we designed the phase 2 AIR trial in IPS, we thought that if we look at the picture to the right, if we just are on par or better than standard of care, but with a better safety profile, that would definitely be a potential game changer. What is important in these type of trials is that we have a secured IPF diagnosis or correct diagnosis of IPF, and we have Professor Davage at Brompton in the UK, who has been involved in the leading studies of BI, Boehringer Ingelheim, and other studies, and he is responsible for securing this IPF diagnosis. And I think we have quite high screening failure, and I think that's a good sign that we really secure the IPF diagnosis. Also, secondly, we have the primary endpoint measure is FVC, which is an objective measure, forced vital capacity. That's also the standard registration endpoint. So those 2 are very important to keep in mind in this study. So next slide, please. So this is the interim data that we disclosed in February this year, which I think was really a momentous day for the company. So what we see here is that there is a first stabilization period of the first 18 weeks, and then there is an increase in FVC, and that's really intriguing. I think comparing to other studies, even though you cannot directly compare to other studies, that increase in FVC is really unheard of. So that was, of course, very exciting, and it's important again to point out that it was safe and well tolerated, and no serious adverse events, including no GI signals. And I think also very important is to point out that even though it's a small data set, it has some caveats. Of those, if we look at week 24, those 9 patients, 5 out of 7 that we have data at 36 continued to improve, and 2 were flat. In addition, we used imputation, which means that we assign the untreated trajectory of those patients that are in the study. So they take the trajectory of untreated patients, which is, would say, a very conservative estimate that patients that have this pattern just for some reason just behave like an untreated patient. But even in that case, we are still on the positive side of the x-axis, so positive slope, which I think is fantastic. And I think this has, of course, forced us to, we have looked for a signal, and we see a clear signal here. Our data is maturing. We are continuing to include patients in the study, but this has really pushed us to think about the next steps. Next slide, please. And I think we have a very interesting business case here. We have an oral drug. It's well tolerated, no GI intolerabilities observed whatsoever so far. And this interim data suggests a stabilization and an increased FVC. So therefore, we have scheduled a meeting with the US FDA to discuss the next steps, which is scheduled in the midst of the summer, and most likely we'll have meeting minutes 30 days or so thereafter. So we are really looking forward to go back and present that after that meeting. Next slide, please. So very short on the ALMI, the DTX program. So as you can imagine, mental health issues are prominent in IPF because of it's a deadly disease. So anxiety is a big complication here. I think you can see this. We work together with Alex Therapeutics, and we include the first patient in the pilot phase very recently. I think it's a US study, and I think it really has been well recognized by patients, patient organization, and it's also a way to get closer to the US as we are moving into late stage, so to say. So I think it's really a tool and a way to get close to the patient organization, and key opinion leaders. Next slide, please. VPCR2 program, not going to say so much. We're still working on formulation, and we're making some progress there. Next slide, please. PAH is, as I mentioned, we have very exciting preclinical data, which and it's very closely related. The vasculopathy link and PH link between IPF, and I think it's a natural extension, and the market there is more fragmented. So 14 approved drugs, as big as the IPF market, but there are no disease-modifying treatments out there, so that really represents a significant market opportunity. We have looked at the Acceleron data, which Merck bought for $11-12 billion, and they were into phase 3 in PAH, and even though it's different studies, so a little bit apple and pears, but we think that we have at least as good or better preclinical data, what is disclosed, and what they have. So I think this is a very exciting opportunity to a natural extension of in addition to IPF. Next slide, please. Yeah, I think we can skip these preclinical data, just tick the boxes. And the total market for IPF and PAH, including those untreated patients, so we have a total market of $11 billion, which is really a sizable market opportunity. Next slide, please. COVID-19 in short, can have next slide. So we are not competing with antivirals or antibodies. We are on the ordinary scale 5 to 6, hospitalized patient, but not on mechanical ventilation. Next slide, please. So the current phase 3 program was backed by very positive phase 2 data that we disclosed in December 2020, where we see at the picture at the bottom left here that number of patients on supplemental oxygen was 11 on those on active drug versus 11 on placebo. And I think we really think that we could restore lung function is the way that C21 works here. And I think this builds together with the totality data that were disclosed before, is building confidence in C21 as we move forward. I think we can move to the next slide. And finally, we are in the midst of this phase 3 program. We up to 600 patients, and we are just in the midst of that study. Anticipate top-line data second half of 2022. Next slide, please. So just in summary, I think really the strong interim data suggesting an increase in lung function is really unheard of, and potential paradigm shifting. And we're also advancing the portfolio, moving from a one-trick pony back in the days only in C21 in IPF, but also expanding in C21, additional indications PAH, but also the new ATRAGs. So moving into a more platform company, multiple opportunities is very exciting. And finally, the strong cash position of 300 million end of Q1 takes us well into the second half of 2023. So next slide, please. I think that's it. Thanks.
Great, thank you very much, Hans, for that presentation. A few questions then. A first one on this very exciting data in IPF. If you're to play the devil's advocate a little bit, with this endpoint of the spirometer assess lung function, there is some degree of patient effort in those kind of endpoints. How should we be thinking about sort of the chance or the risk of that sort of patient effort having sort of biased or inflated the benefit in C21 in a study that's uncontrolled and unblinded, say?
It's a very good question. I think that if there was a training effect in that spirometry in FVC, we would most likely have seen that earlier in the study and not this late in the study. So that is, we have scrutinized and even used the devil's advocate on the data ourselves, but we do not think it's a training effect in terms of FVC, because we would most likely have seen that earlier in the study.
Okay, no, that's interesting. And another question then on that AIR study data, looking at comparable studies in the past with other drugs, how sort of predictive is that early restoration, or I guess in those cases stabilization of lung function for the long-term outcomes for a patient? Does that sort of predict quite well a more long-term benefit in terms of lung function and survival?
Also a very good question. I would say that what's really important with this study is that we have 24 and 36-week data. And what's comparable out there, for example, we can take Galapagos data, I think 17 patients, 12-week data. I mean, FVC there's a big variation, and 12-week studies is very unsure. We see that in our study ourselves. I think what's really ensuring in this study is that we have up to 36 weeks of data, and that is, I think, very, very positive that we have that long data. I think the company's plans back in the days was to run a 4-week study, biomarkers only, which wouldn't have give us anything. But now when we when we redesigned the study and were able to run this long study, I think that's very promising. And I mean, the registrational endpoints FVC over 45 or 52 weeks is, I think that's not far away from where we are, and we see this very nice data. So I think that's ensuring that we have this kind of relative long-term data compared to others. Boehringer Ingelheim, for example, at the ATS recently disclosed data for 147 patients published in New England Journal of Medicine, that they only carried out that data over 12 weeks. And they are now moving into what we think is the pivotal program. So I think we are comfortable, even though numbers are small so far. Data is maturing, but I think we are comfortable that we have this long-term data, so to say, relative to them.
Good. And a question that sort of, I guess, ties into what you've been saying in terms of that longer term data. In IPF, I understand that there's quite a predictable decline, annual decline, that most patients, or at least on a population level, you see this, I think it's 220 to 240 mils per year that you've been quoting that patients typically lose annually. But on a sort of patient level, I guess there is some degree of intra-patient, intra-day variability in their lung function. Again, with the relatively small numbers, is there a chance that the favorable results that you're getting now is sort of perhaps a luck of the draw in a way that these patients were sort of assessed on a good day? I guess the number of patients that you're seeing and the number of measurements would speak against that. But yeah, your thoughts on that would be interesting.
Yeah, I think it's of course a risk with small numbers. It's a random effect, but we do not think it's a random effect. I think it's very unlikely. It could, of course, always be a random effect with small numbers, but we do not think so. Yeah, I think that there is, of course, intra and variation from measurements to measurement, but also within. But on a cohort basis, the number that you refer to is what is quite established data on a cohort basis. That you see this 240 milliliters decline on a cohort basis per year. It doesn't matter if you are mild, moderate, or severe. So it's this, you have this steady decline, and I think that makes us comfortable.
Gotcha. And maybe a final question then on sort of how you're thinking about the positioning of C21 in terms of study design and then also the commercial positioning. We know FibroGen is going for second line, Roche and UTC first line on top of standard of care. What's your thinking for CD21?
Yeah, I think we given this data, we're really surprised with this impressive data, and I think given the clean safety profile, I think it's not. We would like to go potentially first line with this and take it to the FDA, because why carry on the safety issues that the approved drugs carry? I think that's. So we would like hopefully go first line, because it's I think that's the most beneficial for patients and not just be an add-on therapy. So we'd like to position this as first line treatment and not anything else.
Gotcha.
And I think, I mean, we have learned a lot from the current phase 2 study. We really need to bring that into the phase 3 program to make a patient-friendly study in terms of number of visits, to take down the number of visits to the hospitals, because we're still living in a COVID environment. So that is something we learn also. To motivate patients to because there is tough competition out there. So we of course have to design a study, and we're thinking about that, how we are going to balance the study so that patients are motivated to go to our study. And that is, I think, something that we keep in mind. And we are of course looking forward to present more details from our interactions with the FDA down the road.
Great. Hans, we're all out of time. Thank you very much for an interesting presentation and an interesting Q&A.
Pleasure. Thanks so much. Bye-bye.
Welcome back. Our next company is IRLAB, a Swedish biotech focused on the development of new therapies for patients living with neurological diseases. With us, we have here Nicholas Waters, the CEO. Please, Nicholas, the word is yours.
Thank you very much, and nice to be with you again. And thank you once again for this opportunity to present IRLAB. Company focusing on Parkinson's, novel treatments for Parkinson's disease. Just take you through the disclaimer and just a brief overview of the some recent data we presented in the Q1 report, indicating that IRLAB has a very strong cash position, which is a prerequisite in these times, which is also in alignment with our long-term strategy. At present, we are investing in 2 phase 2B programs. So next step for both of those assets will be phase 3. And we have also broadened our pipeline during the past year, and we are now investing in preclinical development of a number of assets. I'll come back to that towards clinical phase 1. As you can see, we have on the left-hand side of the slide, we have thoughtfully built up a cash position over the past 3 years. And since we entered into the Nasdaq Stockholm, we have continuously increased our number of employees from around 12 to 15 to now 30 people. And of course, the cost structure grows with the number of employees, but most of all with the development of the pipeline. We have prepared for this and are now moving forward with our assets in Parkinson's disease. Just a few words on the symptomatology of Parkinson's disease, which is a complex neurological disorder depending on loss of dopamine neurons in the midbrain, which affects basically all aspects of life for patients living with Parkinson's disease. First, typical signs are the bradykinesia, the rigidity and tremor, which are the hallmark symptoms of Parkinson's disease. In that aspect, we are now developing a totally novel strategy for supporting treatment of these symptoms in PD. We are focusing on once daily treatment, which differentiates from today's treatments, which are up to 9 to sometimes even 12 times a day. Patients need to take their levodopa treatments. So we are looking forward to a quite significant shift in the simplicity of treating the early stage symptoms with this strategy. Next symptom complication that comes into the algorithm here is the dyskinesia, the motor fluctuations, and the psychosis symptoms that these patients or individuals experience. There we are developing right now a mesdopetam, which is in phase 2B. We are moving this asset forward together with our partner Ipsen. And this is a strategy to optimally treat dyskinesia, but also psychosis. Later on in the development of the disorder, there is a loss of cognitive function, cognitive impairment, which is usually coupled to dementia, and also apathy, confusion, and depression. Common symptoms in mid to late stage Parkinson's disease. There we have our preclinical assets 942 and 757 addressing these symptomatologies via modern new, or as I say, totally new mechanisms of action. And at the top, one of the biggest problems in Parkinson's disease, which is balance and falls. The loss of balance, which leads to falls, which leads to fall injuries, which is very costly for the patient, but also for society. And here we are developing pirepemat, which is now in phase 2B for this indication. So we are filling up the needs here in PD with a quite comprehensive pipeline. We are using our advanced systems biology strategy, which we have named ISP, the integrative screening process. This is a system where we collect We use phenotypic screening strategies, collect a lot of data, analyze them by means of machine learning, artificial intelligence strategies to define structures and programs. We can also predict usefulness of any new drug. And this highly efficient system, it results in the discovery of first-in-class compounds, which is very important from a competitive standpoint. We also get very strong IPR. We discover molecules which would otherwise go undiscovered by traditional strategies. And strong IPR is once again very important for the competitive edge that we have with our programs. As I said, we can predict which indications are most suited for any new chemical. And we also see, going back in time, around 20 years where we've used ISP, that we see an improvement in the likelihood of phase transitions. And this is one of the biggest issues in drug development today, and it's been discussed a lot in the literature, how to manage the attrition or how to reduce attrition in any pipeline. And with ISP, we discover and develop molecules which have a higher likelihood of surviving phase transitions from preclinical to clinical stages and within the clinical development pipeline from phase 1 to phase 2 and to phase 3. Looking at the numbers we have generated based on our own CDs over the years, we estimate roughly somewhere between 2 to 3 times higher likelihood of reaching phase 3 with an ISP-derived molecule than with the standard industry documented numbers. This, we think, is also adding to the competitive edge that we have. Slide 7 illustrates the pipeline as it looks today. We have broadened it over the past year, as I said, going from the bottom with the P003 program, 757 and 942, preclinical assets now in preclinical development towards IND. And then we have pirepemat for balance and falls and mesdopetam for dyskinesias and psychosis. We have a differentiated strategy, commercial strategy for these assets with showing the partnering with, shown by the partnering with Ipsen last summer for mesdopetam. This was a $363 million deal with double-digit royalties on top, which we are very proud of course. And we have a very, very strong partner in this program, dedicated to developing mesdopetam to the market. We envisage a similar strategy for pirepemat, that is, during phase 2 or after phase 2b data, we expect to join forces with a partner. For the early stage molecules, the preclinical assets, we are discussing the possibility of actually reaching early stage partnering possibilities here. So we have a quite varied strategy towards partnering with our pipeline. Looking at the news flow over the past coming years from each of these assets in the pipeline, and this is just illustrating, slide 8 here, just illustrating the news flow from the pipeline, that is excluding any other major events such as deals or other types of events. So in the mesdopetam program, we expect to see data late this year. We expect to see phase 3 initiation next year and also a broadening of the indication panorama for mesdopetam. The marketing preparations will start also in 24, but that's something that basically starts already when you're working in the preclinical development. And then for pirepemat, we have just recently initiated the phase 2b study. We expect to see data by the end of 24. By the end of 23, we'll see top-line data for the ongoing phase 2b study across Europe. And similarly, we expect for the early stage compounds to reach clinical trials within 23 and 24. So this is a quite rich news flow for a smaller biotech company like ours. A few words on pirepemat, and the unmet needs out there are huge. About 45% of all people with Parkinson's disease, they fall recurrently, up to several times a week. And each fall imposes a risk of severe fractures and hospitalization. Pirepemat is a molecule which we have designed to improve those systems in the brain which govern balance and have the potential to reduce falls. Just looking at the societal costs for these falls and the fall injuries, around $30,000 per patient for a patient ending up in hospital. This is an untapped market. There are no current drugs registered for reducing falls in PD, and it's a huge market. Many patients across the globe. The study is designed as a 3-arm study, 1 placebo arm, 2 different doses, 300 and 600 milligrams. We are looking at a 3-month treatment period after 4 to 6 weeks of screening. That's following the patients to make sure that they fulfill inclusion criteria of at least 2 falls during the past or the 4 weeks prior to randomization. And the primary endpoint in this study is, of course, to evaluate effects on falls frequency. We are proud to have been able to start this study despite the situation over the past year around Europe with the COVID and now also the Ukrainian situation. Looking at the regulatory status here, we have received approvals from all the countries we have applied to start the study in now. France, Germany, Poland, Spain, and Sweden. So this will initially be a European effort. And we will focus now on working with the hospitals and the sites and doctors who will be involved in the study, including the patients, of course. Looking at the preclinical assets, we have 3 or at least 2 programs, and now 2 CDs from the first program, P001. 942 is a molecule specifically designed to improve cognitive function and brain health in neurological disorder. This means that we are looking at potential to broaden our scope a little bit outside of Parkinson's disease as well. But of course, Parkinson is our main target. The problem here with cognitive decline in neurodegenerative disorders is that there is a disruption of frontal cortical neurocircuitries, which we can resurrect with the treatment with 942. And here we see potential for both symptomatic relief, but also for disease modification. The status right now is that we are right now developing the CMC. We are moving towards regulatory studies necessary for filing an IND during next year. With 757, we are moving towards an indication where there are no treatments today. That's apathy. This is a huge problem in neurological disorders. Around 50% of all patients with neurological disorder are at risk of developing apathy. And it's a huge problem for them and for their caregivers. The pathophysiological background or the mechanism underlying this is quite similar to the ones related to cognition, but there is a disruption of frontal subcortical circuitries, and those can be treated or improved based on preclinical data that we have generated, be improved by 757, which increases the neural activity in these regions, specific regions, and in the communication between the regions. Also here we have potential for symptomatic relief and disease modification. Similar to 942, we are now running a CMC development campaign to be able to apply for, and toxin safety studies, of course, to be able to apply for phase 1 during next year. P003, it's the latest addition to our pipeline. We have worked on this for a couple of years. We have now we're looking at a totally new strategy to treat the basic symptoms of Parkinson's disease. And one of the problems with Parkinson's disease treatments available today is that they impose a very strong fluctuation in symptomatology. There are overshoots during certain periods of the day, and there are troughs where there are no effects of the treatments. This, we think, can be resurrected by a long-lasting treatment like the ones we are developing in P003. So we think we have potential to provide an optimal functional efficacy without limitations that you see with current treatments. The mechanism here is similar to what is used today in the gold standard treatment levodopa. That's a full dopamine D1, D2 agonist strategy, but with a much, much improved pharmacokinetic property. We are looking at 2 different strategies. The first generation, we are now in lead optimization. We have previously said that we expect to see a CD from this program this year, which will bring yet another candidate drug into development in our broadening pipeline. And then we have a second generation strategy ongoing on the drawing board right now. A few final words on the collaborative project with Ipsen. That's the mesdopetam program. This is a program which is now fully financed through phase 3 to market globally together with Ipsen. We are looking at improvement of motor function through prevention of reduction of dyskinesias. We have generated data which support the potential prevention here as well, besides the just symptomatic treatment. But there is also a possibility of treating psychosis in Parkinson's disease. There are reasons to believe that this mechanism has antipsychotic properties in Parkinson's disease as well. 30% of all Parkinson's patients have problems with dyskinesia. An additional 30%, 35% have problems with psychosis. And right now, we are running a phase 2b/3 study across Europe and the US. We are recruiting in both these geographical regions. Preparations are ongoing for phase 3 studies, and we are supporting Ipsen's efforts to bring this asset to patients. Besides LIDs and psychosis in Parkinson's disease, there is also an opportunity in tardive dyskinesia, which is actually an even bigger indication globally. It's a side effect of antipsychotics, which can be treated with D3 antagonists, which is exactly the mechanism underlying the efficacy of mesdopetam. So, since we introduced IRLAB in 2017 on NASDAQ, we have now full validation of our strategy towards our vision of being a sustainable deliverer of new treatments for complications in neurological disorders. The science has been published in highly ranked publications over the years. We have had quite good successes with our translation work from preclinical to phase 1 to phase 2 and to phase 2b. The innovation and the novelty is proven by the unique INN names that we have been provided, mesdopetam and pirepemat, indicating they represent totally new classes of CNS disorders, treatments for CNS disorders. And then the commercial value of our work has been validated by the global licensing of mesdopetam to Ipsen last year. So, with those words, I would like to just conclude that we are on a path to deliver strong growth. We have the resources in-house right now. We are building on our competence base, and we will continue to do that during the year. Add commercial competence during the fall. That's our plan. Thank you very much.
Thank you very much, Nicholas, for your nice presentation. I just wanted to remind you if you could turn on your camera.
My camera is on, but I am also showing the... I'll take away the...
Great.
Oh.
Thank you so much.
Can you see me now?
Yes.
Good.
Thank you so much. Now we have time for a few questions. And I will start talking about your interesting approach for drug discovery. It's based in phenotypic screening. And for someone that doesn't understand what phenotypic screening means, could you give a brief explanation on what does this represent and how this compares with traditional screening?
In traditional screening, you start by defining the target you want to address. And then you look for molecules which have properties that can modulate this target. And that depends on a very long period of defining the target you want to address. In our case, with phenotypic screening, which is growing in the industry, is that we allow the living system to inform us about the global effect of any treatment strategy. And the ISP system supports us in discovering drugs which have beneficial effect at the system level, not only at the target level, but also at the system level. There is usually a disconnect between a certain target and a system level effect, and we are capturing the system level effect as a first thing in our discovery. And that means that we don't need to synthesize so many drugs to come to a conclusion of a CD, and we have a very rapid discovery process. It's about half the time in preclinical work compared to a traditional strategy. And that halving of that time is actually very important for the value of the program and the time you have as an exclusive on the market.
That's very, very interesting. And my next question is regarding your newly announced molecule, 757.
Yes.
And you're targeting a space where right now there is no solution. These patients have no solution for apathy. And how come that there is no solution right now? Is it because the pathways that regulate this symptom are too complicated to target?
I don't think so. I mean, if one looks back in history, a number of currently available drugs have been tested with quite bleak results. But using ISP, we have defined and the literature, of course, the scientific literature behind the pathophysiology. We've used ISPs to actually define what the molecule should do. And when we do that, we have the tools in our hands to actually discover molecules which addresses exactly the right mechanisms. So I don't think it's impossible. It's that difficult in a sense. But of course, you need the tools that we have developed to do that.
Yeah, I understand. Thank you. And one last question regarding mesdopetam. As you previously mentioned, together with other key opinion leaders, mesdopetam has the potential to also have an impact, not just on the treatment, but the prevention of PD leads. Could you give us a little bit your view on that?
Absolutely. And the view is based on preclinical data, which we published at Neuroscience a year ago, where we have looked at chronic treatment with mesdopetam in animal models of dyskinesia. And what we can see is that we do see a prevention of the effects of levodopa. Levodopa is what drives the dyskinesia, basically, in patients and in those preclinical models. And it seems that we can actually block that effect over time. So long-term treatment with mesdopetam, hitting the mechanism responsible for generating dyskinesia. If we do that early, we can probably prevent the development of dyskinesia with mesdopetam.
That sounds very, very interesting. Nicolas, thank you very much for being here with us today and for your nice presentation.
Thank you very much for having us, and always a pleasure.
Welcome back. The next company to present is Saniona, and I'm pleased to introduce the CEO of Saniona, Thomas Feldhus. I'll pass the word over to you, Thomas.
Thank you. Good afternoon, and thank you to ABG for inviting me to this conference. And thank you to investors for listening for this presentation. So my name is Thomas Feldhus. I started as CEO on the 2nd of May. I have worked at C-level and board level within the life science industry for more than 20 years. I'm co-founder of Saniona, and I've been CFO for this company from 2012 to 2020. I took the company public in Sweden and listed it on Nasdaq Stockholm main market in 2016. I have significant experience in business development. In Saniona alone, I've made several collaborations, agreements, out-licensing deals, spin-outs, joint ventures, and so forth. I decided to leave Saniona in March 2020 in relation to the implementation of Saniona's go-to-market strategy in the US. I'm now back as CEO to oversee Saniona's refocused strategy on ion channel research and development and to explore partnering opportunities to advance its lead assets. Next slide, please. So Saniona is today listed on Nasdaq Stockholm main market. we are a clinical stage biotech company focused on developing therapeutics for rare diseases. We have made highly valuable preclinical and clinical assets, which provides multiple possible way forwards. We have a unique drug discovery platform, which provide a steady stream of new products into our pipeline. And then we have several clinical assets, including SAN903, which will be ready for phase 1 very soon, and SAN711, where we have completed phase 1 study also very soon. And then Tesomet, which has where we have completed 2 phase 2A studies in 2 rare indications, Prader-Willi syndrome and hypothalamic obesity. The company started phase 2B studies in this winter, but then it has been put on hold. We are currently planning to develop SAN903 internally and to out-license SAN711 and Tesomet. However, we may not end up exactly like this, and this I will come back to. Before that, I think it would be appropriate to take a 2 years back. And next slide, please. So during the last 2 years, Saniona has aimed at to become a commercial stage biotech company in rare diseases based on its lead asset Tesomet. This strategy implied US corporate headquarters and US NASDAQ listing. This spring, it became clear that this strategy has failed. The planned phase 2 studies were put on hold, and the US NASDAQ listing was no longer an option. Market cap has dropped with more than 95% over the last 18 months without any clinical failures. In April, market cap was just about 100 million, which was less than the quarterly burn rate. Everybody could see that this situation was not sustainable. So Jørgen Dreyer, the chairman, reached out to me in April and asked whether I would be interested in stepping in as CEO. I said, yes, I'm a founder, and I know this company. It's a fine company. It has a great pipeline, significant, very interesting platform, and many highly talented people. I'm sure that we can turn this around. So the low market cap now does not reflect the value of these assets. It is dictated by mistrust to the previous management and concerns about financing. Next slide, please. So after the structure in April, restructuring in April, Saniona has again become a Scandinavian company. It is the US operation has been closed, and future annual operating expenses has been reduced by 75%. So 1 month in the old company is 4 months in the new company. We have transformed the company into a more focused and cost-effective organization that leverages our core strengths within drug discovery and development in ion channels and CNS. All the values in the lead assets, including Tesomet, SAN711, and SAN903, have been preserved. We are now focused on establishing partnerships on our lead assets to generate non-diluted funding and become less dependent on the financial markets. We know that model works. Next slide, please. So we know that the market works. We have, until I left in 2020, gained significant experience with this model. We have generated close to 400 million crowns in income through partnerships, and we're still eligible to receive earnouts, milestones, and royalties from several of those agreements. The deal making follows many different paths. Research collaborations with big pharma companies like Janssen, Pfizer, and Boehringer Ingelheim. Development agreements with mid-sized companies such as Medix. Spin-outs where Saniona has capitalized its shareholdings following a trade sale or through the sale of shares held in the company over the stock market. And also then spin-outs where shares has been distributed to the shareholders under Lex Aktier in Sweden. Next slide, please. So I have a strong team behind me. which can and will support me in the operational level and on the BD activities. And it looks like this today. So we have Karen Sania as CSO, Janus Larsen as Head of Clinical Development, and Palle Kristoffersen, who is VP of Research. And then we have Anita Milan, who is supporting me as CFO. She has a lot of experience with financial reportings for listed companies. So Karen, Janus, and Palle, they have deep knowledge of these programs. They work together with big pharmas and biotech companies for more than 20 years. They've been sitting in steering committees. They've been involved in the business development activities in the past. And they have strong communication skills, and they can speak with everybody. So I can take advantage of this. If this is a unique part of this company, the scientists are also working in business development. In addition to this, we have also several consultants, including a medical doctor who will take over as chief medical officer at part-time basis, and then a business development person in the US who have been deep involved in the clinical development and know Prader-Willis and hypothalamic obesity studies. Next slide, please. So we have a fantastic pipeline. On the top, you have tesofensine, which has been partnered with Medix. Medix has conducted a successful phase 3 study for general obesity and filed for market approval in Mexico. The regulatory process has taken longer than expected, partly because of problems with COVID. We understand now that Medix will have a meeting with the regulatory agency in May, and hopefully we can come to a final decision this year. Medix has been granted commercial license in Mexico and Argentina. We were entitled to double-digit royalties on sales, which could provide additional income to the company in the coming years. In case of a positive outcome in Mexico, we would also have the option to roll out the program in the rest of South America and as well as certain Asian countries. Since those countries, we use Mexico as a reference country. Below you have Tesomet, which is developed for the two rare diseases, Prader-Willi syndrome and hypothalamic obesity. Saniona has an open IND and an agreed protocol with the US agency and several other countries to start such a phase 3 study, phase 2B study. The studies have been put on hold due to funding limitation, and we are now positioning for partnering. And there are great interest. Below that, you have Sansamilern. This compound is designed to increase the efficacy of specific neurons in the spinal cord, which controls the pain signal to the brain. So it's a new concept in for pain management. We have strong animal data from animal models in chronic pain, in trigeminal system, and for itching. So it has many potential indications. We expect to report top-line data from phase 1 study over the summer. This study includes also a PET scan study on selected patients where we will be able to see the receptor coverage of the compound and in the spinal cord. And this is important for us because it could guide us for the phase 2 study, because we can see at what dose we will get the coverage which is necessary for the maximum effect of this compound. So there's an important readout coming out of this program this summer, and again, this program is positioned for partnering. And then finally, we have SAN903, which is blocking a calcium-activated potassium channel expressed in many types of immune cells and fibroblasts. The product is designed to prevent proliferation and expression of cytokines by activated immune cell and fibroblasts. This means that it will prevent inflammation and fibrosis, scarring, which is involved in many chronic diseases. We have strong data in animal models of rare diseases such as idiopathic pulmonary fibrosis, as well as more common diseases such as IBD. We are currently planning to keep this product internally, and we will hopefully be able to start a phase 1 study at the year end. Next slide, please. So in addition to this, we have a drug discovery platform for development of new therapeutics which modulates ion channels. Ion channels have been validated for treatment of various diseases, including diseases of the CNS. This important class of targets represent a huge potential for developing of new therapies, since only 20% of the known ion channels has been utilized for development of new drugs. We worked in this field for more than 20 years. We generated a large library of ion channel compounds. We have a huge number of data which has been integrated into our proprietary database, which can be utilized through the use of artificial intelligence for further research programs. This platform may also be used for partnering, such as we have demonstrated with research collaboration with Pfizer, Janssen, and more recently, Boehringer Ingelheim. Next slide, please. So this slide illustrates the ion channel drug discovery engine. CNS drugs are discovered through an iterative process. It requires a lot of disciplines, such as chemical design, precision biology, in vivo stability, target engagement, translation, and so in vivo pharmacology. We have all those disciplines in-house and are only outsourcing wet chemistry to a long-term partner in India. The data from each compound are then collected, put into the database, then we compare to what we have predicted, the physical, chemical, and biological properties, and this enables us to refine the model and then design new compounds, and then finally get to the final drug. Thank you. Next slide, please. So this platform has delivered several clinical assets, including SAN711 and SAN903. But we have further progress just below the waterline of this iceberg. We have informed the market that we will select a new candidate from preclinical and clinical development very soon, which is illustrated with SAN-XXX in this slide. We have also SAN-YYY, which is in lead optimization, and thereby we may be able to leave another clinical candidate within the next 12 to 18 months. These two programs are positioned towards different types of epilepsy. They modulate two different ion channels, which have generated significant interest in the pharmaceutical world. We believe that we have two very attractive and coming programs with here. In addition to this, we have two partner research programs, one with Boehringer Ingelheim for schizophrenia, which also have moved nicely over the last 12 months, and then a joint venture, Cephagenix, in migraine, which again has moved over the last 12 months. These programs are funded by partners and are actually covering 1/3 of our research cost. So there's a lot of active programs, as you can see, and we are able to develop a very large group of compounds internally, which can be explored internally for development or in partnerships. This means also, as you see, that we are able to expand our internal clinical pipeline fast if we are to out-license Tesomet and SAN711 over the next 12 months. Next slide, please. So going back to my initial slide, we have a significant number of assets, and there are multiple possible paths forward. We have an ion channel drug discovery engine, which we expect to deliver a steady stream of new clinical candidates. We tend to develop SAN903 internally and plan to initiate phase 1 clinical studies by the year end. The candidate has many potential indications, including rare diseases such as IPF. And then we are exploring out-licensing opportunities for SAN711 and Tesomet. It may be that we will not out-license we will out-license SAN903 instead of one of these two compounds. If we were to retain SAN711, then I think we would conduct a basket study comprising a relatively small group of patients in different indications, such as trigeminal neuralgia and migraine, neuropathic pain indications, and itching. And the aim here is then to explore the potential in each of those indications for further clinical development. It may also end up that we keep Tesomet. And in that case, I think that we would start with hypothalamic obesity. it is a much easier patient population to work with than Prader-Willi syndrome. We have very strong data in this indication. Recruitment will be faster. The investigator and the key opinion leader, Ulla Feldt Rasmussen, is very supportive and enthusiastic about this compound for this indication. And then many of the patients in the proof of concept study wanted to continue treatment following the completion of the study over 12 months. So this is a very strong sign. And I believe that risk is much lower here. The market is also there. It's basically the same size as Prader-Willi syndrome. So in case of funding constraints, I would prioritize hypothalamic obesity and some of our earlier compounds first. So summing up a little with the coming milestones. Next slide, please. We expect top-line data from our phase 1 study for SAN11 within a few months. We will complete the preclinical development for SAN903 and make it ready for phase 1 clinical studies also here over the summer. Then we hope to come to a final conclusion on Medic's phase 3 program and a market authorization, and then potential for new royalties. We expect to present a new clinical candidate from our ion channel candidate. And finally, we are aiming at entering into a collaboration agreement with at least one of our lead assets within the next 6 to 9 months. Thank you for your attention.
Great, thank you very much for that presentation, Thomas. A few questions then. First one, perhaps on Tesamet and what a potential acquirer would need to do to sort of reinitiate that clinical program. How easy is it to sort of restart that? What is it that is sort of still in place, and what needs to be rebuilt, I guess, to reinitiate those programs?
Okay. As I mean, I think that it's a start over, to be frank. So the clinical you could say the sites were initiated, but recruitment was not really started. Mm-hmm. And therefore there was no patient put on hold. But of course, MedPace, which was the CRO, they have the whole setup. They have contacted and set agreement up with 40 centers for each indication or more. Mm-hmm. And that could be done faster. Mm-hmm. So they can step in the shoes of ours and take it over from there if this is what they want to do. Mm-hmm. Mm-hmm. And there are also potential for finding an amendment, actually doing the study faster. The previous man didn't speak about that because there was not an agreement about that with FDA. Mm-hmm. But that could potentially also be happened. So there's the protocols are written. There's also even amendment to it. So it enabling these phase 2 studies to be turned into a pivotal study, you could say, and thereby spare a lot of years in clinical development. Yeah. Okay. So a lot of work is done. There's an orphan drug indication for both, and there's an open IND. Yeah.
Alrighty. I guess a question on investors' minds will be, given the strained financial situation, how sort of pressured you are to find a potential acquirer of Tesamet or indeed SAN711, and having to sort of agree a quite significant discount. How should one think about that?
So I need to be careful here because we are just about to publish a financial report in a week or so. So we had in cash 356 million crowns in cash at the end of the year. Mm-hmm. And that's quite a lot of money, I would say. And in the current setup, our burn rate will be, on the base case, 70 million crowns. So that could be to several years. But the burn rate was quite high in the old setup. I think in Q4 we had a burn rate of about 130 million crowns. And Q1 is probably going to be in the same level because, they were in clinical trial, they were full set. The real turn change happened at the end of March and actually in the end of April. So there's 4 months burn rate at this level. And then there is cost for shutting down the study. and there will also be severance costs for the existing people who was laid off. And so there is a lot of money coming out of the company in these two quarters here. And the full picture, I don't have it yet, and I will not really comment on it. But still, there's a lot of money, and I do believe that there should be at least a year of cash, and then also longer. But we have also a debt facility with Formula, which has to be paid at the summer 2023. Right? So yeah. So I think that given the current financial market and the company's burn rate and the company's market cap, we will do a lot for making a collaboration with one of these lead assets within the next 12 months, both in order to advance the asset, but also to get some cash in to strengthen the balance sheet.
Alrighty. And perhaps one final question then also, I guess one that will be on people's minds. You're obviously seemingly quite upbeat or confident in being able to find a partner for one or more of these assets. But in a scenario for whatever reason, you're not able to strike a deal for either Testmet or SAN 711, and therefore unable to raise that non-dilutive financing. What is the sort of backup plan, or how does it look in that scenario? Or
then in that case, we will go to the market. I think we have cut it down to what I think is a very lean organization, and we think that the real value is actually our ability to make new drugs. We could make collaborations on the early asset, also on our research platform, and thereby actually going back to a cash neutral situation. The first 2 years of this company's life, we were profitable. So we actually made money by making research collaboration prior to getting listed in Stockholm. So we have been there before, but I do think that there is a significant interest in these assets. I've spent 1/3 of my time in business development meetings since I started on May 2nd. A lot of companies have reached out to us following the announcement in the end of April, and asked to our clinical programs because they may fit into their pipeline. So if you have a cash-rich rare disease company in US with a product on the market, this may fit very well into their pipeline. What's going to be next? So the same. Yeah.
I might wager a last question. I don't know how able you are to answer it, but in these BD discussions that you're having, are you able to say whether any term sheets?
No. I can't do that. As soon as we,
sure.
So the early discussion you can talk about, and there's been a lot. As soon as you go to term sheet, then everything shuts down, of course, because then I would say the same thing. There's a lot of interest, so to say, because this is critical information.
Sure,
yeah.
Fair enough. Thomas, we're out of time. Thank you very much for an interesting presentation.
Okay, thank you. Okay, bye-bye.
Welcome back. The next company to present is Oncopeptides, and I am pleased to welcome the CEO of Oncopeptides, Jakob Lindberg. I'll hand over the word to you, Jakob.
Thank you. Thank you for being here, for allowing me to present today. I'm going to present Oncopeptides, and as you know, we have had a very turbulent year. Happy for Q&A afterwards as well, of course, but first just go through the basics over roughly 20 minutes. The obligatory disclaimer regarding forward-looking statements, etc., and this will also be available on our website afterwards to read for those that are in particular interest of this. Just in brief, we are a life science company in the biotechnology sector. We were founded in 2000, so we're a bit more than 20 years old, and we are listed at the main market here, Nasdaq Stockholm. And of course, we have extensive collaborations with a lot of leading research institutes. What we're having is that we developed a discovery engine for targeted therapies for hematologic malignancies, that is cancers for the blood, in the blood. And our main platform is the peptide drug conjugate platform, which actually uses the power of existing cytotoxic agents, but adding a delivery mechanism to it, which is basically an enzymatic motif. We have several of these in pipeline, but of course our lead asset is a molecule called melflufen. And melflufen is currently approved in the US under the name Pepaxto, but we are not marketing it after we got, as everyone knows, a safety warning letter from the FDA last summer, and we have been working diligently since then to try to resolve that. We have a European application process that is on track for a so-called CHMP opinion in Q2 of this year, and assuming that we get approved in Europe, Germany would be the first market to launch later this year. Highlights for this year. First of all, we have an ongoing review process with the European Medicinal Agency, and it is proceeding as expected. And this actually includes both the Horizon and the Ocean data sets, and this is a critical point, and I'll come to that. We have, of course, through this period also, despite everything that has happened with melflufen, continued our focus on research and development, and primarily on the preclinical side. And that includes both the next generation of peptide drug conjugates, but also we have a very advanced stage, actually, NK cell engager platform that is peptide-based with very good preclinical data. We have not talked about this so far, and the reason is that we thought that it would be distracting from the melflufen process, but this is something that we hope post-CHMP opinion to give much more flavor around, because this is, as people might know, a very hot field. Last year, we voluntarily withdrew from the US market in October, but actually we rescinded that voluntary withdrawal on January 21st, or we reported it on January 21st. So right now we are still approved in the US and in dialogue with the FDA regarding our approved product, but we are not marketing it. We published the data from our phase 3 Ocean study in Lancet Hematology in January, and of course this complete data set have been shared with regulatory authorities. As to the regulatory update, I already gave the main highlights. The current process with EMA is proceeding as expected. Last week we had a scientific advisory group meeting, that is called a SAG, with EMA. That was on May 11th. And we are continuing the application process and expect still, according to plan, a CHMP opinion now in the second quarter. The EMA application process was originally only based on the Horizon data set, but now also includes Ocean as a confirmatory study. And I think this is very important because that opens up the possibility for full approval instead of just conditional approval from the beginning, since the confirmatory data is part of the application. Please note the indication is still based on the Horizon data set, that is in triple-class refractory patients. We published the phase 3 data, and as you know, this study and its result has caused a lot of discussion. It was a study that met its primary endpoint of progression-free survival, but with a very heterogeneous overall survival result that that is basically the topic of discussion also with regulators, how to interpret this overall survival result. The article was well received, and as you can see, the conclusions from the scientific authors is that these results also suggest that treatment with melflufen should be carefully tailored on the basis of a patient's previous medical history. And this is a direct link to the heterogeneity in the overall survival result of this study. And we'll come to that within short. The main challenge in interpreting this study is kind of summarized in this graph, and this is a complex graph. As you can see, it contains a red line with a solid line with dotted red lines around it, and a solid blue line with dotted blue lines around it. The dotted lines depict the 95% confidence intervals. The red line is pomalidomide, the blue line melflufen from the OCEAN trial. Further down, you see the patient age in the study, and then you see the hazard of death within each treatment arm. So you cannot make comparisons between the arms here. And as you can see, in the melflufen arm, the hazard of death is rather similar across the age spectrum. Taking the 95% confidence interval into account, it's a straight line. While in the pomalidomide arm, you see very large differences, and they are significant in the risk of death for various patient groups depending on the patient's underlying age. This is at the heart of how to interpret the OCEAN trial. For anyone reading the pomalidomide label, if you go in Europe or in the US, there is no mentioning that the treatment effect of pomalidomide would differ by age at all. So a core question is, of course, is this result representative of the pomalidomide treatment effect, or is this an outlier? In the study, and you can see here the survival data across the pre-specified age groups, and you can see that in the table. You see that melflufen was 70% worse in terms of survival than pomalidomide in patients less than 65, but more than twice as good in patients from 75 and above in terms of survival. And this is very dramatic in terms of differences, and it's statistically significant in both ends. This is obviously at the heart of every discussion with the regulator. And the question is if this is a result that exhibits what is called true heterogeneity, or whether this is a random find. If this is true, this has a lot of implications for how to interpret the results. If it's not true, then this needs to be established in future potential clinical trials. Our argument is obviously that this is a matter of true heterogeneity, and one of the primary drivers behind this is actually pomalidomide, and this can be seen in multiple studies for pomalidomide as well as other immunomodulatory drugs. As soon as we can, we will come back with regulatory updates regarding this, obviously. As we have stated, in Europe, we will have more updates towards the end of Q2. And whenever we can, we would like also to share update regarding the FDA dialogue. More than 70 patients are included in the early access program in Europe. We are preparing market access in Germany, assuming a positive CHMP opinion. And if we are approved, we will be ready for commercial launch in Europe before the end of the third quarter. It's important, and we will come back to this, given our position after what happened last year, we will definitely need commercial partners to fully commercialize this product, both in Europe as well as in the US. I see it right now as highly unlikely that we would build the correct infrastructure to do this completely on our own. Actually, I rule that out, as a matter of fact. As stated previously, we rescinded our voluntary withdrawal of Pepaxto in the US based on further review and analysis of the heterogeneous overall survival data from the OCEAN study and other relevant trials. We are not marketing Pepaxto in the US at this time, and we have a dialogue to try to reach a mutual understanding and interpretation of this data set. Right now, I don't want to speculate on the timetable and the outcome. We are, however, committed to provide US patients continued access, and this is done through what we call the expanded access investigational drug application process. And this also required the FDA to actually, on a single patient basis, approve a patient for treatment with melflufen. I mentioned preclinical development, and I will just briefly talk about this. We will come back to this point. We have several other peptide drug conjugates in development, and the most advanced is one called OPDC3. But more importantly, we have an NK cell engager platform that we call SPIKE, the SPIKE platform, where the preclinical data is tremendously strong, actually, and we want to come back to this. And this is actually a fairly complex peptide molecule that is trivalent. It binds 3 modalities, 3 binders between the NK cell and the target cell. And we will come back to this, but we have, for anyone following that field, there are recent deals that are very large, and we are at a similar stage of development as the companies that have made those deals. With that, I open the floor for questions and answers.
Great, thank you very much, Jakob, for that presentation. To some questions and answers then. Obviously, there's a particular topic that is, of course, of interest to most people, and I'm cognizant of your limitations in terms of what you can and can't say. But I'll ask away. So the SAG meeting was held last week, I believe we said, and Oncopeptides was on the CHOMP agenda for the May meeting.
Correct.
And perhaps just to begin by clarifying, what was the purpose of the agenda item for Oncopeptides on this CHOMP meeting? Was that to discuss the outcome of the SAG meeting, or was it to decide on regulatory recommendation?
So basically a scientific advisory group meeting in the EMA system. They opine on specific scientific topics, so not regulatory topics. So EMA and the CHMP group asks certain scientific questions to the SAG. The SAG answers them and comes back with specific answers, and they need to be very specific in how which of course boils down to also how well are the questions put, right? But to be honest, very good questions. So then the answers in written format goes back to the CHMP to say, okay, in light of those questions, these are the regulatory actions we take based on those answers. And so they need to translate a scientific conclusion to regulatory conclusion. And that is then what the CHMP is doing. And that was exactly what they did at this meeting. So they asked questions, SAG came with answers, then that is processed by the CHMP and results in, so this is our regulatory position.
Right.
Yeah.
Okay. And at this CHOMP meeting that's happening, I guess it's finishing today, was that regulatory decision on the agenda? Was that what they were deciding on now, or is that for the June CHOMP meeting, or do we not know?
So what we got now was then based on the input from the SAG, then this is our position, and then it goes back to us and say, are you okay with this? And normally what happens, right? And I know there was a lot of discussion regarding the wording regarding oral explanation in the current agenda. The reason is, based on the SAG output, they will say, okay, so this is our position. Then that goes to us. If we are of a different opinion than the CHMP, then we will ask for an oral explanation. But if we are in agreement, there won't be an oral explanation.
Right.
So, simplistically.
Yeah.
And so that is why. So there needs to be possibility for an oral explanation. But we'll see. Right now, we're just looking. Obviously, this will occur then between now and June 8th to see if these last topics, so to say, if we're in agreement or not.
Okay, alrighty. And that was going to be my next question. I guess the timeline that we're operating under now. So the CHOMP decision is referred back to you, and you, I guess, review it and have an opinion on it, and that happens up until June 8th, were you saying, or was that? So
the maximum timeline is to June 8th, and then either we have reached a consensus, and then there's no oral, or we haven't, and then there will be an oral explanation.
Okay.
Which is kind of up to us how much we want to fight the conclusion, right? And otherwise, then the output of that, with or without an oral, then goes into the CHMP meeting that is held in June, with then a decision on June 23rd.
Okay, alrighty. Okay, no, that makes sense then. And I guess in parallel to this, there's the FDA process, which is less well defined in terms of what the processes are and the timelines. I don't know if there's anything much you can say. I guess you explicitly said that you weren't going to comment on the timetable. So, but is it reasonable to say at least that it is likely to whatever that process is to come after the EMA processes?
Honestly, I don't know. What I do know is that there is a lot of communication between EMA and FDA regarding this. And then to what extent EMA has a set timetable. Whether the FDA wants to sort of be aligned with that timetable or if they run their own process here, I don't know. But I would guess and assume, but please note, that's speculation, that they would try to align roughly with the FDA with the EMA timetable in the end, which doesn't mean on the day, right? But not to be 6 months later, I think. But that's my guess. Sure. We don't know. We don't know.
Okay. And then I guess a question on the other pressing issue of the financial state of the company, specifically on the EIB loan facility. We know that's being renegotiated, the terms of it. And I guess firstly, if there's anything new you can say about what those new renegotiated terms are, and in particular, will that line of credit be dependent on regulatory approval, either in the EU, I guess, primarily?
So first of all, the renegotiation is simply that the old agreement was negotiated in light of the US approval. And so now, basically, how we already approved in the US, but obviously it's an interest of EIB here that we resolve the FDA issue or not, right? So then we need to define a milestone around that rather than the approval, etc. So it boils down to a lot of fine prints. The second is that in a structure like this, you always have a debt component and a warrant component. And given our share price, the warrant component needs to change, obviously, because warrants have a different value now than they had before. All the milestones will be connected to Pepaxto, or Pepaxti, as it will be called in Europe, approval and milestones. Yeah.
Okay, alrighty.
And the first one is, of course, European approval.
Sure.
Yeah.
Okay, well, that makes sense. And I don't know if you're able to answer, but of that 40 million, the loan facility, how much roughly would be sort of tied to or made available immediately on EU approval if that were to happen? Is it a small proportion of it or a reasonable chunk of it, I guess?
A reasonable chunk.
Okay.
What we're talking about, please note, since there's no ink on the paper, but it's roughly a third.
Okay, already. Okay. Okay, and I guess a next question on the, again, maybe tricky to answer, but going back to the FDA process, I guess this is back in 2021 when that announcement came that you were rescinding your approval. There was this mention of the need for a new dose finding study. If we imagine a scenario wherein Pepaxto is approved in the EU and there's a favorable resolution in the US, how should one think about what would need to happen for Pepaxto to be in a sort of a commercially ready state in the US?
So this is a very good and technical question. The first thing is that, of course, the need for a new dose-finding study was based on the assumption that there was a safety signal in the OCEAN trial. So let's assume that the conclusion is that there is no safety finding general. And please note, the safety warning was on the population level. There was no discrimination, like all patients are at risk with melflufen treatment. Let's assume that the conclusion is not that is not true. then the question is, why would you do a new dose-finding study based on that, right? So there are a lot of moving parts in this dialogue, and I think I cannot answer today if EMA now comes to a different conclusion. What conclusion, how will that affect the FDA? And based on that conclusion, what is then required according to them? And there are a lot of unknowns there. But I mean, you should read the conclusions about dose finding was very much in light of a preliminary conclusion that there was a general safety find in the study. So if that is not true, then the question is, and I don't have the answer to it.
Okay. My recollection of it was that it was tied to the number of dose adjustments and dose reductions, but perhaps that is still seen in the light of...
Absolutely, in light of that.
Yeah,
yes.
Okay, alrighty.
Because the fundamental aspect of, and I think this is sort of was a general conclusion, there was no toxicological safety findings in the melflufen arm. And then the conclusion was, since we say that there is a safety warning, it must depend on something. So then the dose must be too high. So it was like the other category, right? But that is still, it still follows from the preliminary conclusion that there was a general safety find with the drug. Yeah.
Okay. Alrighty. And you mentioned in your presentation here the core question, I guess, for regulators and the EMA now is whether this heterogeneity as it relates to the POM efficacy, whether what we're seeing in the OCEAN study is representative, whether there is in fact true heterogeneity, or if it's a random finding. I guess from a regulator's point of view, how do they go about making that determination? What do they look at?
All other trials with pomalidomide and IMiDs. And when we referred to that, we had looked at other data in January when we rescinded our voluntary withdrawal. It was because we had done a meticulous effort to collect all studies that isolate the IMiD treatment effect. And I can actually show are my slides still visible on screen?
Yeah, I believe so, yes. Yeah.
So when you look at, there are not many trials that actually isolate the IMiD treatment effect. And on this screen, you can see that there were the original 2 lenalidomide trials, there were a bunch of academic trials with thalidomide, and then over a decade, nothing. And then suddenly trials come in. And you also have at least 8 clinical trials outside of myeloma, and then some supporting data from other trials. We collected all of this and went through it. And our conclusion was that this is consistent. It's not a random find, it's absolutely consistent. Now, it's very important to note that this is our conclusion. And we have been a little bit careful to talk about this because this is, it's problematic, right? Because this is not what is written in the labels of these drugs. We'll see if the regulators agree or not. But for me, scientifically, for me, this is crystal clear. There's a significant survival benefit difference based on primarily patient age with these drugs, and you don't see it on progression-free survival. But you see it on overall survival, which is ultimate. Progression-free survival should act as a surrogate for overall survival, and in this case, it doesn't, which is a huge regulatory headache in itself.
Yeah. Alrighty, no, that's interesting, and I mean, we've done a similar exercise, less meticulous than yours, I'm sure, and I guess I've reached a similar conclusion on whether the OCEAN study was a spurious finding as relates to POM and that effect, or if it isn't. And I guess a question then that follows is, whether the assessment that you make will be the one that the regulators make, and a question then is there any precedent to sort of point to? Has this happened in the past, or anything similar that we can kind of look at?
I mean, I don't have a good precedent. And just so that the listeners understand, the fundamental problem is that this puts into question the information value of the ITT result. As you know, our overall survival ITT hazard ratio was 1.1. And, the 1.1 is supposed to be sort of an average that is representative of the whole. So you have a sort of a distribution around that. But if the ITT result is more akin to let's say you go, I pulled this example earlier today. Let's say you go to a metabolic clinic, and at this clinic you have 40 severely anorectic patients and 30 morbidly obese patients, and you say the average weight is 72 kilos. What information value does the number 72 give you? Nothing. It's actually even misleading because, of course, you have one group that has a very low average weight and one group that has way too much. The average doesn't tell you this. So it actually obfuscates the truth. And if you have true heterogeneity, it puts into question what information does 1.1 give you? And obviously then you need to look at the subgroups to actually establish where you have benefit and where you don't. And that is very much our position, and then we'll see what the regulators ultimately say here. Scientifically, I think this is fairly clear, to be honest.
Okay, Jakob, we're out of time, I'm afraid, but thank you very much for an interesting presentation and Q&A.
Thank you very much. Thank you.
Hi again. Our next company is PCI Biotech, a pharmaceutical company focused on the development of of therapeutic solutions for improving treatment of cancer. With us, we have here today Ronny Skuggedal, the CFO. Ronny, the floor is yours.
Thank you for the introduction and welcome to PCI Biotech's company presentation here at ABG's Life Science Summit. My name is Ronny Skuggedal and I'm the CFO, and I take the opportunity to say thank you to the hosts for the opportunity for this company presentation. So next slide, please. So please pay attention to our important notice and disclaimer. We can go to the next slide. So PCI Biotech in brief. PCI Biotech is a listed and innovation-driven company. We focus on oncology. Our vision is to develop and commercialize novel therapeutic solutions to address unmet medical needs. Our technology, PCI, photochemical internalization, is a technological platform that enables drugs to reach intracellular targets. We use this platform technology for 2 different programs. We call them FimaVac and FimaNAc. FimaVac is our lead program, and here we apply a unique mode of action to enhance the essential cytotoxic effect of therapeutic cancer vaccines, with the aim to turn immune cold tumors hot. Here we are in the planning phase of a phase 2 study. We plan to initiate it next year and results anticipated 2024. We also have a preclinical program called FimaNAc, and here we utilize the platform technology for intracellular delivery of nucleic acids, such as mRNAs. And here by that, we address one of the major bottlenecks facing this emerging and promising field. And here we target on applications suited to the specific strengths of the PCI technology, and we have a collaborative approach with several collaborations ongoing. Next slide, please. So some words about our platform technology. PCI consists of 2 elements. One photosensitive molecule named fimaporfin and light. Fimaporfin is designed to attach to the inside of endosomal and lysosomal membranes. When we apply light on the cell surface, fimaporfin takes up the energy from the light, and this triggers destabilization of the endosomal and lysosomal membranes, and this generates endosomal release, intracellular endosomal release. And this again enhances the access of therapeutic molecules to their intracellular targets. And this we apply in FimaVac for enhancing the essential cytotoxic effect of therapeutic cancer vaccines, and for FimaNAc to overcome the challenges of endosomal escape for a nucleic acid delivery. Next slide, please. So our pipeline, FimaVac, lead asset, therapeutic cancer vaccine entering phase 2, FimaNAc, preclinical delivery of nucleic acids. Next slide, please. So our lead program, where we enhance cellular immune responses, important for therapeutic effect of cancer vaccines. Here we have compelling preclinical results. These results are translated into man via a phase 1 study in healthy subjects. And here we demonstrated safety and encouraging immune responses with peptide vaccine antigens. Next slide, please. So our strategy before we take this into phase 2 is that how can we in best way demonstrate the platform's potential? And checkpoint inhibitors are dominating the immunotherapy market, but with limited response. And this is an attractive opportunity space to leverage the FimaVac platform. Because we want to sensitize non-responding tumors to checkpoint inhibitors and thereby improve their efficacy. And we seek an entry indication with a high perceived likelihood of response, where there is a high unmet medical need and on commercial opportunity. We seek an effective trial design to maximize the possibility to show anti-tumor activity and turn tumors sensitive to checkpoint inhibitors. And by this phase 2 study, we want to establish the fimaVacc treatment concept and have an asset ready for partnering. And by that, expand the clinical established fimaVacc platform into follow-on indications. Next slide, please. So why is fimaVacc excellently suited for this purpose? It's because cytotoxic T cells are the preferred immune cells for targeting cancer, and this requires MHC class 1 antigen presentation to be induced. A major hurdle in cancer vaccination programs have been to achieve this kind of MHC class 1 antigen presentation. And this is exactly what we can do with fimaVacc. We overcome this challenge by enhancing MHC class 1 antigen presentation. This activates and induces infiltration of cytotoxic T cells, resulting in tumor cell killing. So we co-inject fimaporfin, our photosensitive compound, with vaccine antigens. We illuminate the injection site. We get intracellular delivery of the vaccine antigens. This generates a rerouting and enhancement of MHC class 1 presentation of the vaccine antigens. And this again leads to activation and infiltration of cytotoxic T cells. So that's why the technology is excellently suited for the purpose of turning cold tumors into hot. Next slide, please. So with all that in the planning phase, we are now ready to approach phase 2. And the approach here is, as I said, use fimaVacc to increase cytotoxic T cells and their infiltration into the tumor. We combine it with immunomodulation, and this is expected to convert cold tumors to hot and thereby sensitize tumors to checkpoint inhibitors. The entry indication we have pinned down to is recurrent and metastatic head and neck squamous cell carcinoma, progressing on checkpoint inhibitors as first-line treatment. Around 70% of these patients progress within 6 months, and cold tumor contribute to this resistance. So here is a clear unmet medical need. So the study design here is optimized to explore efficacy of PCI vaccination combined with immunomodulation to sensitize tumors to checkpoint inhibitors. That's because we want to turn non-responders into responders. And by that, we are able to design a proof of concept study with a feasible budget and timeline. Next slide, please. So some more details about the study design. The main eligibility criteria: recurrent or metastatic head and neck, progression on pembro or nivolumab within 6 months, and HPV negative patients. We foresee up to 20 patients where we combine our peptide antigen vaccine with an adjuvant, a checkpoint inhibitor, and chemo. The main endpoints are 6 months progression-free survival, objective response rate, safety, and immune response in tissue and blood. And this is an open treatment study, so we will review accumulating data along the way. We aim for a European-focused study with around 10 clinical sites. Core clinical investigators and external experts are engaged with the program, including Professor Kevin Harrington from the Institute of Cancer Research in the UK and Professor Ezra Cohen at the University of California in the US. Next slide, please. So the timelines here, preparations now in 22, study conduct 23, and results 24. We will have accumulating data along the way, and we aim for an asset ready for partnering along the way. Open treatment study enables news flow as results accumulate. And importantly, we have a strong clinical core group of QS to support the study preparations and the performance of the study. Next slide, please. So here for this phase 2 study, we have a focused opportunity positioned to drive value. We have a solid foundation. We have solid preclinical data, including anti-tumor responses, and we have solid understanding of the mode of action. Preclinical data strongly enhances T-cell immune responses, and these data are translated into man in a phase 1 study in healthy subjects, where we successfully demonstrated immune responses in healthy subjects. And the treatment was safe. The scientific rationale is solid as well. The mechanism of action with rerouting and enhancement of MHC class 1 presentation of the vaccine antigens are expected to induce T cells necessary for checkpoint inhibitors to work. We aim to turn cancers sensitive to checkpoint inhibitors and then turn them from cold to hot tumors. We have a broad IP portfolio. The entry indication we have chosen, recurrent or metastatic head and neck patients, have a high likelihood of response to study treatment. There is a high unmet medical need, and there is an ample opportunity for value growth. And we have in-house expertise in clinical development, and our internal expertise is supported by core clinical experts, and our concept for the study and the clinical study design is endorsed by international and renowned KOLs. Next slide, please. That was our clinical program. Now let's speak a little bit about our preclinical program, where we use the intracellular delivery technology to deliver nucleic acids. Nucleic acids has a main challenge, and that's that they are not able to freely pass the cell membrane. They need to get into the cytosol to work, and this is where PCI is very well suited for this opportunity, because PCI can effectively deliver nucleic acids intracellularly with targeted and triggered delivery. And within the FIMAVAC program here, we use this for intratumoral delivery of nucleic acids. And here we have strong evidence from preclinical studies of enhancement of mRNA transfection without off-target delivery. These results compare beneficial to LNPs, both with regard to transfection and off-target effects. And we have seen systemic effects upon treating one tumor. In FIMANAC program, we use the intracellular delivery technology to deliver mRNA. And here we are targeting dermatology, an application which is very well suited to the specific strengths of PCI. It's easy to illuminate, and we aim to develop a topical application and a custom light device. Next slide, please. So here are some examples of our preclinical data from delivery of nucleic acids. On the left-hand side here, you see intratumoral delivery with excellent results with our technology in animal experiments. And on the right-hand side for fimaNAc, delivery of mRNA to skin via intradermal injection. Next slide, please. So this preclinical program has several research collaborations established, and these offer valuable scientific know-how for PCI Biotech. We generate encouraging results, and this is an opportunity for intellectual property establishment for PCI Biotech in different fields. And we are, of course, pursuing new and value-adding collaborative opportunities. Next slide, please. So to summarize here, we have a broad, innovative platform, triggered endosomal release, endosomal escape, can be used in many different ways. We focus on fimaVac and fimaNAc for our programs, and this generates a wide pipeline opportunity for the company, where we have fimaVac as our lead asset in the clinic. We have compelling data from the preclinic with fimaVac and from the first phase 1 study in healthy subjects. We have a clear clinical development strategy, which is endorsed by KOLs in the EU and the US. This is a focused opportunity with efficient clinical translation and an ample opportunity for value growth. We have strong leadership, experienced team in drug discovery, development and commercialization, and supported by these clinical experts. So the clinical development plan for our lead asset, fimaVac, study preparations now in 22, conduct 23, results 24. Next slide. And by that, I conclude my presentation.
Thank you very much, Ronnie, for such a nice presentation. And now we have time for some Q&A. And I will start asking you about your phase 2 proof of concept trial that you will run in head and neck cancer patients. Could you comment on the patient population that you have chosen? Because as you just mentioned, you have chosen HPV negative patients. And why did you? What's the rationale behind choosing such population? Is it more like a commercial reason, or more like something related with the biological process behind this type of cancer?
I think the right answer to that is a combination of these two. We aimed for an entry indication where we see high likelihood of or perceived high likelihood of response to our treatment. And we also looked at how can we design a very efficient, cost-efficient, and time-wise well-designed study. So there are a lot of elements into the equation when we chose this. And more details about the scientific rationale, I'm just a CFO, so I'm not going into those discussions.
I understand. And my second question would be related to the CPI that you're planning to use in such phase. Do you have any strategy behind this? How are you planning to acquire the CPI? Is it going to be through a partnership, or you're planning to get it on your own?
We are working on both paths for this, of course. And as you know, it's a tough fight to get these ICIs on board, but we are working on it. But since it's a small study, relatively small study, up to 20 patients, we don't consider this to be a showstopper for the study.
Great, I understand. Thank you. And another question, and this one is regarding your fimaNAc platform. Right now, I guess that the big accomplishment that you have to show is that you can deliver nucleic acids to the right cells in the right disease. Could you let us know, in the hypothetical case that you managed to prove the whole preclinical phase, how are you planning to move strategically to the clinical phase within this platform?
And here we are 100% dependent on partners. We don't have nucleic acids in-house. We just have the delivery technology. But we seek partnerships, and we strongly believe in the technology. And intracellular delivery is still a major hurdle for this class of drugs. So we continuously work on this project for generating partnering interest.
Great. And I guess that you have been in contact with key opinion leaders and clinicians. Could you elaborate a little bit on the vision that they have around your both platforms, both fimaVacc and fimaNAc?
I can comment on fimaVacc at least, that the clinical expert group we have established here for this study in head and neck cancer patients. This clinical expert group, they are very eager and supportive and very engaged in the program, and they have been to great help for the company in the planning phase. And we think it's a flying start to have these key opinion leaders on board and already the clinical expert group established, meaning that we have around 5 sites already on board. And that gives us a flying start in the kickoff process for the study.
Yeah, sounds very interesting. And one last question before finishing. Regarding your cash position, and where are you right now on this, and what are the key activities that will drive your costs before starting the phase 2 proof of concept trial?
Yeah, the key costs for in the planning phase is, of course, the CMC part of all supply of study treatment material is the key for us now. And regarding cash position, we currently have financial run rate towards the end of next year. But this phase 2 study is not financed as it is today. So we are doing all the preparations and exploring financial opportunities to finance the study.
Runi, thank you very much for your nice presentation and for being here today with us.
Thank you for the opportunity.
Hello and welcome back. Very excited to have Synthetech MR with us here for the next presentation and also the last presentation of the Life Science Summit of 2022. And with us today we have Ulrik Harrison to present the company and how you're innovating the MRI space. But I'll leave the floor over to you right away.
Thank you very much, and thank you for letting us have the opportunity to present Synthetech MR in this Life Science Summit. Next slide, please. My name is Ulrik Harrison. I've been now with the company since mid of 2019. Joined Synthetech MR from Hermes Medical Solution, another company within medical imaging. And have throughout the years been in different kind of international commercial roles within companies like Pfizer, HemoQ, and Danner Corporation. Next slide, please. So today I will have the opportunity to address to you some details around the company. That is a leading provider of innovative software solutions within the MRI area. We will go more into details on the fact that we have today existing partnerships and collaborations with the bigger part of the MRI OEM businesses in the world. We will also be addressing some information about the changes that were made just recently. For example, implementing a new business model with a recurring revenue solution setup. And also discuss a little bit more on the possibilities we have in order to continue to grow and where to find opportunities to implement our and accelerate our growth initiatives. Next slide, please. So as I said, we are an imaging software solution that we develop and sell that is supporting a faster workflow and improve patient care within the MRI and in the healthcare. We have a very clear vision on that we empower clinicians with efficiency and confidence to improve patients' outcome. Important to mention is that parts of the mission is that we will make our quantitative imaging solution as standard of care, and by that support a more precise diagnosis, reliable monitoring, and personalized treatment strategies. We are today located in Linköping in Sweden, where actually Marcel Vanches founded the company and started up in 2007. And looking into the next slide, please. And I would like to share with you some details here on this timeline. As I said, Marcel Vanches founded the company. We are very happy that we still have the innovator of the solution in the company as a board member, as one of the key people in our innovation strategies and work, being the head of innovation. Looking at the first part, you can say, or the first few years of the company history, on the left-hand side on this scale, you can see that we initially started to work with a company that's named Sectra, a company that is active within, you can say hospital, radiology, picture, and imaging and archiving solution platforms. Whereas Samurai, this is the name of our solution, is being used. We also, back in 14, 15, and 16, signed more general collaboration agreements with Philips Healthcare and Siemens Healthineers. But I would say that the major milestone that we reached early in the early phase of the history so far is back in 2014, when we signed a global license and distribution agreement with GE Healthcare that definitely supported our efforts to really build into and starting to establish SciMRI as a vital part in the healthcare. As I said, I joined in the mid of 2019, and by that time we actually identified and started to really forcefully implement, accelerate some key initiatives, key growth initiatives. One of those is that we by then started to implementing and investing in our own sales force, our own sales resources in some key countries around the world. The US, we started in India, in Japan, as well as also in some parts of EU. It is obviously very important to us to be there with our own sales resources, both in order to have our direct sales to the hospitals. It's also, of course, very important to us to have the direct contact with the healthcare providers, with the healthcare in general, you can say, and also to kind of support back information, voice of customer into the headquarter organization. But not at least, it's very important to us in our ambition to continue to develop and strengthen our collaborations with the different OEMs, to be able to also work with their local sales organizations. So that was one of the first things that we did and spent a lot of time in doing that, mainly the second half of 2019. What we also have been doing now during these years from 19, 20 and 21 and actually up until today is that we now have continued to find ways to strengthen the collaborations with both the existing but also with new OEM partners. What we have managed to do now is to have an even stronger global distribution and license agreement in place with GE Healthcare. They are now having the full functionality of SciMRI selling in their global sales organization. The same thing, definitely a strong leap that we took when we, during last year, signed new agreements, both with Philips Healthcare and Siemens as well, being on par with the one that we have with GE Healthcare, meaning that we now also have these global license and distribution agreements with them as well. During 2020, we also added to our partnership another PACS company as a sector, and that is Fujifilm, having a very strong market share in Asia and mainly Japan, of course. And not at least, we now also signed, actually last year, signed a new product development agreement with Canon Medical. I will come back to that a little bit later on, as well as also adding a fourth MR scanner OEM manufacturer, and that is United Imaging. That is a big player in China with very clear global ambition. We came from the very important first agreement with GE Healthcare, having a business model in place or a pricing model in place that were kind of focusing on perpetual licenses, meaning that we had one sale of a license and that then were in use for the full time of the lifetime of an MR scanner, which is around 10 years. What we have been doing now for the last 3 years is that we have implemented, I would say, only time-limited licenses. It could be 2 or 3 or 4 years, but in most cases, the customers decide to buy a 3-year license, and in general, for only a 3-year license to a higher price than what we had before with the perpetual license. And as I mentioned, just a lifetime of a scanner is 10 years, which means that, in theory, you can actually sell the same license 3 times, and each time to a higher number than actually for the full that we had before. That is also something that we have been focusing a lot in implementing as well. And obviously now we have, with these 4 different GE, Philips, Siemens, and United Imaging, we have together with our own direct sales force a really strong muscle in terms of sales, sales resources, sales organization on a global level, working and promoting and selling SciMRI. Next slide, please. So the MRI market today is heavily consolidated on a few OEMs. Looking at the right-hand side here, it's concluded that around $5 billion is the value of the MRI market. Of course, with all the scanners that are pretty expensive, that is the most of the cost related to that. Important to say is that the market grows around 5 to 7% on a yearly basis. That means, of course, that they are including a change of worn out MR scanners, but actually also there are being more scanners implemented into the market on a general basis. So that means that today we say that around 40,000 systems are out there on a global level, and obviously then that number will increasing by every year. Around 5,000 new systems are implemented into the market every year. And usually we say that around 50% of those 5,000 systems are seen as an addressable market for us, meaning that our solution is then compatible to those different camera versions or scanner versions. And I didn't mention it, but I usually say that the price for a 3-year license is in the interval between $50,000 and $100,000. You can see here how big of a market share the different players has. And you can also then conclude with the also product development collaboration that we now have with Kernel Medical Systems. We are today covering more than 95% of the global market within MRI. Next slide, please. So there are some clear challenges in the radiology, in the hospital, in the healthcare, in the radiology today. On the left-hand side, you can see some examples of chronic diseases that is, having people affected on a global level that we can see are increasing in this case. And in all these situations, using or having an acquisition of an MR scan being made is relevant, both in terms of making a diagnosis, but also in terms of, looking at progression of a disease or monitoring a treatment that has been implemented. So you could say that the number of MRI scans, they are increasing due to this fact. It is being used more and more, as I also mentioned before, because the market is growing on a global level. And as it is in many different areas, of course, there are also a higher increased pressure cost. So this means that there is an increased pressure on radiologists, healthcare in general. And it is an even a need for a more efficient workflow, something that kind of supporting that, as well as also the demand for having objective assessment tools is being higher as well. And with our solution, and taking the next slide, please, we actually address those different needs from the healthcare and from radiology and from the clinicians that refer the patients to radiology. As I said, the name of our solution is SciMRI. This is SciMRI actually being performed on the brain, which is SciMRI Neuro. A faster workflow, you can, with our solution, have multiple grayscale images in only one go. I will come back to that a little bit later. Of course, with having examination being done in only 5 minutes, compared to maybe 20-25 in using the conventional imaging, that is also supporting a much better patient experience, as one example. Lower cost, increased throughput as well, addressing the faster workflow and need for that. Our technology aid the clinician in order for being able to make an improved diagnosis, and as I said, also monitoring treatment and follow up. we are providing with an objective measurement rather than a subjective assessment. And I will come back to what that means a little bit more in details. And please look into the next slide there. So here you see an example of how objective assessment will look like. So up left in this slide, you see two different grayscale images, as we are used to see them since decades back when MRI was being implemented into healthcare. To the left, we see a brain and an image from a brain from a healthy person. And to the right, you can see that there's an image from a patient suffering from multiple sclerosis or MS. Doing as it is used to be done and still is being done, obviously, you are doing a visual inspection, and we can see here that maybe there is some differences if you're looking on the two different images. Maybe it is the fact that the brain looks a little bit smaller to the right, but also that you could see that the in the middle of this image that these kind of black parts are a little bit bigger actually. Doing this kind of visual inspection is a subjective assessment using these grayscale images. Bottom left, using SciMRI in only 5 minutes, as said. What our technology is doing is to measuring the absolute properties of the brain, meaning providing with a numeric value for each and one of the small pixel or voxel in this case. That means using these metrics, you can do a lot of things. For example, you can be able to do automated volume segmentations. In this case, you have been doing segmentation on a vital part in the brain that is called myelin. That is a biomarker that is supporting the speed of how a neurosignal is actually moving in the brain. And you can see here on the left-hand side, the same healthy person, right-hand side, we have the MS patient, that there is less of green in the right. And that means that the level of myelin is lowered only by using the visual inspection. So that is a support to the visual inspection that still needs to be done. But using the measurements, as I mentioned, you can see the exact value of myelin in this specific case of a healthy person, 178 milliliters compared to 126 for the patient suffering from MS. And you can also see the fact that the brain is smaller in relation to the total volume of the skull. That is what BPF stands for. So with a healthy person, 88.5%, and then you have the MS patient, 83%. Moving on, please. Quickly, another example in this case. So you see again, this is a 3-month-old boy that has been referred to MRI. You can see here the first scan to the left, and then we have a follow-up 5 months later. You can see that there are differences, of course, in this case, and that is because the volume of cerebrospinal fluid, or brain fluid, actually, you can say, is higher after 5 months than in the beginning. And that is because this little boy suffers from macrocephaly. Again, it is challenging, it is time-consuming doing this visual inspection, and it relies on a subjective assessment. Using SciMRI, and looking into the next slide, please, you can see the same. And that means that top left, if you look at the right-hand side here, and you see the graphs, but starting to look at the different images, top, you can see the same that we saw before, and then you have a little bit bluish color. That means that we have used segmentation looking to cerebrospinal fluid. So in this case, we have support for the visual inspection. You can look at the first scan up to the left, both of them, and then the follow-up. But in the different diagrams, you can see the gray shaded area. That is actually a population of healthy individuals. And the yellow dot is the value for this specific patient. So that means that you can also see in the heading on the different graphs that for The first scan is yellow dot shows it is outside of the healthy group, so to say, and the exact number is 178.2. And the same goes for, and you can also see the change after the 5 months. And this is exactly why our different OEM partners now wanted to come to us and starting to promote and sell this solution into the healthcare. Next slide, please. So today, based on the fact that we are going to be and have the ambition to be standard of care, we are now having many highly reputable hospitals around the world that is using our solution in different countries. You have Karolinska sjukhuset in Sweden, for example, Mayo Clinic in the US, as well as also MD Anderson Cancer Center using this in their clinical workflow. Next slide, please. So based on what we now have built, based on the momentum that we're having, and based on this strong partnership that we have built and continue to build, we continue to implement those key initiatives to support for future growth. For example, the pricing model, as I mentioned before, we continue to find ways to work together with different partners, global, new, existing ones, and so on and so forth. We will also continue to implement and investing in implementing our own sales resources in our existing markets. We have proof that they are the right ones, but also into new ones as well. For example, South Korea, China, Brazil, Southeast Asia. And we are also putting a lot of focus in to continue to build our product roadmap. And next slide, please. In this case, we are moving from what we have today in the marketplace and what's also our different collaborations are based on, that I mentioned before, the sales and promotion agreements with our different OEM partners. That is relating to the so-called 2D solution that we have to the left-hand side here. So I mentioned the different kind of parameters related to them in terms of faster workflow, objective assessment, and so on and so forth. But we now have, I would say, deep collaboration, as I mentioned, with Canon, and also with the other OEMs as well. And a specific one of those that I cannot mention right now, we have come far in the development of this joint product of a 3D solution. And what does it mean? Well, the images that I showed before shows actually what you can have acquired an image from only in one plane. But imagine when you have the same, let's say, a lesion. I mean, let's say that you are kind of suspecting that there might be some oncology change, some kind of, let's say, a tumor. If you are able to see that not only from one plane, but also from the side and from the back, will add so much more value to the radiologist and by that also to the referring clinician. So that is something that we are very excited for and investing a lot of money and time into continue to work in order to bring to the market as soon as possible. So having addressed one example on the different key growth initiatives that we continue to implementing, by looking at the next slide, please, I'm open to take some questions.
Perfect. Thank you very much, Ulrik. This is very interesting for sure. And we're a bit short on time, but I'm going to squeeze in 2 questions either way. And as you've said, you cover most of the major OEMs, and 95% of the market right now with the partnership. But I believe the attachment rate is roughly 25%. Please correct me if I'm wrong. But what needs to happen in order for that to move towards, say, 50-70% attachment rate to new MRIs?
I would say that actually the attachment rate at this point is a little bit lower, to be honest. And I say that based on the fact that we see a great potential for our solution to be implemented and sold. Well, what needs to be done? Now we have a lot in place. We have built a momentum in terms of working together with both, well, with the OEMs, of course, but also with our own sales resources. And we have many key customers right now as references. So it is about executing. That is what it's all about right now. And that is what we continue to do very forcefully.
Okay. And I also believe that the sort of revenue share in this partnership are quite generous at this time. I've heard somewhere between 50-50 split or something in the range of that. But maybe that's on the low end now to sort of incentivize and drive adoption of this software. But thinking about future iterations, like the 3D product that you talked about, do you think that you will be able to sort of keep a larger share of that?
You know, I can, as I mentioned before, and compare the price that we had in the earlier agreements and to what we are selling for now. It shows that, of course, that is the ambition, and we are able also to execute on having a higher price and value into the company. And yes, you're right. That is, of course, the ambition. And one, clear milestone that we will reach that will give an opportunity to actually continue to execute that will be when we are launching the 3D solution pretty soon.
Okay. Thank you very much, Ulrik. In the interest of time, I'll need to limit myself to those questions. But I really appreciate you being a part of this. And also thank you for everyone, to everyone listening in to the Life Science Summit as well. This has been the last presentation of today. So just want to thank everyone for participating.
Thank you.